Great. I think we're going to get started. If everyone could take their seats. Mic check. Can anybody hear me at the back? Okay? Yes. Great. Fantastic. Okay. So in the interest of keeping us on time, I'm going to get started. First of all, I want to thank you all, everybody, for being here. This is a great turnout. And it's going to be the start of a, I think, wonderful day. I wanted to thank not only all of you for attending, and obviously Peter. Where is Peter? Peter.
There are two of us.
That's right, Peter and Peter. But I also want to thank the rest of the local organizing committee who helped organize this. Derek Manzello, Ian Enochs, Bernhard Riegl. Couldn't have done it without them. So with no further ado, I'm going to pass this over to Roni Avissar, the Dean of the Rosenstiel School, for a note of welcome. Thanks.
Thanks, Andrew. And let me join Andrew to thank everybody for being here, and especially also the organizing committee of Andrew, Derek, Jan, and Bernhard that put that together. It's a great idea. It's always with mixed emotions. That we say goodbye to one of our faculty that after a brilliant career is leaving us. I mean, Peter has decided not to leave us completely. He's still coming, and in fact, okay, I think that he's kind of a role model for a lot of people that notice that even after retirement, 4 years after retirement, the light in his office is still on, and we still see him quite often at the school. It's a true pleasure from that point of view to continue to share the opportunity to work with Peter. As many of you, if not all of you, know probably better than I do, because I've been around only for about a decade, but you have seen Peter's and you have known Peter for many, many years. He has been around for a while. He started his career getting a Ph.D. in 1963. So that's before I was born. But I wished. But he has been around also in terms of institutions, okay? He has provided expertise in the field that is now a very well-developed field, both at the Scripps Institute, at UCLA, at Stanford, and then tired of California and the cold weather, he decided to move to more reasonable place like Puerto Rico, and of course the Smithsonian Tropical Research, as well as being with Rosenstiel since 1984, so for the past 36 years. Which I think that the University of Miami has tremendously benefited from Peter's insights. And all of you in the room know that even better than I do. Peter has continued publishing over the years, including after retirement. And we are going to hear today some of the work that he has been doing over the years with some of you. So without any further delay, I want again to thank the organizing committee, thank Peter for a brilliant career and brilliant contributions to the Rosenstiel School, and thank again all of you and the organizing committee for putting that together. I look forward to seeing you again this evening. Unfortunately, I am not going to be able to spend the day here, but I look forward to seeing you this evening as well. So thank you very much, and have fun, and see you this evening. Thanks.
Thanks, Roni, and thanks again to all of you for attending. So today is going to be, I think, truly fantastic. It's great to see so many familiar faces in the audience, and again, thank you all so much for traveling from wherever you traveled from. We're here to celebrate 8 decades of marine science and exploration on coral reefs with Peter Glynn. And I hear you say 8 decades? How can that possibly be the case? And so we're stretching the truth a little bit here, because if any of you read the tribute that's available online, Peter's first foray into the world of coral reefs began in 1959 when he hitched a ride aboard a Mexican fishing vessel to go and explore, I think, the Belizean Barrier Reef. So if you count the 1950s as a decade in which he worked and stretch it through to the 2020s, you'll find there's 8 decades there. So that's where that comes from. You know, I should also mention this is not the first time that we've tried to organize a retirement symposium for Peter Glynn. Some of you might remember a failed attempt to do so in 2008 where I heard rumors that Peter was retiring, started to organize, and then Peter quietly pulled me to one side and said, rumors of my retirement are somewhat advanced, and we decided to pull the plug on that one. So I'm very pleased because we got another 10 years or more of Peter. And in fact, Peter's been retired officially for 3 years, but as Roni just mentioned, many of us don't actually know that because Peter's still here, he's still working with undergraduate students, he's still publishing prolifically, and I don't anticipate that this retirement symposium is going to change any of that. So please don't feel that this is a farewell to Peter. This is just something to mark the occasion as we needed to do, and I'm just glad that we were able to do so. So for my own remarks, I find myself somewhat unprepared actually, but I wanted to touch on my 4 decades with Peter Glynn. Again, 4 decades following the same logic I just explained. I'm not that old. And just point out that, you know, many of us, and I think you'll hear great talks today from Peter's field explorations, touching on various aspects that you'll see here. Thanks to everyone who contributed photos. I'm shamelessly using them here. But my first interaction with Peter came when I came from Britain as a graduate student on a Fulbright scholarship to come and work with Peter from the United Kingdom. And right about the time I started, which was August 1993, this paper had been published in the May of that year by Bob Budemeyer and Daphne Foten, a very controversial paper at the time it was published. One might say it's still a controversial paper today, but it really captured my imagination as a graduate student. And, you know, Peter being Mr. Coral Bleaching, I, of course, wanted to talk to him about this paper, get his advice, and although Peter, like many people, was somewhat dubious that there was necessarily the evidence there for it at the time, he was incredibly encouraging of me to go and pursue ideas that I thought were interesting and to find the right people to help me in that endeavor. And he encouraged me to go to the Smithsonian Tropical Research Institute, which I'd been with him on a couple of cruises that he was doing at the time with his graduate students and colleagues. Met other scientists at STRI, Nancy Knowlton, Rob Rowan, who were working on this. And as a result of that, was able to do some of the first work sort of testing this idea in the tropical Eastern Pacific on some of Peter's very reefs. And there are many tales to tell about this, but it culminated in a paper in Bulletin of Marine Science, which of course is RSMAS's own marine science journal, Glynn 2001, which showed for the very first time, in fact, that there were algal symbionts inside corals, the possession of which imparted bleaching resistance to corals, and that these symbionts were in a clade that at the time was coined clade D, and now has been changed to durastinium. And this was the first paper to show that clade D was bleaching resistant. And we turned that into a more in-depth study over time, looking at the transitions in symbionts and the shuffling of symbionts over time, and the Panama data set was included in this. And really, I think Peter had a hand in helping sort of begin this whole field, which has really become quite a burgeoning field of research at this point. I have to point out that some of you may have heard that there was a report recently published from the National Academy of Sciences looking to investigate and test ways of intervening to increase the resistance, the resilience and persistence of coral reefs, in particular to climate change and in particular to bleaching. And a number of experts were convened to sort of advise on that. And the reason I bring this up is that there are fully 3 people, former Peter students that were on that committee, Tyler and myself and Bob Richmond, who sadly isn't here today but sends his greetings. So fully, 25% of that committee were from Peter's legacy, and I think there's something to be said about that. And as I mentioned before, this has developed into a field that I think is going to command much of the research agenda for the next decade, with the results of that study being published, and now a lot of interest in testing interventions through a variety of methods to try to increase the bleaching tolerance of corals on reefs around the world through a variety of different mechanisms. And so I think, that all sort of lays the groundwork that Peter's tentacles or mesenterial filaments have stretched across the ages and across fields. But I also have to say that my own personal experience of Peter's is maybe slightly different from many of you here that have a lot of experience of Peter in the field. Most of my experiences with Peter have been, more prosaic, more here at RSMAS in the office or in the lab. And of the almost 7 years that I've been in the US, 21 of those have been in Miami and at RSMAS. And so I've overlapped with Peter a lot. And the lessons that I've learned from Peter, I think, are not necessarily the field-based lessons, but the lessons in conduct, scientific conduct, I suppose. And I have, for one, I'm sure many of you share this, benefited greatly from Peter's advice and interactions. And for me, he sort of reminds me of another time, a time that we don't normally have perhaps access to in today's day and age, where it seems like we're all getting busier and busier with less time to talk about scientific ideas of interest and less time to sort of smell the roses and ponder out loud. And Peter really, for me, epitomizes that approach to science, which I still think is incredibly valuable. And I've put this up here. James Dana is a sort of a hero of Peter's, and in fact, this photo is outside his office. And although James Dana was famously wrong in claiming that there were no significant reef buildups in the tropical Eastern Pacific, nevertheless, he was a tremendous contributor to coral reef science. And this sort of reminds me of Peter's attitude towards science and that, I suppose, gentlemanly attitude towards science. And in many ways, I think of Peter as a coral maker, as a maker of corals. And by that, I mean that he inspired an interest in corals, a fact-based interest, which if you read that paragraph there is important, but which for me at least never dismissed the mystery of corals. And I think that's the attraction for many of us in the room. These corals and coral reefs are mysterious places. And I suppose in tribute to Peter, you guys will probably have just been distributed this souvenir bookmark, which I made. Hopefully you all have one. And that's the purpose of this little tribute to Peter. I called it Corals and Courtesies. And for me, this little illustration, if you can make out in the lower right, just below Peter's name, depending on your perspective, these are either coral polyps or diadema. And the little diadema that's wandering off near the word Glynn is Peter. And I think of this as Peter the polyp, wandering off from the rest of us as he pursues the rest of his career. Some of us are stretching out our tentacles, trying to get him back. Or maybe if they're diadema, they're our spines. But I hope you'll take this with you and think of Peter whenever you next read a book, which some of us still do. Again, I just wanted to thank you all for coming. Thank Peter for his tremendous contributions to the field, and looking forward to a wonderful day. Okay, I'm now going to transition to a few housekeeping remarks. So we have a lot of speakers, and people have a lot to say. We've got some tremendous PowerPoints, but in order to keep us on time, I've created some little reminders for speakers. So the different session chairs will be seated at the front here and will prominently wave these signs at you that say 3 minutes, 1 minute, and time's up. So please respect the signage if you can before we have to stand up and sort of wiggle you off the stage. If you haven't got one already, please pick up a name tag, also a badge, a metal badge, as well as your souvenir bookmark. If you don't have a name tag, there are blank name tags at the back. Please, I encourage you to use them and write your name on them. One other thing, many of you are attending tonight's banquet. If you're attending the banquet and you're sort of paid up, you should have a little dinner plate icon on your little badge. If you think you've paid and you don't have an icon, please let me know. Can I also tell you, every one of you who has such an icon on their badge, to please, at some point today, go to the back and pick up your wristband, which will give you free drinks tonight. So don't forget to do that. I think that's it in terms of housekeeping. We have a coffee break coming up, and there are going to be 4 different sessions, as you can see from the agenda. And now it's my honor and pleasure to introduce the first speaker of the day, who is actually a plenary speaker. We have 2 plenary speakers, Jorge Cortés right now, and Peter J. Glynn, Peter's son, this afternoon who have extra time to tell us their insights into Peter. So I welcome Jorge Cortés from the University of Costa Rica to the stage. Thank you.
It's a great honor to be here today. It's a real pleasure to see you, Peter. And Andrew, thank you very much for this invitation to give this plenary talk, and all of you for being here. When Andrew invited me, I suggested the title, Peter Glynn's Influence in Marine Science in Latin America. But when he mentioned that I was going to give a plenary, not just a 50-minute talk tape, I've been thinking more about it and expanded it a little bit. So I'll talk a little bit about his impact on marine science, which a few things have been mentioned, and many will be mentioned today. His Latin American connection, and then finally the topic of my talk. So as mentioned, he did his PhD in 1963 at Stanford, but before that, he was in South Dakota, and way up north, and then, fortunately, he moved to the coast and started looking at the sea. He went to Puerto Rico from 1960 to 1967, and that's where he started looking at reefs. And then, in my opinion, it was fundamental, his move to Panama, to Smithsonian Tropical Research Institute, because that's when he started with the Eastern Pacific, which is what most of us know him for and have been working on. And then in 1984, he moved up here to RSMAS, which was, for me, was very fortunate because the next year I came to do my PhD with him here at RSMAS. And he started very early on publishing. His first paper is from the 1960s. And it's a very interesting title, A Class Exercise on Marine Microcosmos, published in Ecology. And during the 1960s, he did a lot of work on the marine intertidal in Monterey Bay in California. That's where he did his PhD. And there's a whole series of papers on the intertidal organisms. And from the very beginning, you can see the detail of his work with the different groups looking at the individual organisms, looking at the communities, and how they all interact. His first coral reef paper is from Puerto Rico in 1964, and that was the impact of this hurricane in La Parguera. And then, well, in Puerto Rico, and when he moved to Panama, he published a series of papers in the intertidal zone. That was his first connection, so he did all these papers on the intertidal zone. But then from 1972 on, he started publishing his Panama papers. And that's when we start seeing all these seminal papers from the Eastern Pacific. And most of us know him for his work in the Eastern Pacific, but he was a taxonomist, a naturalist, an ecologist, a geologist, a historian, and of course, a great mentor. And as Andrew said, he's done so many different things and worked with so many people from all over the world. And maybe many of you don't know, but he was a taxonomist. An isopod taxonomist. He described many species. This one is one from Venezuela on the left. And then this is a very important handbook on the isopods from Puerto Rico. And I was surprised when I saw his first papers on isopods. And many of these species still stand, which for taxonomists, that's very important. And we know him mostly as a geologist and ecologist, but he was a geologist at heart. And this is really in Golfo Dulce, Costa Rica, for my PhD, with his good friend Ian McIntyre, with whom they did a lot. They had a lot of adventures all over, not only the Eastern Pacific, but in the Caribbean, drilling, going to the reef, drilling, and figuring out the history of those reefs and how they change over time, and let us understand what is going on right now with all those reefs. And that was the routine, just all day drilling and then sending them out in the boxes, and then a couple years to analyze all that and come up with some story. He started exploring the Eastern Pacific in Panama when he moved to the Tropical Research Institute, and I should mention James Porter, who did a pre-doctoral fellowship as Tri, and also I don't know if there was like sort of competition, because there's a paper, similar paper by both of them from Panama, the comparison of both sides, the reefs on both sides of Panama. And that's the first time that we realized that the reefs are very different. In fact, it's the first time that we realized that there are reefs in the Eastern Pacific. All the classical papers by Darwin say there's no coral reefs in the Eastern Pacific. And it's these papers from the 1970s that start putting all these reefs in the map. And now we know that there are big reef areas in the Eastern Pacific, and some of these have been there for thousands of years. In Panama, for 5,600 years, there have been reefs growing there in Panama. And the list of colleagues and students working in Panama, it's very long. And that's what we'll be hearing during the day today. Many of his students and colleagues that have done work with the coral reefs, mainly in the Panama area. But he moved into other. Panama became too small for him, so he started moving around Latin America. And his first publication was in Colombia with Henry von Prahl in the 1970s. And then he told me an interesting story. In the 1970s, he met this pilot from the Panama Canal, and that had a plane, so he convinced him to just fly around Central America, taking pictures, flying low, taking pictures. And at that time, it wasn't that problematic just to move into another country, take pictures, and go back to Panama. So that's when he started taking pictures, and he took some pictures from Costa Rica that intrigued him. So he went back to Costa Rica and published a very, very interesting paper. And it's the impact, possible link with the Little Ice Age with Ruffle and Dunbar. And what's innovative of this paper is that they combine the dating of the reefs plus the use of stable isotopes. And what they find is that most of these reefs in the northern part of Costa Rica stopped growing about 300, 400 years ago. And the signal in the skeletons is of cold water. And this is, from what I know, is like first evidence of little ice age in reefs in the tropics. And then he went back to Costa Rica again. He did a lot of work with Jerry Wellington. And I know Jerry because his first work was in Costa Rica as a Peace Corps member. He went to Costa Rica in 1970, and the first work on reefs on Costa Rica was done by Jerry, and it's a manuscript, and I talked with him many times that he should publish that, but he was too busy with all his work, and never got around to publish it, but that's why I have this closeness with Jerry. And Jerry and Peter got together and worked a lot in Galapagos and came up with this famous book, Corals and Coral Reefs of the Galapagos Islands, which I'm sure every one of us have used, worn out. And the important contribution was including John Wells, and that was like the first time someone put all the corals from the Eastern Pacific together, and we can start identifying some of those corals that we're looking at in the field. And in Galapagos, he also worked quite a bit with Fernando Rivera and Patricia Martinez from from Ecuador. And in this picture, this is 2016 in Honolulu with Bob Richmond that had been mentioned here. And I met Bob in Panama when he was doing a postdoc with Peter, looking at coral reproduction in the Eastern Pacific, and that started off a whole series of coral reproduction papers from that region, which is an uncommon region because the corals there are not reproducing sexually as much as other regions. Most is fragmentation, and we see all the gonad development, but very, very little recruits of corals. And of course, that's a problem for the recovery of reefs in this region. And he went back to Costa Rica in 1988, and that's where we did my work for my dissertation in the Urvina, which probably some of you remembered from STRI. That was the research vessel from STRI. And this is in Golfo Dulce, going out to the field to drill. It was a great experience together with Ian. Just spent days and days drilling and tide there 3 meters, and we start in the reef flat, so we in low tide, and then tide will start coming up, so we have to put tanks and keep on, start working underwater, and then the tide will go down again, and by the afternoon we were outside the water again. And one anecdote was that one evening Ian is making the notes about the dive. He said, "Do you realize we dived for 8 hours today?" The dive officer will never believe this, but it was dive like that for 8 hours a day. And then he did work in Mexico also with Gerardo Leite Morales from the Universidad del Mar, and they published a paper the first paper in 1996, and then a series of papers. And what's interesting of this paper is Huatulco is in this region. This is the Tehuantepec Strait, and it's one of the seasonal upwelling areas in Mesoamerica. Here in southern Mexico, southern Nicaragua and Costa Rica is the other area, the Papagayo upwelling, and then in the Bay of Panama. And that was a whole other area of research that he developed, and it was the influence of upwelling on reefs in that region. And afterwards, there have been a whole series of papers related to those topics. And he also went to Chile. And the Eastern Island, or Rapa Nui, there are reefs there. There was a previous paper from people from Chile, and he went there and he worked with Yves Wieters and Sergio Navarrete, and he went there with Jerry Wellington in 1999 and 2000. And these publications really put the reefs in Chile on the map. And it's interesting with my Chilean colleagues, I talked to them about the reefs in Chile, and they would say, no, there are no reefs in Chile. And I said, yeah, check out Rapa Nui and Peter's paper. There are reefs, very interesting reefs in this island. And that culminated in this monumental work, Coral Reefs of the Eastern Tropical Pacific, which I was very happy when he started editing this book that was going to put everything together. 22 chapters, 657 pages, 77 authors from 11 countries, and 37 of those authors are from Latin America. So I think it's become obvious his connection with Latin America, his work with people from most of the countries in Latin America. And now he's going back, natural history and basic ecology. And his latest publications are with benthic ctenophores, which at least for me, was ctenophores are these jellyfins that float in the water. And it turns out that there are some that live on the benthos. And he's published a whole series of papers on the benthic ctenophores right off here. And I see that as going back to the basic natural history and ecology. And as Andrew said, we need to do more natural history. That's for me, that's the basis of all the other sophisticated science that we're doing. But that was my title, Influence on Marine Science in Latin America. And I think with the mention of all these people that he touched on in Latin America, you could see the connection. But his connection goes way back. And his connection was with Carmen from Puerto Rico. And I guess that's where he learned Spanish. And then moved to Puerto Rico and eventually to Panama. And that, I see that as his first Latin American connection. But he visited most Latin American countries with coasts in the Eastern Tropical Pacific, at least every country that had large coral reefs. He didn't go to El Salvador, as far as I know, but those are very small reefs. But Mexico, Costa Rica, Panama, Colombia, Ecuador, Chile, he did work in all those areas. And he worked with local researchers. And that's something important for us. And in Latin America, many times we're very much disconnected from the rest. And we see a lot of scientists coming in from first world countries, go to our countries, do research, leave, and we find out that they were there when we see the publications. Peter got in touch with people from every country, did work with many, many people, and many of us ended up being his students up here at RSMAS, but others that were not even his students were connected with him. And he's the mentor of a large number of Latin American students and researchers. And to finish, Andrew sent me this photo. I had it somewhere, but I couldn't find it. And you'll recognize some of the people there, Josh, Mark. And that's only 20 years ago. It's incredible. But here we see Nohora Galvis from Colombia, Juan Maté from Panama, that's me. Hector Reyes Bonilla, which was Peter's student from Mexico. Fernando Rivera, and Hector Guzman from STRI. And plus, of course, many other people there, Andrew and many other people. And so he really has been a powerful influence in the region, and very proud to have been his student. And even some of my students wanted to come because they say that he's their academic grandfather. So many of them, Juan Jose, send his regards, and Odalisca and Celeste send his regards. My students now working on reefs, looking at deeper details of those reefs. Thank you very much, Dr. Glynn.
So we have a few moments for questions, and actually this reminds me of something I forgot to mention. So we are live streaming, so hopefully if you didn't get the link and have sent that to your friends and colleagues so they can see it. But it does mean that if we do have questions, if you could please use the mics, and so we're going to have mic runners coming up and down the aisle so that we can capture your question for the live stream too. Are there any questions for Jorge? For you immortalized on the live stream. The live stream's being recorded too, by the way. I should just mention that. Another little housekeeping point of notice is that can the next speaker, who of course is Mark Eakin, please come up in advance so that you can get mic'd up and we can do the transitions really quickly. Questions for Jorge? No? Okay. Thank you very much.
Well, thank you very much for having me here, and it's great to be here speaking at the beginning. Especially Jorge and I have this ongoing discussion about who was Peter's first doctoral student. I was here first. He just got out first. Anyway, it's great to be here, and really the appreciation we all have for you, Peter, and what you have done for coral reef science is just to the depth of all of our souls. And I'm going to tell just one little story about a piece of this and sort of where Peter became involved in a major area and what I've been doing now for 15 years at Coral Reef Watch. And that was Peter's role in the rise of coral bleaching, global coral bleaching. Now, I'm not going to imply that he's at fault. We all know correlation is not causation, but Peter was there at the very beginning. So what's going on now is, of course, we all know about this problem of coral bleaching that's going on and is now the biggest threat to coral reefs around the world as climate change continues to get worse and worse. And right now, in fact, we're looking at an event going on in the Great Barrier Reef, the third major bleaching event in less than 5 years. The heat stress started building in the north, it's spread on down through the south, and is intensifying. We have reports of bleaching all the way from Heron Island in the south up to Lizard Island in the north and points in between. And this is, of course, from our Coral Reef Watch products, an animation of the last month. If we look at the climate model-based forecast that we do, this is what it looks like for the coming month. We're looking at a tremendous amount of heating going on. Hopefully it will not be as intense as what we saw in 2016 and 2017, but more widespread than either of those events, which is quite unfortunate. And all of this was a part of this global bleaching event that happened from 2014 to 2017, the biggest global bleaching event we've ever had, the first time that a global bleaching event has gone from not just for 1 year, but for 3 years in length, from May of 2014 to, or June of 2014 to May of 2017. But we're not here to talk about what's going on now. We're here to talk about where it started. Peter Glynn and the work that he was doing, the pioneering work that we heard about from Jorge in the Eastern Tropical Pacific, in particular the work that he was doing in Panama and then very quickly after in Costa Rica and Galapagos with the first big bleaching event. 18 March 1983, Peter was on a trip going out looking at the reefs at Uva Island in Panama. Small reef, many of us have been to. It's a little bitty reef on a little bitty island, a dot north of Coiba that is hard to spot on the maps. But it was there that he went in, and the reefs that he normally saw that were these beautiful fields of post-saloprid corals intermixed with various massives had changed. And there was a tremendous amount of bleaching going on. And later, the bleaching was extreme. Some of the corals were dying. And this led to the first paper on this event, part of the 1982-83 El Niño's impact on the Eastern Tropical Pacific, and the part of the first really widespread bleaching event that ever happened. Peter found that not only was it happening at that little reef in Uva, but it was happening throughout the Gulf of Chiriquí, throughout the Gulf of Panama, and had reports coming in from colleagues in the Galapagos that was happening down there. But at that point, being the consummate and careful scientist that Peter was, the paper that he wrote talks about possible causes, the copper mine project, dam construction, transshipment of oil, pesticide use, extreme midday exposures, an El Nino event that was going on involving anomalous warm temperatures, a strong earthquake that had been in the area, and finally, no causative agent can be identified at this time. It's something we forget, that it's easy to jump to conclusions, and that's something that Peter avoided. And that's a very important part of what he did and that we'll be talking about. Of course, not long after that event, Peter moved from Panama to here in Miami. And Miami was one of the places that were innovators of satellite remote sensing. And some of the early data from the satellites, and the data that we have for good sea surface temperature data only go back to 1981. 84, depending on where you call it good at that point. But what he was able to find working with folks here at RSMAS was that indeed there was a big heat mass, massive warm water, early marine heat wave report as a part of that 1982-83 El Nino that affected that whole area across the Eastern Pacific. Here you can see 1983 on the left, normal conditions in 84 on the right with the upwelling going on off the coast of South America. So at this point, the identification is clear that this heat stress was going on. But Peter, again, causation is not the same as correlation. So you've got to know what's going on. And so Peter wanted to do an experiment, and I was here as a student of Peter's during the time of this. And he went for an NSF proposal. And a part of that NSF proposal, Peter was going to go down to Panama, was going to take the corals from the eastern, from Gulf of Cherokee and from Gulf of Panama, was going to expose them to high temperatures and prove that could be the cause of that bleaching event. At least one of his reviewers said, there's no need to run that experiment. Everybody knows that's true. Peter ignored NSF's instructions that that wasn't something that needed to be done as a part of that. Published this paper comparing corals from Gulf of Panama and Cherokee at 4 different temperature regimes. We won't go into the axes and the various chlorophyll and zooxanthellae counts and things, but what you can see is higher temperatures, everything starts crashing. And Peter was able to demonstrate, without a shadow of a doubt, that those high temperatures were a causative agent behind this. So despite the reviewer who said, you know, nobody needs to do that, some people thought this was pretty good because this was the best paper award in the coral reefs for that year. Well, at the same time here in Miami, there were some other folks who were starting to look into these and look at these early records of satellite oceanography. And Don Atwood, who was across the street at AOML with Jim Hindy and Antonio Mendez, did some work looking at a whole series of time series. And what was going on then in the 80s were all of these bleaching events in the Caribbean, the early Caribbean bleaching events. And looked at the time series of temperatures, compared those to what was believed to be about the temperature where bleaching was going on, and started looking at something, the early analysis of anomalies, and the anomalies of temperatures above what you normally saw in those sites, and getting up above around 1 degree Celsius and seeing coral bleaching. Also at the same time, Al Strong, who some of you may know, and was my predecessor at Coral Reef Watch, started working at the U.S. Naval Academy. He was planning on being detailed to the Naval Academy, teaching midshipmen about satellite oceanography for a year or two, and ended up being there for 11 years. During that time, he started working with a number of the midshipmen, the new officers of the Navy, and doing projects on this issue of coral bleaching, because this was something that interested them. The work that Richard Montgomery did, developing an SST index for coral bleaching off Bermuda for the Oceanography Society. We have the one for the American Meteorological Society, Michael Gleason, correlation of coral bleaching events and remotely sensed sea surface temperatures, where they started really getting into this issue of anomalies. And it was about that time that when Al and Michael Gleason were working together that another thing was going on. And there had just been this election. Brought in a new presidency, and the vice president, Al Gore, who as a senator from Tennessee, had held Senate hearings on coral bleaching. And here we have one of his famous breakfasts that he would hold, these environmental breakfasts. And you can see Vice President Al Gore, Michael Gleason, Al Strong, and our Peter Glynn. They're having breakfast with the vice president and talking about the problem of coral bleaching. This is where all of these worlds start coming together. And the follow-on to that is Al kept working on this problem of heat stress on coral reefs, looking at developing these satellite products. Before it was even called a hotspot, this early idea of this SST minus the monthly mean climatology, some of us started talking with him and saying, based on the kind of work that Peter did, it not only was the temperature, but it also was the time of exposure. So you've got to figure out how to bring both of those together. And that led to what many of you know as the degree heating week product here during the 1997-98 El Nino, and really driven by that great work, a simple experiment that was done that identified how heat was so critical in driving this and identifying how much heat was needed to do so. Now at Coral Reef Watch, we've moved on. We were using not those old 50-kilometer products. We're doing work at 5 kilometers, sea surface temperatures, anomalies, hotspots, the early product, the degree heating weeks, and you've seen many of the bleaching alert area images that we use. And taking that same thing and not only applying it to satellite oceanography, but applying it to climate models and being able to predict it. Here, the work that was going on in the last bleaching of the Great Barrier Reef, bringing it back to what's going on today. This was 2016. Not only are those products accurate at estimating the amount of bleaching, but here you can see the impact that you can see of heat stress on changing color, the mortality of corals across the GBR. And by taking those, applying them to climate models, we can forecast up to 4 months in advance. Here you can see the forecast from July of 2015. This was the actual observations from the satellite data in Hawaii. We'll get back to Hawaii in just a second. These forecasts are now being used around the world. In Thailand, using these forecasts and knowing that bleaching was coming in advance, they closed off a number of dive sites to help protect them from the impending bleaching event. And that event I mentioned a second ago in Hawaii, they became concerned about many of their rare endemic corals. They went out. They set up this wanted poster to know where they were, went out and collected corals, brought them into nurseries on land in case the bleaching was severe. One of those, Porites dordeni that you see, at the time this picture was taken, that is the entire living population of Porites dordeni. It was wiped out in the field in Hawaii. They have raised it back up using tools that were developed in the Florida Keys for microfragmentation, took those and within less than 4 years of being wiped out in the wild, reintroduced this species into the same habitat it had been collected from. All of this because of the ability to go in and identify these things in advance. So for all of that, Peter, we thank you for your contributions. Questions? We had a quiet crew this morning. Everyone's just enjoying. Alina.
So I've been a very long believer in the effects of temperature on coral bleaching
Oh,
disease.
wait, microphone's coming.
Okay. So I was an early adopter of the temperature stress cause of bleaching and coral decline way back in the, in 87, 88, when we had our first really big bleaching here in the Florida Keys, et cetera. And one of the trends that sort of disturbs me is that people seem to be forgetting about temperature. It's like human thing, we get tired of hearing the same old thing. So now every paper I see out there, they try to invoke ocean acidification as the reason for the corals are going downhill. And it hasn't acidified enough to have a physiological effect on corals that include any mortality. If it slows down calcification by 5% or something like that, we're not going to see that. Yeah. And so, I worry because people sort of take their eye off the ball of the temperature if you start getting bored with that and looking for another cause of decline. Is the acidification community doing anything to come up with products, anything similar to this? But the data doesn't really show that corals are being affected by acidification at this day and age. Yes, 100 years from now, if there's any corals left, that's a whole nother matter. But what can you add to that?
Yeah, in fact, there's been work that's tied these together. I was just talking with Chris Langdon this morning about some of the work that he's doing on ocean acidification in corals, and he's finding in some of the corals what's been found previously, where acidification can even make the corals more susceptible to bleaching. So those two can go hand in hand. But you're right. I mean, we need to be considering ocean acidification, and in some areas that is the big issue, but not for coral reefs at this point. Because for coral reefs, that really is dealing with the rate of growth, the amount of erosion. It's huge in affecting the erosion, what goes on in the whole reef. But it really doesn't matter as much if a coral's growing a little slower if it's now dead. And that's where the temperature comes in. You know, it's just so much more of a direct effect. So you're right. I mean, we need to be worried about ocean acidification, but I think there's still enough attention going on. And one of the great things, it's interesting, these products that we use for coral bleaching gave rise to the whole concept of the marine heat waves that are now getting this whole attention for communities all around the world and so forth. And this whole idea, tying it back, it's the marine heat waves that are causing the coral bleaching and mortality and such. And it's really the problem is the one of mortality, you know, what's going to be around. We lost 50% of the corals in the Great Barrier Reef in 2016 and 2017, in a 2-year period, back-to-back bleaching. It just, you know, it's huge. And that is the big driver at this point, and that is what we need to really focus on. Don.
Peter looked at this before. Is anybody really looking at, I mean, light and temperature are obviously correlated because sunlight heats reefs, but light also stresses corals directly. Are people trying to look at that correlation?
Absolutely. I mean, light is important. This is photothermal bleaching. You know, the dirty secret is it's not temperature that causes bleaching. Temperature just causes the corals to bleach because of the response that's going on in the zooxanthellae as a, you know, in terms of the photochemistry that's going on. However, the big driver is the temperature. Now, light is a contributing factor. There have been some good studies in that. We've started developing some products. We actually were very close to having products out that use geostationary satellites looking at the light in combination with temperatures, and we have a whole physiological model we've published for doing that. Currently, we're kind of stuck in not being able to produce that because of things that happened with the data streams on the light data, but we'll have that going again soon. But it's important. Again, it's a secondary thing. The real important one is the temperature.
Okay, thank you very much. We now have Juan Maté from the Smithsonian Tropical Research Institute in Panama.
Thanks, Andrew, and thanks to the organizing committee, and glad to be here celebrating Peter's career and be part for a few years of that life. Let's talk a bit about early work that Peter did in Panama. He moved to Panama in 1967, and he spent 13 years there. When he moved to Panama, he was only 1 year old. And I never met him until he left Panama. He was already at RSMAS when I started interacting with him. He worked mostly from Panama Bay in the NOAA Marine Laboratories in the Amador Causeway. Imagine the Rickenbacker Causeway that divides in Panama. The Amador Causeway divides the Panama Bay in two parts, the Panama Canal entrance and the bay. The lab was located on the canal side of the causeway. All the big science that happened in Panama on coral reefs in the early years of Peter happened in this little laboratory. This is the Bridge of the Americas. This is the Panama Canal entrance. Everything happened there from 1970 to 1983. The lab is no longer there. It's now a public attraction that people go fishing, sightseeing, relaxing. But all the big science that started identifying reefs in the Eastern Pacific happened in this little place, and of course, all the field areas. The first work that Peter did started describing the reefs in the areas of the Gulf of Panama, which was an upwelling center. And during a dry season that runs from mid-December to mid-April, temperatures drop from 28 to 29 degrees all the way to 15 degrees Celsius. And it's one of the largest changes in temperatures that are recorded in the ocean. While in western Panama, in the Gulf of Chiriquí, there is not such upwelling. While Peter visited many reefs, there are some particular reefs that were recurrently visited. The Uva Island that Mark mentioned in his talk, and that's Peter's favorite reef, and that was the reef that many of us still work on that area. We got the Secas Reef, and in the Gulf of Panama, Saboga Island, and Contador Island, and those continues to be some of the most important areas. But for Peter, this is the most charismatic and interesting reefs of all in Panama. All these reefs in the Gulf of Chiriquí are around 550 kilometers from Panama City. So if you wanted to spend there several days, you either tow a small boat or you'd be there on site. And there were many reefs, many boats that were used to sample these reefs. The Teris in between 70 and 72, Estenella 72, 78, Benjamin 78 and 94, and that's when I started working with Peter, and the Rica from 94 and 98. Well, the boats were not available. Peter was using, or before these boats were available, he was using a Navy boat, or we started towing small boats to go to the area. And if you look through all the publications from the 1970s to date, you will see a reference to these ships and the captains of these ships in the acknowledgement section. So you can really track which boat was used on the operations. The early work, Peter started describing the diversity on these reefs, and he was the first to report fire corals in the Pacific coast of the Americas. He already identified 3 species of fire coral, and then just moving out of Panama, when he trip to Clipperton Island, he identified 4 species. Those were the only 4 species reported for the Eastern Pacific in Panama and Clipperton. I'm going to do a big jump into El Niño 8283 that Mark made reference to. And one of these species, Millepora platyphila, was bleached, died, and was wiped out from Panama. So our Millepora diversity was reduced from 3 to 2 species. This species in the Pacific, so it's currently present in other areas. The second species, Millepora bushmite, was extirpated in 82-83, so only was observed again a year later, but the 96, 97 El Niño completely wiped out the species. We haven't seen it on Mars, anything else in the Eastern Pacific, so it's gone from the area, but appears to be present in the Central Pacific. But at one time was reported as a local extirpation. And Millepora intrincata is the only remnant species in the area. The main difference between these 3 species is that these 2 species occupy shallow areas on the reef, while Millepora occupy a greater depths. So the variability in depth allow these species to survive in deeper areas. So the diversity of fire corals was reduced from 3 to 1 in this area of Panama. He also did a lot of interactions with species, looking at predation by the crown-of-thorns starfish. Let's see. He was a coral predator. Wait, it's not going back. It's not changing here. Just frozen. He was looking at this interaction between species. Acanthaster, the crown-of-thorns starfish, was a corallivore. It was identified as a species causing major problems in the West Pacific. It was present in Panama in lower numbers, but also have a natural predator that was feed on them. There was a little worm, shrimp, the clown shrimp. And when the shrimp were beating and creating bite marks on the starfish, the worm got into the starfish and started eating the starfish from the inside. Let's see if we can recover it. Yes. A lot of these interactions allow the species, the crown-of-thorns star, to be a check, so it did not become as a plague as found in other areas. In branching corals that Mark mentioned, the Poeciloporas, there is a symbiotic relationship between the corals and crabs and shrimp that defends the coral from the crown of thorn attacks. That's not mine. We have an issue. It's showing mine on the screen, but there's another one on the. Let's see. Yeah. Okay. Okay, there it goes. So here's the clown shrimp, Hymenocera picta. It's a shrimp that lives in pairs, male and female, and it's a coral starfish predator. Allow this little worm to get in and start eating the starfish inside, and their biological controls over the species. Also, there is defensive behavior by little trapecia crabs and Alpheus shrimps, and you will see some here. They're highly aggressive. They're like a coin of 25 cents size, and they deter the starfish from eating the large corals. Branching corals were the preferred prey compared to massive species. Let's see if we can see some few more. And you can see all the publications that came from these interactions and predation. You can see how aggressively the trapecia attacks and defends his coral host. The trapezies are symbionts and can only live within the coral and feeds on the mucus of the coral. So once the coral starts stressing, the symbiont will die. Another important description that Peter made was that because we have extreme tidal change that maybe between 5 to 6 meter amplitudes, a lot of the reef flats became exposed during low tides. And when those low tides coincide with midday, corals were cook and fry. And there was a lot of mortality going in that area. So there is a continuous process of mortality, slow recovery, and mortality. And normally we have these big tides occurring 2 or 3 times a year during spring tides, full moons and new moons. So there is this dynamic on coral recovery in these shallow areas. And this is the Uva reef that we mentioned before. Early work also focused on comparing reefs in the non-upwelling Gulf of Chiriquí, Secas and Uva, and the upwelling Gulf of Panama, Saboga and Contadora, and there were like 10 additional reefs on this paper. Even though reefs began to form at the same time, Gulf of Chiriquí reefs were a bit more thick. They attain larger thickness, they grow faster and quicker. Posilopora, the branching coral, is the main species growing up, but it's not responsible for that differences. Corallivores, like the crown of dorsal fish, other snails like Generia, could be responsible for that, but it was believed that upwelling was probably responsible for these low growths on the Caribbean side. And I think Rick will have a different hypothesis that he will be presenting today regarding these conclusions that we made in the early 77s. From all the work on the description and El Niño bleaching event, there were 2 main papers that people presented. Mark already mentioned this one, that obtained the best paper award in the coral reefs, where they did microcosm experiments comparing temperature effects on corals from the Gulf of Panama and Chiriquí. He did this paper with Luis de Cross, that became my advisor at the University of Panama there as an undergrad. And this is his 5th more cited paper in Google Scholar. And also he did a major problem on coral bleaching ecological perspective that got also in 93 the best paper, when he described most of the bleaching events recorded today, major outbreaks in mass mortalities, relation to the scientific information being generated by the establishment of the main marine labs through the different oceans. And this is still Peter's most cited papers. I never met Peter until he left Panama. He came to RSMAS. And even I wanted to work with sharks. They were not shark science professors at the University of Panama. So I started working with Luis de Cross, that told me he was working with these scientists from the university, from RSMAS, that at one time was a STRI. So I started looking at the synergistic effects of UV radiation and temperature. That was the line we're doing in. And we were doing basic work, doing zooxanthella counts, coral extractions, and so on. So that's how I started working on on coral reefs. We were from a small pier that the Smithsonian had at that time. And from there, I got my undergraduate. Peter was coming to Panama in '89. I met him like a month before this trip. I escaped my undergraduate program and just went to the field with him. I dropped my semester at that time. And we spent 2 weeks in Uva Island. And you can see Morgan that was working on his carbonate budget and his dissertation, and Dave Mead that was working on the reproductive ecology of porrites. So that was my real first interaction with coral reefs everywhere in the science world. I was diving already in other areas, but scientifically that was my first interaction, and that's kept me hooked for the rest of my life. Then on a Fulbright scholarship, I came from my master's and then remained for my full PhD, almost 10 years here. I spent a lot of time. I went through a coral reef symposium in Panama, and in '97, I did El Niño there. I started in coral reef ecology, and then I became the TA for the course a few years later. And you can see how the years have treated us all through all this time. Once I started working with Peter, a lot of the effort was also placed on coral reproduction. Few of the papers were already published on Poeciloporas and porrites. And people were looking into recruitment, fecundity, and how corals can recover, reef can recover after El Niño coral bleaching, and how this will provide resilience to the reefs. Many of these species were gonochoric, that means separate sexes. Only 2 of them were brooders, that means they released the larvae into the water column, and many of them were broadcast spawners. So we have 12 species that have been published. We still have 1 fire coral that needs to be worked. We hope we can do it sometime in the future. 18 researchers participate in all these papers. So I participated in many of the collections every month in Panama. I was the point man in Panama for the collection, so keep me in the race for some time. As I was doing my dissertation on pavonas, I spent a lot of time working on the reproduction. You can see the spawning of one of the pavona variants. And that was one of the 3 species that we observed spawning. All the other species were observed mostly by histological examination. We knew the brooders because we got the larvae. For the spawners, only pavonas. At these times, we know we have 2 species, but most times they were considered siblings. We got enough information. We finally observed them spawning. One of the species, pavona variants, spawned just before sunrise, and the other spawns just after sunset. So there were 12-hour differences almost in their spawning, so that means there were some barriers to be able to fertilize each of the gametes. Eggs were differently, so there were evidence that they were at one time different species. So we finally described the 2 species as separates. We call this Pavona churiquensis, that is mostly an encrusting coral that live mostly on basaltic rock, and variance is mostly a reef species, but is also found in rocky areas. We also found a new species called Fungia curvata. It's a solitary coral. All the paleontologists have been dredging deep areas and has only come as dead skeletons. We found it on the second reefs as alive. In this year, in 2001, we found only 2 colonies. In last year we visited, there were already 15 colonies, so that means they're reproducing in the area. And it's the only place in Panama where we can find these species. And until then, it was only recorded as dead skeletons. In honor of Peter, Hector Guzman and Nancy Budd describing a Siderastria coral in the Eastern Pacific was the first export to the region called Siderastria glynni. And you can see and was named Peter based on his pioneer work on the Eastern Pacific. And only five specimens were known at then, and one was sacrificed for the description and the type specimen. So you can see the quality of work that he did, that he has a species name on his name, and then several years, he needs to get rid of that name so that he doesn't have a coral species name. Why? One early researcher appears to be transplanting Siderastria syrea from the Caribbean that assumed to be dead, And then they started recovering. When they came later into the reefs, they found that there were some smaller colonies being grown into larger colonies. Those were later removed, and they're no longer in the area, so we don't have any more Serratia glynni in the area. In 2001, I was asked by the Panamanian Postal Service to provide some, I was at that time the only coral reef biologist in Panama, to provide some images for a special postal issue. They asked me for a Caribbean reefs and Pacific reefs. Again, Uva reef was showcased, and I showcased also 2 species that was named in the name of Peter. Serratia glynni, that have the glynni name there, and Pavona chiriquiense that we have just described the same year. So Peter is in the postal just circulating stamps all over the world too, not just in scientific papers. Coral reproduction also was done into Austria. We spent a lot of time on one of the boats, Rica, looking not only the information that is zoological, but looking on field on information. And we know we're looking at recruitment and looking at the lunar phases. And most of the corals in this particular are what we call lunatics. They follow these moon phases where they prefer also the warmer months of the days, of the month, of the year. Our main paper on coral bleaching was the 2000 paper. We looked for almost a year on the responses of corals. I was in Panama doing my dissertation then, so I spent almost every month, so we were able to capture this bleaching event. The main features, there were 2 phases, one in 2007 for a couple months. The threshold for bleaching was around 30 degrees Celsius. Once the warm waters start cooling down, then corals start recovering, and again in 1998, they start bleaching again. The waters hit 30 degrees Celsius and start bleaching again, so we were able to capture that. And as Andrew mentioned in his presentation, the Poeciloporas, we were observing a shift on the zooxanthella that they were present, the symbiont that was present in the corals, from a sensitive algae that allowed the coral to bleach quickly to a more tolerant or resistant algae that allow corals to survive. More recently, we have been looking at cryptofauna, simulated reef frameworks. That work still needs to be done. We were 4 years collecting a lot of data. We have a lot of information, so we need to spend time there. A lot of the work has been done on ships, and at nights, after finishing the work, there were a lot of interactions, but the most typical part was poker nights, where cable ties were the chips for the work. If you lose, then you don't have any cable ties to tie your samples the next day. So finally, you have an array of photos, a collage of photos from everybody. You know why no one told Peter no when he was in the field. And finally, a last message from Peter's assistant, Aníbal Velarde. He was his assistant in Panama for 13 years. He's now retiring after 50 years of service in Panama. And he sent just his kind regards to Peter. And that's it.
Going to be from Derek Manzello across the street at NOAA's AOML labs.
Thank you very much. I usually don't bring my cheat sheet, but I felt like this was more important than my science talk, so I had to make sure I didn't forget anything. So my talk's going to be a little different. I'm going to talk about Peter's influence on my career and my life. So my name's Derek Manzello. I was Peter Glynn's last PhD student. Actually, that's not true. There were two more PhDs that came after me. I would later find out that there were no less than 5 PhD students that were Peter's last student. And this speaks to the man we've already heard so much about. He just could not help himself. He had to keep mentoring students because Peter Glynn, above all, is a teacher. And And this is why he's one of the most world-renowned coral reef scientists that has ever lived. Everybody, no matter how accomplished, learns something every time they talk to Peter Glynn. For Peter, sharing knowledge on coral reefs and shepherding the next generation of coral reef scientists along on their careers was something that he is still flat out addicted to. So before I get into my talk, I want to point something else out. I want you to pay attention to the dynamic that exists between Peter and all his students. When we are around Peter, all of us instantly revert back to being his student, desperately trying to impress the man. I've taken great pleasure over the years watching people from their 20s all the way up into their 60s and now, pushing that, trying to impress their old teacher. And this is because Peter's always challenging us to be better and be the best. He makes everyone around him better, and he is the most inspiring and smartest person that I've ever known in my life. So Peter Glynn was my professor for Introduction to Marine Science at the University of Miami as an undergraduate. Now, when I took Peter's course, I was having a bit of an existential crisis. I wasn't really sure if marine biology was what I wanted to do. This is because my Intro to Marine Science course, based off this book you see here, ended up being very uninspiring for me. Now, this course was mainly focused on physical oceanography, satellite oceanography, mostly open ocean, large scale, principles of the marine science. The topics were as abstract to me as a Picasso painting, and it made the oceans seem both incomprehensibly large and small at the same time, with everything represented by a tie-dye swirl of colors superimposed over the 7 seas. It was dry, it was boring, and a lot of kids dropped out of marine science right after that course. Now, I didn't take any marine science courses my second semester of college, so by my sophomore year, I didn't know what the hell I was going to do with my life. So it was time for Introduction to Marine Biology, or MSc 230. I can still see the professor walking in. He had on a baseball cap, and he looked more like Jimmy Buffett than the pompous assholes I was used to dealing with. He wore a contagious smirk that immediately put everyone in a good mood whenever he was around. He was unassuming and he was soft-spoken. He taught the class with an enthusiasm and emotion of a child seeing a shark at an aquarium for the first time. I hung on to every word and soaked in every bit of knowledge he shared with us. It was more than halfway through the course before I really learned who Peter Glynn was. Now, the course was based on the book by James Nyback, and you see there. And Dr. Glynn baited us for weeks about the lectures on coral reefs, like they were the final season of Game of Thrones or Return of the Jedi. I guarantee I was more excited about those lectures than anyone else in that course. And I vividly recall sitting in my dorm room in Pearson Residential College, reading Chapter 9 in Nyback, and the semester was more than halfway over, and the burnout of that semester was taking its toll. At first, I didn't notice it, but after about the 3rd time I saw the name Glynn pop up as a citation, I thought to myself, wait a minute, is this the guy teaching this class? You have to remember this was 1999. The internet, going online for information was an exercise in frustration. The internet took forever, it was slow, and there wasn't much content on it, so I couldn't just type in Peter Glynn and go, oh wow, this guy's a esteemed coral reef scientist. I still wasn't sure, is this my professor? Is that even how he spells his name? Finally, I figured it out. The humble man teaching me Introduction to Marine Biology was the same guy in this book. I remember flipping to the reference section and boom, 10 citations, by far the most of anyone in the chapter. My professor was a goddamn rock star. This made me even more scared to approach him. Believe it or not, I'm a shy, quiet guy. I'm never one of those gregarious undergraduates that would sit around and shoot the breeze with their professors. But one day before class, I was sitting there taking notes on that week's creature feature. Once a week, as part of Peter's course, he would highlight a marine organism, and we had to go in and check it out, write down notes, draw a little sketch. Peter would always come to campus early and lurk in the lab so he could interact with students. He approached me and said, "You are doing very well in the course." I nervously said, thank you for noticing. Now, this simple act of, pardon my language, giving a shit about students was huge and meant so much to me. Things like this don't show up in publication metrics or professors' point systems for tenure. He took the time to interact with students and encourage those who were doing well to keep doing well. Peter challenged his students like no one else, and his exams were notoriously impossible. He'd pull out some random factoid that he said in his aside, or include questions on a topic that were buried in one of the many pages of reading materials he assigned as a test to see who was really reading the material. Students that were used to doing well their whole lives bombed exams and freaked out. Me, I killed it. I was one of the few people that managed to read everything he assigned. I was the only one in the class he couldn't trick. One of the questions that I will never forget was to spell out the name of the common foram in the Caribbean, Homotrema rubrum. I was the only one in the class that got that right. It was one of my proudest moments. I have never met anyone who is more in awe and in love with coral reefs than Peter Glynn. So of course, I fell in love with coral reefs. From then onwards, I put coral reefs and coral reef science on a pedestal as the apex of careers in marine science. That was the holy grail. So one of the things we take for granted today is that scuba diving is a relatively safe activity as long as you are well trained and dive within that training. Put differently, we all know from years of people diving that it's highly unlikely that you're going to be eaten alive by a shark. When Peter and his buddies like Jerry Wellington started exploring reefs, no one knew just how dangerous it was to go swimming around the ocean. Now imagine yourself falling off the back of the boat in the northern Galapagos Islands, hundreds of miles from any basic form of civilization. There's no one else around, descending and seeing this. Just think about the courage that took. Now that thousands of people have put in hundreds of thousands of dives, we take for granted the knowledge that we aren't going to be eaten alive when we go into the ocean to scuba dive. Peter never had that luxury. He was one of the first people to ever see sights like this on scuba. He was the literal guinea pig. If I were in his shoes and I fell in the water and was one of the first people to see this on scuba, I would probably be an accountant in Nebraska right now. Between my junior year and senior year at college, I did an internship with Diego Lehrman at RSMAS. Now, Diego was an amazing teacher and person in his own right, and unfortunately, he couldn't be here today. I spent the first few months of that summer doing experiments on corals and doing field work in the Florida Keys and Florida Bay. Diego instilled with me the confidence that I couldn't actually be a coral reef scientist. So there was no turning back. It was coral reef science or bust for me. As my internship came to an end, I went around to the marine biology professors at RSMAS, whose research interested me, and emailed them to sit down and talk to them about opportunities for Ph.D. students in their labs for 2002, as I was determined to stay in Miami for graduate school. I talked to people like Sam Snedeker, John McManus. Jerry Alt never returned my email. I'm not pissed about it, though. Now, even though word on the street was that Peter Glynn was retiring and no longer taking students, I decided to meet with him anyway. It was July 2001. I hadn't seen the man since handing him my final exam in December 1999. He immediately agreed to meet with me. He welcomed me into his office that was lined wall to wall with reprints of his publications, coral specimens in shoeboxes, and other knickknacks. He sat me down and he said, "Unfortunately, as you know, coral reefs are in decline. But the one benefit of this is there are many opportunities opening up in coral reef science." That was all the hope I needed. A day or two later, Peter emailed me and told me there was an opportunity to work with a guy named Jim Hendy at NOAA. In just a few weeks, on July 16, 2001, my 21st birthday, I began working at NOAA. And it's been over 18 years since that day, and I'm still working in the same building. A few good words from Peter Glynn can change a life. Shortly thereafter, I received an email from Peter informing me that he could take me as a student in the fall of 2002. Also, he was curious if I'd be interested in joining his 3-week research cruise to Panama in March. Are you kidding me? Euphoria kicked in. In March 2002, I joined Peter Glynn on a 3-week research cruise to study the coral reefs on the Pacific coast of Panama. I was still an undergraduate. How cool was this? Nobody I knew of, either from high school or in college, was doing anything close to as cool as what I was doing for spring break my senior year of college. This was the first time I had traveled outside of the United States. I had been to Canada, but that doesn't really count. I vividly recall when my face went underwater and I saw a Pacific coral reef for the first time at Isla Cavada diving with Tyler Smith in the Gulf of Cherokee. I had heard tales of these reefs in class with Dr. Glynn, and seeing them firsthand for the first time was mind-boggling. For almost 3 weeks, I was almost continuously underwater from sunrise to sunset, and then at night, I'd fish off the back of the boat. It was a tropical paradise. I experienced a sustained bliss that I've never experienced since. It was incredible. So after my first semester of grad school, Jim Hendy invited Peter and I to a workshop he was hosting at Leastocking Island in the Bahamas. Now this was a small meeting, about 20 participants, but it included some of the who's who of heavy hitters in the world of climate change science on coral reefs. I mean, just look at this list of people. Danny Gleason, Mike Lesser, Todd LaJeunesse, Mike Risk, Ray Berkelmans, Bill Fitt, El Strong, who Mark already talked about, Andrea Gratoli, Ilse Kuffner, Mark Warner. It was almost impossible to attend this meeting and not end up having a successful career after being exposed to these people. My favorite part of this meeting, though, was when at the end, Mike Risk got up and literally told us we were all a bunch of idiots. Now, this type of experience is something very few graduate students get to have. To see that scope of work presented, as well as be challenged by someone like Mike Risk, was beyond priceless. I can still see Peter standing up and introducing himself to everyone, despite the fact that everybody in that room knew goddamn well who Peter Glynn was and knew his work like the back of their hands. Peter stood up and said, "I'm Peter Glynn from the Rosenstiel School. I'm still in mourning over the fact that the Miami Hurricanes football team lost the national title to Ohio State on that BS penalty. Peter, it's been 17 years and I'm still not over that game." At this point, I was only one semester into graduate school and still searching to really nail down my research topic. I knew I was fortunate enough to have access to the Eastern Pacific, but I was really looking for the best possible niche to find. Now, it was late in the meeting and Joni Klepis got up. Now, bear in mind, the term ocean acidification hadn't even been coined yet. She put up this slide. Now, this basically shows carbonate ion concentration in pre-industrial times and with a doubling of atmospheric CO2. I immediately noticed that much of the eastern tropical Pacific was experiencing conditions of low saturation state, similar to what was expected to occur for the rest of the tropics with a doubling of atmospheric CO2. And the rest is history. Here is a photo of me proudly displaying my CO2 sampling bottles in the Galapagos Islands in May of 2003. Now, that workshop, hosted by Jim Hendy, ended up being the most important meeting of my entire career, as I met people that I collaborated with on projects for the next 15 years, and it set my career in motion. In closing, Peter Glynn was undoubtedly the seed that sparked my entire career. Being a student of Peter Glynn opened doors for me and led to a career that I cannot even believe happened. If you would have told me 20 years ago when I was reading that chapter in that book that I would be standing here today, I would not have believed you. Being associated with Peter was huge. It set me above the common graduate student, because being Peter's student carries immense weight. Peter didn't just take anyone. You had to be special. In closing, there is no career accomplishment that I ever wear as proudly as the fact that I was, I am, and I always will be a student of Peter Glynn. Thank you.
We have a couple of minutes for a question, if there is one.
Uh-oh.
I want everyone to know that Derek was known as the 100%er in my MSc 230 course, because I could never trick him on any question. Every quiz, every exam, he got 100%.
Thank you.
Thank you. Okay, we're going to head into the break now. We have a half an hour. You can catch up then with Peter, the man himself, or any questions for Derek, take them at the break. And we'll be back here, please, at 10:30 promptly for Mitch Colgan's talk. All right, self-explanatory. All right.
But I can figure it out.
Well, here's the thing. There are a whole bunch of blank cards in a pouch
Yeah, I
there.
saw that.
Yeah, so you could just use those.
Okay.
If I don't have time to print more out. Seem to be. Yeah. All right, we'll make that one. That's the next one, yeah.
How long is the week?
Half an hour, so till 10:30. Actually, let me get rid of this stick.
should be. Like the first guy's coming.
It's the last. This station.
Oh, yes. Well, Peter only has short remarks on it. Yeah. His big talk is the same.
So we have it here, but it doesn't, it's not recognized. Yeah, it's not recognized. So I think it's going to be safer if you talk to your laptop, or you need to talk to the guy who knows about your
I'll give you a hug.
Did you get everybody a badge and a bookmark? Did you get a super bookmark as well? Good. And sure, if you just ask them. Yeah. Oh, awesome.
Nice to meet you.
Hello.
This is the lady with the apple. So she's going to have to do a whole swap. And she is. So we've got 3 talks.
nervous? It's okay.
I
Once
don't
you
want
get
to embarrass
started.
No, I don't want to embarrass you. And, the others had a very different approach to their lectures than I had.
Oh, okay.
We'll see it. So I,
Waiting with bated breath.
I know. It's completely different.
Where's, you see Mark?
Mark was up.
That beautiful picture of Uva Island.
Well, mine is scary.
If everyone can take their seats, we're going to get started for the next session.
Yeah.
I finally got to see this in 2012. I was so excited. Wow, that's funny.
Yeah, I know. Often, probably. Starting right after 3 minutes.
We good? I think we'll go ahead and get started. Our next speaker is Mitch Colgan from, what is it, Charleston? College of Charleston?
Yeah, College of Charleston.
Cool. I met Mitch today for the first time, but I'm well aware of him from his contributions on Galapagos coral reef development and the Urvina Bay uplift, which I finally saw in 2012, and I was very excited to see that. Without further ado, Dr. Colgan.
Well, thank you. It's my pleasure to come and speak before you to tell you stories about Peter. What's interesting is that so far, before we begin, so many of the stories that were told at the beginning were the same feelings that I have about his special relationship to science and to my personal life. And so you kind of understand maybe the title of this. The Urvina Bay uplift is on the Galapagos Islands, or how Peter turned a clean ecologist into a dirty geologist. There's a few times in one's life that one understands the exact moment when your life turned. And I can point to that time. Here, covered with mud, we just went through our Neptune party. And this was after being cleaned up a little bit. And I'm sitting there thinking about coral reefs and the other things that I used to do, sitting above the Arctic Circle, covered with mud, studying sediments for the U.S. Geologic Survey. But all the time in the past, I dreamt of coral reefs. The coral reefs was what I wanted to study. I loved them. The first time I started studying coral reefs, or it got the interest, when I got a book from my mother when I was 8 years old. And I went to the Guam and to the Philippines as a Peace Corps volunteer and Guam as a student. And I finally was there doing what I always wanted to do, study reefs. Well, then there was a fateful meeting. Peter Glynn came into my life, and that changed everything. I worked as his assistant on studying acanthaster and the crab trapezius, and looking how they would protect the coral during their attack. And so from that, we spent a lot of time together, studying reefs at this place on Gun Beach in Guam, where our day would be spent underwater for 6 to 8 hours a day. We'd be sitting there, and we have a person on the boat handing us tanks. We never got out of the water, we just got handed tanks. It was the most intense working period I ever had. We came close, and I got married on Guam, and Peter was my best man. And so, just to tell you just a little story about that, when as a best man, there's certain responsibilities, and so one was to get me to the wedding on time. So we cut our research short. Instead of staying out to 6 hours, to 6 o'clock, we came back at 3. So we got there, still wet. We had the lovely sunset wedding, and then for the honeymoon, 12 o'clock next day, we have work to do. Now, I know all of you have had that same experience in slightly different ways, but it was the idea of the importance of the work, getting it done, and the enjoyment of doing it even under the most interesting times. Well, my father died when I was 10 years old. Peter knew that. And so after I was married, he had the talk. Because my honeymoon was interrupted, so he still thought he should have the talk with me. But it's not about what you think. It was, you should be a geologist. Well, I was enjoying my life. I'm thinking, this is the highlight of my thing. I was never any happier, and you should be a geologist. You have responsibilities now. So I was faced with a dilemma. 2 roads. Well, Peter has a great deal of belief in the fact that there wasn't much future in ecology for me. I don't know what that meant, but I got the hint that, So we took a slight detour, and instead of going into continuing ecology, I started at UC Santa Cruz as a geologist on my road to doing oil work. Because Peter gave me this one inspirational thought. He says, you know what? You could study coral reefs as fossils and still do the same thing. And I said, oh, that's fine. And Shell Oil actually hired me to do just that for a while. So Peter changed my life at this one time, and he made me, and in this part, to explore places I never thought I would ever go to, even dreamed of. From working at Shell to working with the U.S. Geologic Survey. I was the director of the South Carolina Space Grants Consortium, working with NASA for 15 years. I even was part of a team that put a payload on board the shuttle. And for almost 18 years, I was the chairman of the geology department at the College of Charleston. All of that wouldn't have happened, and many other things wouldn't have happened without Peter sitting down and having that talk. But first, another important conversation with Peter. As a graduate student, was kind of suffering, figuring out what to do, lost. I know Derek didn't have that problem. But so we had an intervention. And so with this long conversation, Peter suggested I could be a geologist and study a coral reef without getting wet. So here, if you've seen the picture of Jerry Wellington, John Wells, and Peter at the Urvina Bay uplift, I was tasked with his suggestion and prodding that I should study the Urvina Bay uplift. And this was a marvelous opportunity to study a reef in its wholeness without having to worry about staying underwater for a short period of time, without having to worry about the sharks, to having to worry about anything except getting sun bleached and tanned. So off to the Galapagos Islands. With the help of the NSF grant, I went to the Galapagos Islands and went to this little white dot. So here's Isabella, Alcedo, Darwin, Ferrandina, and that white dot. That white dot for 3 different field stations would be my home. And that is here at the Urvina Bay uplift. Now, we know about the Urvina Bay uplift exactly when it happened because it happened in, oh, sorry. Oops. Oh, I'm back. I hit the stop button. So this is the earliest aerial photograph of the area in 1946. You can see that the bay here, this is Urvina, and this is all underwater. And then in 1954, you can see Mickey Mouse is out there, because they had a film crew going there for the filming Disney's Enchanted Islands. And they went in and they surveyed this and they left the area. And they came back to find this. The whole section was uplifted. We think it was uplifted overnight. In that, the people that were there talked about the great smell coming off of the uplift. The fish were everywhere. We could still find, even after when we weren't there, find turtle skeletons, sea turtles that were trapped in pools. And so we knew exactly when everything stopped. And we had this opportunity to study, to walk around what was originally, all of this was originally underwater and was frozen in time. So for 3 months, we lived in tents on the beach, and we did our first survey of this uplift. And the survey allowed us to have the opportunity to really see. I mean, the idea is, instead of seeing small transects, we were able to see this entire area and its relationship, the spatial relationships between the corals, and dig in longer periods of time than you would have by diving. And you can see here, you can see the way the land was. You can see low sea level and high sea level. You can see the markings here. So we knew the water depths that we were in. We knew the surroundings that we were there. And so we began to explore. And our exploration came to finding this very unusual place. If you look way back here, that is the shoreline right there. The old shoreline is the white sand beach. And you have this whole area right in through here is Palosophora colonies that are spread out. You have corals with trees growing out of them. And so you have this very strange and unusual place that we explored. Because we had the opportunity to see so much of it, we started to ask some questions. And one of the questions that I thought was most interesting, studying from Guam and knowing about other reefs, is why, excuse me, why the reefs in Urvina Bay were so different. Because what we found, unlike what you see in the Eastern Pacific and other parts of Eastern Pacific and see in places like Guam and the Western Pacific, is that corals dead corals were there, but new corals grew on top of them. And so we have what we call an antecedent framework, that corals grew on top of corals. That when a coral died, another coral could grow over, and we have this sustained growth of developing a true framework. We don't have that in Urvina Bay. We have corals that are sitting there as small colonies sitting on top of rock, on top of the basalt. And we had colonies were all of the same age or similar age structure. And one of the things that we'd find is large corals like this that had no other corals living on top of them. In fact, instead of recruitment taking place, they were being destroyed. And they were being destroyed from the inside out. All of these are boring lithofags that were coming in there and tearing out the coral and weakening it. What we saw as a community that was on balance. It was very healthy as long as it wasn't disturbed. And as soon as it was disturbed, everything fell apart. In this area that looks like it's a very robust coral community, we have to look underneath. And when we look underneath, we found all the corals were hollowed out, that no coral was a solid coral. We saw this also when we would go diving, that there was a hollow structure. And underneath these corals, we found Eusoderis. The sea urchin was there that came and scraped off algae and tore up the coral. And the corals began to collapse after death. And we can see that here. This is a large Postlepora colony. And from the edges, they're being chewed and reduced. And they're coming in from the underside and toppling these corals. And the corals are simply crumbling. Nothing else is growing on top of them. They're not being recruited to. And the corals that were once there simply disappear. And you can see here the hollowness. And so, of course, this led me to only one factor, one idea, the beaver effect. It doesn't matter how much material is being removed. If you remove enough from underneath side, the coral colonies collapse. And that's what we saw. The underside of the corals were collapsing from the effect of the sea urchins gnawing on the coral edges. And so we have places like this, we're perfectly available for corals to settle on, and the corals are gone. We see that there isn't very much reproduction taking place, and that the coral areas that could have corals were gone. We also found huge corals that were one time there that have been erased. And this, for example, here is one of those corals. We've found a number of them in which there was a thriving community and then was gone. Well, with that, I'll end my talk about the fact that we just see this change that happened, a change that happened in Urvina Bay, and a change that happened in me. And just, we have this one last remembrance here. As Peter came to visit us, we had I have no idea why we had chef hats out there, but he got a chef's hat, and therefore he was the chef of the island. All right, well, thank you very much. I appreciate it.
Thank you. I don't know how you spent 3 months there. I spent about an hour there, and I ran back to the ship in the air conditioning. It was miserable. Our next speaker is Dr. Tyler Smith from the University of the Virgin Islands. Tyler and I overlapped a lot and spent a lot of time in Panama together. Had a lot of good times. Without further ado, I'd like to introduce Tyler. Oh, I don't pull up his talk, sorry.
Great, thank you. Thanks, Derek, Mr. 100%er, for preparing so much for your talk and really setting the bar so high. So mine's gonna be a little more off the cuff. That's all right, though. I'm really pleased to be here. I'm really pleased to be here to celebrate Peter Glynn's career, legacy, and the man himself who was so important to me and so many of you in this room. And I guess I should note, the title of my talk, No Stress with Coral Stress, is because Peter would stress you in ways that were subtle, but in terms of being friendly and positive and just always having a great time in the field, in the lab, wherever, that positivity always radiated from him, was always just made any experience with him so terrific. So that's the title of the talk. That's where it comes from. So who am I? My name is Tyler Smith. I'm a research associate professor of marine biology. I grew up in St. Louis, so like Derek, a Midwesterner, we make the best marine biologists because we love the ocean so much. I did my bachelor's of science at Western Washington University. And there I did a cruise around the Pacific for a summer. I was a retiring professor. He had always done this trip to Hawaii. And he decided, as he was going out, he would do this one trip with students all the way around the Pacific. So 9.5 weeks, we circled around the Central Pacific, and it changed my life. And I knew I wanted to do coral reefs. I sort of screwed around for a while and finally got into grad school here at RSMAS. And I had an opportunity to really study coral reefs. And that was exciting. So I did my PhD here from 1998 to 2005. And since then, I've been at the University of the Virgin Islands. I had planned to do a PhD based more on coral physiology. Actually, I talked to Alina when I was coming in, and she informed me right as I was coming in that I'm leaving this year, so I'm sorry, I won't be taking you as a student. So I was kind of a little adrift, but I had an opportunity because I had a Maytag fellowship to look around to find a place where I was comfortable. And I talked to Peter, and he looked at me and he said, oh, this is good. You know, I see you used to get B+, and I also was a student who got B+, so I think you could be okay. So it's kind of a compliment. But from that, I switched to coral ecology. And I really, like Peter, I was interested in the biology of coral reef systems. I really wanted to study how this worm A interacts with coral B to affect algae C. That's what I really wanted to do. And of course, as things are going with coral reefs, none of us are actually doing that anymore. But the lessons I learned were extremely important. So I studied actually using the upwelling system in Panama. I looked at how that upwelling affects the algal system in those reefs and how that affects the competition with corals. But it very much prepared me, my interaction with him and the Eastern Pacific, and the fact that it's one of the longest histories of thermal stress really prepared me for understanding my current job, which is understanding coral decline. So this is coral cover in the Virgin Islands since I've been there, just a little bit prior to when I've been there. You've seen a dramatic decline in coral cover, and so that's what we're studying now. Most of that decline was from the 2005 bleaching event in the Virgin Islands, and I was already primed. I actually started my job while the bleaching event was going on, and I mean, I was very fortuitous to be so well trained in the lab of Peter Glynn to understand that event and take the reins right there. I'm the coordinator for the coral monitoring program in the Virgin Islands, and so boom, I had that experience and could go right into it. So let's switch to the Eastern Pacific. And I want to get a little scientific here and talk a little bit about the ways that Peter has influenced our understanding of both El Nino phenomena, coral bleaching, and how corals respond to bleaching in different areas. So we know that the Eastern Pacific has experienced, in modern ecological literature, thanks to Peter, 3 El Ninos, 1982, 1997, 98, and 2015, 2016 El Nino. Each of those was about as strong as the next, and you'll see that in a subsequent slide. But interesting is the coral response to that is not the same, and that's what I want to talk about. And a lot of this, as you've seen before, comes from this small little island, Uva, in the Gulf of Chiriquí. In 1982-83, the reef was almost essentially wiped out. It was left with remnant corals. But you can see, this picture from 2005, that it came roaring back after that, and it survived through numerous events. And so what I want to go through is a little animation of a plot that was set up by Bob Richmond, a 4x5 meter plot that to record coral recruitment right after the bleaching event in 82-83. In 1984, they set this up. This area, so anything in the plot behind there that's green is dead coral rubble. That was all killed by the Nino. This plot was about 75% coral cover during that period. Anything blue, those little dots, is living post-lapid corals. And then red, which you'll see later, is dead post-lapid framework. So I just want to go through the animation. So this is 1984, almost nothing there. And you can see the corals starting to come in and recover up to 1997. And you would expect then, 1997 was actually probably a stronger event than 82-83, that this coral then would die back from 97-98. But instead, the next time the measurements were taken in 2000, the corals barely registered that event. And that's, again, what it looked like in 2005. So these corals had become somewhat resistant, apparently, to that thermal stress. Fast forward to the 2015-2016 El Nino, and now that recovery is almost complete for that framework. And what's going to happen here is we have this next El Nino, very high coral stress. During that event, we did see bleaching partial of the framework, even though it was very, very high registered stress. There is the 4x5 plot you can see during that event. And just a little blip down. So this reef framework has really shown tremendous resistance now to thermal stress. Now, I don't know if that can hold out through future levels of climate change, but it's interesting, at least in this part, that we're seeing as well as other parts of the world, that some corals can develop resistance to stress, either by being culled and you're left with remnant survivors, or through other adaptations. So this is the temperature record over that time period. And you can see the coral thermal stress is on the top graph of the red lines. So 97-98 was probably even more stressful at about 13-degree heating weeks than even the 82-83 El Nino. And then the latest El Nino also was at least 10-degree heating weeks in 2 separate bouts. And there is, again, the coral cover through that period. And I know this is not going to play. But essentially, what it is just a lot of bleached coral. So the surface reef, the shallow reef, was highly bleached, actually, in some areas. The massive corals were very bleached, but the post-operative framework that builds a reef did quite well. But as you look deeper, and this was an idea that Peter saw after looking at Melepra, these fire corals, and realizing that one of these species, Melepra intricata, actually had a depth refuge over that zone in the Gulf of Cherokee, whereas the other two species went extinct regionally, is that there might be a depth refuge, and he posited this idea, and I think Bernhard Riegl also at the same time posited the depth refuge for reefs he was looking at in South Africa. And so I spent a lot of time after that looking at this phenomena, particularly in the Virgin Islands and now also in the Eastern Pacific. So this is a record from 2014, 15, 16 at Uva Island and the Gulf of Cherokee in general. And it shows that for Melepra, which is the little inverted triangle, that they all died in shallow water, 0 to 6 meters, after that event. They suffered bleaching, about 60% at 17, 14 meters, and almost no bleaching once you got at 15 to 24 meters depth. And then they all died out in terms of partial mortality by 2016 in shallow water, but survived again. This is the third time we've witnessed them surviving in deep water, and probably when we would populate after that. But it also, for other coral species as well, for massive coral species, kind of consortium of massive coral species, and for postalaprids, they did much better with depth. And part of the reasons for this, and perhaps the Eastern Pacific is a little unique in this, is a very strong thermocline, shallow thermocline with depth. These are the 2 years, 2015-16 record on the graphs. It's a little hard to see the different colors, but you get the gist that even by 10 meters, there's much cooler temperatures. That blue line on top is the bleaching threshold, and that protected them in terms of degree heating weeks over that period. And the reason for that is a very strong pycnocline. So you have very low salinity water and very hot water sitting on top of very cold and salty water. That leads to a very, very strong resistance to mixing. And that creates a condition where even during El Nino events, water is not downmixing. The hot water is not downmixing. It's staying at the surface and protecting these corals. This is not a phenomenon that occurs throughout the world potentially, but at least in the Eastern Pacific, in this Gulf, it's highly protective of these corals. And so that brings us to the people. So I was looking at all my photos of Peter Glynn and our time in the field, and was trying to be inspired, and I think, oh, maybe I can categorize all these interactions into different things. So I came up with these themes of the photos and what we were doing. So obviously there's a lot of photos of field sampling. Here's Peter with Jerry Wellington, Peggy Fong with her daughter Caitlin, who's now also a reef ecologist. And of course, taking group photos is an important part of any activity, right? That was, this is in Okinawa, and I believe this is 2010 in Panama. There's little baby Victor Brantanaris, isn't that cute? Of course, being on boats, as always. Fishing, of course, Derek already mentioned, we always were fishing until we were so successful in highlighting the natural importance of the reefs that we were studying that they eventually made them into no-take marine reserves. I think I thank Juan for that, for Uva. It was really great yellowfin tuna fishing that we're no longer able to do. Drinking rum was very important. Playing poker, as Juan mentioned as well, and eating were all very important. There's Adrian Correa, I'm very excited about this meal. And of course, I think the thing we did most and is most memorable is sorting those damn rubble bags. I don't know where Peter got this idea that we should look at this, but actually fully like a quarter of my pictures are people sorting rubble bags, crouched over. So we all had to do our part. And the best thing about Peter is that with him, everything was full care and service education. You know, he raised you from the cradle to pushing you out and did everything for you. And here he is feeding me in Panama. Very nice of him. And so I thank him so much for what he's given me, as we all do. And I'm very honored to be here again to talk to you about Peter Glynn and share a little of his work and life and the inspiration that goes on and continues on after him. So thank you very much. Thank you, Peter. Uh-oh.
Thank you. One cruise, we were out on St. Patrick's Day, I seem to remember, and there was some celebration going on. And you notice we had inflatables. We were usually diving and working off the inflatables. And I remember we got some Irish music going, and Tyler jumped onto one of the inflatables and did some amazing river dancing.
I call it transom dance.
I just want, do you still do that?
Less frequently, yeah. I tried to find a picture of that, but none exists. No record exists of that. So there were other salacious ones I didn't put in.
Thank you, Tyler.
Thank you.
If there are no more questions, we'll move on to our next speaker. Our next speaker, Ian Enox, was the best man at my wedding. Ian is not only a close personal friend of mine, he's also become my most valuable colleague over the years, and we met in Panama doing sorting rubble bags for Dr. Glynn.
It's on? Yep. Thank you, Derek. That was me, I think. Not number 9, it was up. Perfect, thanks. It's a privilege to be able to talk to you guys here today. My name is Ian Enox. I have the distinct honor of being the last student of Dr. Glynn. And you're going to note throughout this Ph.D. student, Victor, I'm sorry. Throughout this talk, I am going to be referring to him as Dr. Glynn because he's a man that has given me so much and is really responsible for everything in my entire career and so much of what I have. And he has he's so incredibly humble. But I can never, and I think as Derek kind of mentioned, I can just never escape seeing him as Dr. Glynn. And I see this all the time with some of his students. So I just want to put that out there. My introduction to this world was when this gentleman on the right handed this young child on the left a copy of Marjorie Riaka Kudwa's chapter on coral reef biodiversity in her Biodiversity 2 book. And in that chapter, it discussed how if the majority of biodiversity, metazoan biodiversity on a coral reef, or rather in a rainforest, are the insects, then the analog for coral reefs are in fact the cryptofauna. And this is a multi-phyletic assemblage of organisms, including all sorts of crabs and shrimp, and as people have talked about in prior. Thanks for stealing my image, Juan. These are organisms that really support that huge amount of biodiversity that we actually cherish about coral reefs. And this understanding and drive to appreciate biodiversity is something that was continually reinforced with me during frequent fishing expeditions with Dr. Glynn and biodiversity fishing, so getting as many different species as we could. And I think I found about 5. It's amazing, actually, how many pictures I don't have of us because we were always working, but then I ultimately come to just fishing pictures, so it's pretty funny. One of the brilliant things about Dr. Glynn is he can see a pile of rocks and start an entire field. We've seen, people talk about bleaching. We'll be hearing about physiology, coral physiology. We've been hearing about, interactions, small-scale interactions of invertebrates. But really, to be able to look at this and start an entire field of looking into biodiversity on these reefs, and this was my first actual real introduction to coral reef biodiversity, and these are the so-called infamous rubble bags that Tyler mentioned. And they're infamous because it took so much time to sort through all of this dead rubble, breaking these down into trying to identify them to the species level where possible. And this is something that we've spent hours upon hours, months, and years, in fact, doing. Now, the Eastern Pacific is ideal for this type of work because it's surrounded by the Eastern Pacific Barrier, obviously on the left here, in the west, and the Americas on the right. And this creates a fairly depauperate fauna which is ideal because if you're going to be studying an obscenely biodiverse community, you want to start in the most simplistic system possible. At the same time, the reefs there are structurally complex, formed primarily by Poecilopora damicornis, allowing for abundant crypts, cracks, and crevices for these organisms to occupy. And they're also fairly horizontally homogeneous, meaning that we could undertake replicate sampling over the extent of the reef. Now, one of the most amazing things about Dr. Glynn, and this has been subtly hinted at several times, is the amount of trust that he puts in his students and in his employees. And that trust is absolutely terrifying. And it was with this in mind that we set out to Isla Contadora. This is Dr. Lauren Toth and Victor Brandt Neris. And to this house, Casa Rosada, of questionable proprietorship. And we set up shop on this island with a lot of help from Dr. Juan Maté, and started to really investigate in detail the biodiversity of this cryptofaunal community. We also had some fun, too. So we collected a huge number of organisms. This graph, you can kind of see the black lines show the total number of individuals, and the green and red are the proportion in which they are associated with live and dead coral substrates, respectively. And you can see that there was a huge number of species that we had to actually identify. And that most of them were associated with these dead coral habitats. Now, those that were associated with the live were incredibly abundant, relatively more abundant, and ultimately led to higher amounts of biomass and enhanced trophic potential. And this is because if you are specialized enough to get past the wall of mouths that is a coral colony, and you're able to actually live within this potentially inhospitable environment, there's all sorts of abundant food for you to actually utilize. And this was something that we were able to look at. We also capitalized on those, the different zones on this reef and the horizontal homogeneity by sampling different types of rubble and reef framework in several different levels of degradation. So we looked in the most intact here on the left of the screen of intact coral frameworks. And as we proceed to the left, you can see like medium degradation framework and high degradation framework and all the way to rubble. And we conducted rarefaction on these communities and actually found that the highest amount of biodiversity was associated with the most degraded, the most eroded actual substrate materials, the rubble. And this seems on face value kind of counterintuitive in that this is something that's not classically considered to be the most valuable part of a coral reef. But in fact, it makes a lot of sense because this is the area that you have huge amounts of heterogeneity with respect to the food sources and habitats that are available. What was also quite interesting, and I should say that the numbers of species associated with these decreased as you went towards more and more intact environments, which also makes sense because you get competitive exclusion in some of these areas. What's also very interesting is that we found the highest amount of biomass of this community in areas that were of intermediate levels of degradation. And so if you look at this and you want to manage a reef and understand what, to maximize the ecosystem services of this reef, you really need to target two different types of habitat for these two different ecosystem services. Now, I have pivoted, like many of us have, in terms of my focus on my research. But no matter where I turn, There's always Dr. Glynn that has always, kind of created whatever field I'm looking at. And I use this figure all the time, which is from one of his chapters. I now, most of my research involves bioerosion and calcification, looking at the permanence of reef structures, which I would say are critically linked to ocean acidification, Alina. So this is something that it's been really nice to and also been very pioneering in terms of all the work that Dr. Glynn has contributed. So more recently, I've been collaborating with Lauren Toth and Rich Aronson, and we've actually put out these pieces of Porites lobata on the reef in these two different gulfs of Panama. And we've used, which are typified by an upwelling, different conditions with respect to nutrients and thermal conditions and ocean acidification conditions. And we use CAT scanning to look and see how these different little pieces of habitat have changed over the amount of time that they've been there. And we're able to, using this, actually isolate out the different influence of the different invertebrates that ultimately affect these habitats. And so this is an ongoing project that we're currently working on, and it's been really nice to kind of be able to come back to the Eastern Pacific and where I really was able to start. But I think one of the biggest and greatest legacies of Dr. Glynn is the community of people that he has introduced me to, and the people that are my amazing friends and colleagues. We've had a lot of really amazing adventures, and we've produced a lot of really great work. I recently went through went over my CV, and a full 93% of my publications have a Glynn student as a co-author in them. And I think that is extremely telling, because it shows how competitive and the drive behind all of his students, but it also shows how much they work together and cherish each other's contribution to the field. So if there isn't a better legacy, I don't know what it is. So thank you.
Thank you, Ian. We have time for questions if anyone feels so inspired. All right. We'll go ahead and roll on to our next speaker, who is Monica Medina from Penn State University. Monica is oh, that's right, she's an Apple person, making things difficult for us. Monica is world famous for her work in genomics.
Oops. I stole my thunder. There. Okay. Well, it's a great honor to be here honoring Peter Glynn. I recognize many faces, and I'm finally able to put faces to names, so it's fantastic. Those of you that know me know that when I was a student at RSMAS, I didn't work on corals. I was working on aplysia, actually. Doing genetics on aplysia, but the coral reef group was so large and influential in general that it really drew the rest of the students. So I participated in a lot of expeditions, both with Alina and Peter, and I was always ready to be the volunteer diver to go help out. So that really influenced my career as a coral reef scientist years later. And I want to thank you both and all of you for that. So even though I wasn't Peter's student, I have some anecdotes I'd like to share. I won't be here tonight, so I'll share them now. When we went on some of the cruises in Panama, there was the cook Lucho was his name, right? Luchito. And Lucho was always catering to please Peter. So he had a bucket full of candy bars right on the deck as we got out of the water. And I always miss that from that moment on. I haven't been on any other expeditions where you could get out of the water and have a Milky Way and M&Ms and all kinds of stuff. And one night we counted how many candy bars Peter ate, and it was 18. Nobody could beat that. So also when I was a student here, one of my committee members was Jack Fell. And because I was working on Aplysia californica, he said, before you defend, you need to read the log of the Sea of Cortés. Otherwise, I won't let you defend. And you need to tell me where the sea cow ran aground. And because of that, I started on this journey of reading John Steinbeck. I'll get to Peter soon. So I started reading John Steinbeck's books. And on one of the cruises, I think I had just read Cannery Row. And Cannery Row was a very different place in Pacific Grove next to Hopkins Marine Station. And before Cannery Row became what it is today, this very posh, fancy place, it was a working class seaman kind of place. And there was a place where ladies of reputable fame lived, and there was a time where Stanford Hopkins students lived there. And I asked you, and I don't know if you remember this, but you said that that you lived in one of these dormitories where the ladies were transitioning out and the students were taking over the space, right? So I was a professor, I was a scientist and a professor in different institutions in California, and we had to go to Pacific Grove to do our DSO dives. And I wandered those streets and wondered all the time what was the place that was your dormitory and where the ladies were. So I do have some fond memories of you.
Monica?
Yes.
$25 a month was the rent. That was the big reason. Yeah.
All right, so I'm going to tell you a story. I was going to tell you about coral bleaching, but Andrew discouraged me from geeking out on too much genomic stuff. So I'm going to tell you about a more recent type of work that I've engaged in. I'm originally from Colombia, so this is a very special project to me. And it has brought me in new directions that I think is always fascinating in someone's career. So this reef is called Varadero. It's in the Cartagena Bay in the Caribbean, Colombian Caribbean. And it's a very small reef, but it has amazing coral cover. And it is the last reef standing in the bay. So this is a map. North is here. This is a map of colonial times, and that's what makes this reef really, really fascinating, that we have a lot of good history. Here is Cartagena. This is the bay. This is Tierra Bomba Island. This no longer exists. This is called Boca Grande, Big Mouth, and this is called Boca Chica, and Varadero sits right here. What I want to highlight is this, the Canal del Dique. Cartagena was the most important city in colonial times before the ships did the crossing. They went to the Antilles and then they did the crossing. So it was very important to protect the city. And they turned the Magdalena River, which is a very torrential river that comes down the Andes, into the Cartagena Bay. And they started to change the bay from 500 years ago. However, let's see what's next. That changed more dramatically in the 20th century when the Colombian government made this channel an irrigation channel, and that's when really all the reefs started to decay. And I'll show you an image, satellite image of that. But in 2017, something else happened in Colombia. and it's the peace accord between the FARC and the Colombian government. And that triggered unprecedented development, non-sustainable development that is affecting ecosystems nationwide in very, very dramatic ways. So one of the things that is happening in this region in particular, now you see the right orientation, north is here, this is Boca Grande, this is Boca Chica, this is the Dique Channel. So there is a lot of turbidity being dumped into the bay, and all the reefs have disappeared, except for this reef that sits right here. So now you see here Tierra Bomba and the Barú Peninsula, and this is where the reef sits. And the port has grown so much that they want to expand the channels, the navigation channels, and they want to dredge the reef. So if you look at, if you zoom out, what you see is then again the Dique Channel here, this is Varadero, and then this is a marine protected area, the Rosario Island. So we have a gradient of turbidity. If you look at the optical properties of the water in Rosario, you have a similar light availability in Varadero only at 3 meters of depth, whereas in Rosario it's at 12 meters deep. This is work that I'm doing in collaboration with Roberto Iglesias Prieto, who's a physiologist, coral physiologist, and very good optical person, which I'm not. But we've been focusing in the genus Orbicella because this is the species that I've done a lot of genomics work on, so we have a lot of tools developed for them. For those of you that don't know them, Orbicella annularis is a shallow species. This is a close-up of them. Fabiolata has a more broader distribution range from shallow to deep, and then the Franksa you find in the really deeper waters of a reef. And in Varadero, you find them really compressed. So I'm not going to show you a whole lot about the optical properties of the water because it's not my comfort zone. So I'm just going to summarize this work that Tomás López, who's Roberto's student, is in final stages of publication in communication biology, where they examine very carefully the light properties of the water, and what we see as a reef that is thriving is actually probably not the case, because the light availability that is conducive to good reef survival, and these species in particular, is compressed at the top. So what you see here is the distribution of the three species, in this case in Rosario, the marine protected area. So you see the annulares, the Orbicella, and then the Franksa at 30 meters deep, whereas in Varadero you only find annulares and Fabiolata, and the Franksa have pretty much gone extinct. So if we continue on this path, it's very likely that the reef is going to disappear. Even though, because we have been so vocal, and this has engaged the local communities and activists, we have been able to delay the dredging. So the dredging is not happening just yet, but if there is no modification to the Dique Channel, the reef is not going to have much longer. So we also did a reciprocal transplant, and this, what we call Cartagena Bay, is at the mouth of the Dique Channel. We transplanted Orbicella fabiolata from Varadero to the Dique Channel mouth, and then from Rosario to the Dique Channel mouth, and then we did reciprocal transplants. This is the work of one of my students, and we've been studying coral microbiomes, coevolution between corals and their microbiomes all over the world using the coral phylogeny to look at this. So we decided to look at the Fabiolata microbiomes in more detail here. And this got recently accepted for publication in the special issue that Coral Reefs is putting out for exceptional reefs, or I forget the name, the Reefs of Hope or something like that. So you can keep an eye out for that there. So what is interesting, even though this is not statistically significant, unfortunately, what this slide summarizes is that when we transplanted, it didn't matter where they came from, the corals, that went to Varadero, from Varadero to Varadero and Rosario to Varadero, the survival was higher. And I think this has to do, I didn't explain that in a lot of detail because I don't have a whole lot of time, but what Roberto has found is that the location of the reef and why he thinks it survived is because it's right where oceanic waters are coming in and the decay water channels are coming out. So there is a lot of fluctuation in light quality. So they have a break, and they also can do a lot of heterotrophic feeding. So they're actually thriving in that small window of good light. So we did this a couple years ago, and the transplants are still alive, only in Varadero though. So when you look at reef microbiomes, this is a bit complicated slide, but I'll walk you through it. This is a PCOA analysis. What you see here in the larger oval distributions is we sampled right when we collected the fragment, then we let them acclimate to the location of the reef transplant, and we sampled again, and then we sampled 6 months later. So that's what all of this is, the mother colony and the initial pre-transplant samples. So and these are the microbial communities. In each one of the dots is a sample or a coral fragment. So there is a big spread and not a whole lot of overlap between locations, but then when you transplant them, they all kind of converge to a very similar microbiome. And then if you zoom into this plot, what you see, I think is also very interesting, is that you do have a location-specific microbiome, so they converge to an environmental profile that selects for specific microbial communities. And when you look at species richness, this is alpha diversity. What you see here is that a decay channel, the species richness is greater, which we kind of attribute to what is now known as the Ana Caterina effect. That is a paper that was put out by Becky Begard-Herber's lab, where they say that all happy families are all the same, but all dysfunctional families are different in their own way. And so if you have a very stressed environment or stressful environment, you're going to have microbial communities that are opportunistic, microbial species that can be pathogenic, that also colonize your coral fragment. So I think that's what we're seeing here. So to summarize, the prospect for Varadero, unless there is some change in policies, first, it can be dredged, but also it's not that the reef is really thriving. It's having a progressive death in the deeper zones. And I would like to thank my collaborators, Roberto. This is a project funded by NSF and the Colombian colleagues, and then all the lab members in both labs that have contributed to the research. I'll be happy to take questions.
We have time for questions, if anyone's. So ask any.
Hey, thanks, Monica. Do you think there's a transition where you're going from microbes to cryptofauna, where something switches, and when you get these larger corals where you have high diversity? I'm seeing a lot of high diversity of the cryptofauna microorganisms, but that's not necessarily a good thing for the corals. Can you sort of speculate a little bit on that transition zone where there may be a flip?
So not only at the microbial level, but like microfauna?
Right, but so you might have very low diversity microfauna when you've got a healthy, lots of corals, for example. So this whole thing could be misinterpreted somewhat, because you've got lots of diversity, but you haven't really got this thriving reef.
Yes. And in the case of microbes, we've been looking at a lot of coral diseases, and we see much greater microbial diversity in all the diseased corals. I don't know in terms of larger organisms. It's not my I don't have data on that, so I can't speculate. It seems like you do. But I don't know how that, in the case of deceased corals, how that's happening. You know, the microbes stay there until the end. I don't know that all critters necessarily stay when a coral dies, right? So there is an earlier migration out of the host, whereas the microbial species just turn over in composition as they decay and chew on whatever remains. Yeah. Thank you.
If not, we can move on to our next speaker is Rich Aronson, former president of the International, what is it called now? Coral Reef Society. Talking to us more about reef development in Eastern Tropical Pacific.
Thank you. I can think of no greater honor for a scientist than to have somebody go out and test his hypotheses, and that's what I'd like to do for you today, Peter. But before I do that, just a little bit of personal stuff, and that is that I came to the Eastern Pacific and I came to Peter Glynn's mob much later and much more obliquely than the other people who have spoken today. So I want to just say a very little bit about that. I knew Peter kind of, sort of, and I had read, of course, read all of his papers, and I thought, geez, I wonder if I'll ever get to the Eastern Pacific. And it was a chance trip that Harris Lesios at STRI was able to organize for me and Peter's great buddy Ian McIntyre out to Uva Island that got me out there to see Peter's old stomping ground, and it's really quite a fortuitous place to do your work because, of course, there is this incredible upwelling gradient with which you're all familiar in this room of strong upwelling, at least during the winter season in the Gulf of Panama, then intermediate upwelling over here along the Ushuaia Peninsula, and then weaker upwelling in the Gulf of Cherokee, and you know, a lot of people say there's no upwelling, and I think that's to some extent a semantic issue because there certainly is thermocline shoaling, and so it's there, but it's weaker. So I was able to get out there, and I started mucking around. I thought, oh man, I better start working out here, and not that long after that, Peter did me one of the greatest professional favors that anybody's ever done me. He probably doesn't even remember it. In fact, I'm sure he doesn't remember it, which is that Lauren Toth was his undergraduate advisee, and she came into Peter and said, so you know, what should I do about grad school? And he said, well, you should call Rich. And so that was the beginning of a collaborative relationship that's lasted many years, and so Lauren, of course, is certainly a co-author of this talk. So Peter worked a lot in the Eastern Pacific, of course, and was trying to explain what was going on there. And in the 1970s, he and Ian did a bunch of probing on the reef frameworks that exist out there along in the islands in both the Gulf of Panama and the Gulf of Chiriquí. So along this gradient of upwelling, from strong upwelling in the Gulf of Panama to lesser upwelling in the Gulf of Chiriquí, and what he discovered was that the bulk rates of accretion, reef accretion, that is to say, how long it took these reefs to grow up, were slower in the Eastern Tropical Pacific than they were in the Caribbean. As a related but somewhat separate matter, in the early 1980s, he was the first one on coral bleaching, and he witnessed this mass coral reef bleaching event in the Eastern Pacific, and he identified two climatological causes of slow reef accretion, putting these two things together, and they've served as guiding hypotheses for the ways in which we think about reefs of the Eastern Pacific, or at least the ways in which we try to approach them. And first is that strong ENSO events at decadal to multi-decadal scales suppress coral growth and therefore vertical accretion. And this is, I'm taking some liberties with the second hypothesis. It's not exactly, I think, the way Peter framed it, but lower water temperatures suppress coral growth during the upwelling season, more so in the Gulf of Panama than in the Gulf of Chiriquí. So we went out to try to revisit these hypotheses, which of course are living hypotheses. And again, I think there's no greater honor than to have somebody run around and try to show that you're wrong. And so that's what I'd like to talk to you a little bit about today. So starting with the first one, strong ENSO events at decadal to multi-decadal scale are responsible for poor reef development in the Eastern Pacific. Lauren and I, kind of separately and in tandem, have cored several reefs along this upwelling gradient, one at Contadora, an old stomping ground for Peter and his students, Isla Iguana, and then over at Canales de Tierra and in that sort of Coiba area along this very nice gradient. You've probably seen these photos before. This is Ian McIntyre and Steve Vollmer and me coring at Iguana. A number of you have had the pleasure of doing this before. You take a big irrigation tube, you cram it in, pound it in, work it in with handles, put the cap on, and then the work begins because then you have to pull it out against the pressure head. But having done all of that, and in very brief, we identified a hiatus in reef development starting around 4,000 to 4,200 years ago. So reefs which were dominated by Passalopera, at least in the zones we were coring, were going gangbusters, and then they stopped, and then they were going gangbusters again. And this little layer of crappy-looking corals, taphonomically degraded corals, crappy-looking. I know we're being filmed, and I thank Derek actually for using a lot of swearing in his talk, because I thought that was off, but I guess I can do that too now. And I love to do that, being from Queens. But anyway, there was this very thin layer of crappy-looking coral, and if you did the radiocarbon and uranium dates, it turned out it was an enormous interval. It was 2,500 years, which is a lot, you know. And the accretion rates of reefs go down dramatically. No big shocker, right? It takes up a lot of time, and it's a very thin layer. And it's true along the entire upwelling gradient, although there are differences in the length of that hiatus along that upwelling gradient itself. Now, so to the question, do reefs of the Eastern Tropical Pacific accrete more slowly than reefs of the Caribbean? And this is Lauren's analysis. The answer actually is when times are good, nuh-uh, they don't. So this is, you can see this is our work, and then Galeta, and then some earlier work I did in Bocas del Toro, Bahia Almirante, and then other work that Bill Precht and I did on the Belizean Barrier Reef, the central portion of the Belizean Barrier Reef. So when times are good in Pacific Panama, they're accreting just as rapidly. So basically, what does this mean? Well, reefs of Pacific Panama experienced this 2,500-year shutdown, and the causes are related to changes in ENSO variability, and Lauren's going to talk about that in her talk, which is coming up next. The reefs in both Gulfs grew as rapidly as Caribbean reefs, apart from this 2,500-year shutdown. So poor reef development, which is to say low bulk accretion rates in this part of the Eastern Pacific, that is to say off the Panamanian coast, turned on this ENSO-driven hiatus that covered about 40% of the Holocene history of these reefs. And so rather than a multi-decadal scale shaving back of coral reefs, there was this enormous millennial scale hit that knocked these reefs back and was responsible for the sum total of slow accretion. So Peter was correct about the importance of ENSO in limiting reef development, but the tempo and mode of ENSO action on accretion are different from what any of us expected. This was just a complete shock to me. Okay, second thing, lower water temperatures during the upwelling season suppress coral growth, more so in the Gulf of Panama than in the Gulf of Chiriquí. So here's what Peter found earlier on. Rates of linear extension of Passalopera, the dominant coral out there, were faster in the Gulf of Chiriquí than in the Gulf of Panama. And so we went out to revisit all of that. Reefs in the Gulf of Chiriquí, where upwelling has been less pronounced during the Holocene, that we know from geochemical data as well as from data on the modern, stopped growing later and recovered from this hiatus sooner than reefs in the Gulf of Panama. So that fits together with differences in the growth of coral. That all makes sense. And the question is, are corals and coral reefs in the Gulf of Chiriquí, will they continue to fare better than reefs in the Gulf of Panama as the climate warms? Now, it would be pretty damn stupid of me to put up this question if the answer is yes. Obviously, the answer is no. And I want to show you some recent data that we have, which in no way negates Peter's findings, just that things are, of course, changing. Well, here's the climatology. Both of these gulfs have warmed significantly over the past 150 years. The Gulf of Chiriquí is warmer, and it is warming faster than the Gulf of Panama. And the Gulf of Chiriquí is also becoming more variable in temperature than the Gulf of Panama as time goes by. And this is a plot of Peter's original data and some data from Derek and our data. So our data are in the colored bars. The dashed horizontal line for the Gulf of Chiriquí is Derek, and then those vertical black bars are Peter. And what you see is there's some changes going on in both the Gulf of Panama and the Gulf of Chiriquí. Gulf of Panama, coral growth seems to be increasing, linear extension seems to be increasing. It seems to be going down in the Gulf of Chiriquí. This is a plot that Derek published in Coral Reefs in 2010, showing a decline at Uva Island, about which you've heard rather a lot already this morning. So this is a decline in linear extension rate. And then out at the right, those are our data for 2016 to 2018. We're right on the curve there. Coral growth is declining in the Gulf of Chiriquí. So Gulf of Panama, higher than historical growth rates in the fall and winter, and possibly higher than historical growth rates during the spring and summer, which is the non-upwelling season. Gulf of Chiriquí, the opposite, lower than historical fall-winter growth rates and possibly lower than historical spring-summer rates. Finally, these are the same data plotted in a slightly different way, and what you can see is that coral growth is now as fast or faster than in the Gulf of Panama. And I see a sign's about to come popping up here, so I'm almost done, not to worry. 3 minutes, good. Okay, so I was going to give a little squeal there, I kind of did, Until recent decades, coral growth was faster in the Gulf of Chiriquí than in the Gulf of Panama, and it's now reversing. Climate change is at some point going to make it too hot for corals in the Gulf of Chiriquí, but warming temperatures in the Gulf of Panama are now attenuating the impacts of upwelling. So upwelling is now offering a temporary, it is going to be temporary, refuge from the thermal impacts of climate change, which is to say bleaching because of high temperatures combined with high light intensities. So ecological conditions have changed substantially since Peter took those original data. And again, this is not in any way negating or falsifying what Peter found. It's just saying, okay, this is something new, and this is what's happening now. So just to recap, strong ENSO at this decadal to multi-decadal scale suppresses coral growth, Peter's original hypothesis. The tempo of variability in vertical accretion is actually multi-millennial, not multi-decadal. And the mode is response to millennial scale rather than decadal scale changes in ENSO variability. Fair enough. Lower water temperatures during the upwelling season suppress coral growth, more so in the Gulf of Panama. That's changing. Situation is now reversed. Rising temperatures are suppressing coral growth in the Gulf of Chiriquí, and the Gulf of Panama is now a temporary refuge from coral bleaching. And I just want to end this up by paying respect to Peter. Peter's pioneering work on coral bleaching has set the tone for decades of research, and for that I thank him. And his observations have led to these really important hypotheses. New generations of scientists, I'm not in the new generation of scientists, I mean newer generations of scientists, continue to search for the questions he asks. And again, that is, I think, the greatest honor that a scientist could ever have. So thank you, Peter, and thank you all.
We have time for a few questions.
Okay. Mark.
Yeah, you have this millennial gap where there's a reduced reef accretion. How can you separate out what you're seeing in terms of reduced coral growth versus a massive amount of bioerosion that may have taken out a lot of the record?
Yeah, bioerosion, it just can't account for it, and I think Lauren can knock that one off. I mean, we've fretted about that rather a lot. Yeah, no, I'm just gonna let Lauren do it because I don't want to deal with it. Yes, sir.
In terms of the change in upwelling, there's also a huge halocline involved there. So for long-term change, how much is it changing over time? Because the thermocline would be very different if you didn't have the halocline in the Gulf there.
Mm-hmm. We haven't even considered that, so thanks for that. Got to look into it.
I mean, I think if the waters were getting saltier, that would probably be good for the corals because you have low saturations. Not paying attention to my answer.
Yeah.
But you have low saturation state and low salinity, so if it was getting saltier, I think it would actually be a good thing.
Yeah, who knows? I mean, it's hard to say. All right.
Okay, we can move on. Our next speaker is Lauren Toth. I actually met Lauren in 2004 at a house party in South Miami. And today I'm honored to call her a peer and my friend and colleague.
All right, thank you. I have to say I was pretty intimidated when I was thinking about giving this talk because I know I haven't known Peter as long as many people in this room, and I haven't worked with him as closely as many people in this room, but I think his real legacy and the reason he's so special is because he really touches everybody who he interacts with, and he certainly helped shape the path of my career, and that's what I'm going to talk about today. So like Derek, when I was first thinking about Peter's influence on my career, I thought back to his intro to marine biology class. And I thought, okay, I'm gonna look back at my notes and I'm gonna find some like fantastic piece of wisdom that I wrote down, in the margins that he might have said. And Peter, I'm sure you said lots of really inspirational things. I don't think I wrote them down. I wrote down a lot of tremendous notes, and it was really great to go back and reflect on all I had learned in that class. What did stand out in my notes, which I'll highlight here and I'll blow up for you in case you can't see it. I credit Peter with a lot in my career, but I guess the one thing I can't credit him with is inspiring me to get into geology, because even by Coral Reefs Part 2 of that class, I was pretty sure I was not going to be a geologist. And of course, I'm now a coral reef geologist working at the U.S. Geological Survey. So that changed at some point, maybe not because of Peter. After my undergraduate career, like Ian talked about, had the fantastic opportunity because of Peter's support to live and work on Contadora Island. And this was really one of those turning points in my life, and really a moment that shaped the way that I think about science and set me on the path of my career. Not only did I get to collect some of my first coral reef cores there with Rich and Ian McIntyre, finally dug into geology a little bit at that point, but I think even more importantly than that, like many people in this room, I just had the opportunity to spend a tremendous amount of time out on these reefs, learning about them and seeing them every day, and just really immersing myself in this incredibly unique environment. And at that same time, I was also studying all of Dr. Glynn's papers. And as you can tell from my obsessive highlighting on these papers, I found almost everything in these early studies to be tremendously important to the work I was doing. And I promise I don't highlight all papers like this, but when I was going back and looking through my folders of Dr. Glynn's papers, pretty much all of them have this insane amount of highlighting, because all of that information is just so important. And really, what I took away from reading all of those papers and later my experiences on these reefs is really just how dynamic and how high variability these reefs can tolerate. This is an incredibly unique environment, and it's actually pretty amazing, as Rob Van Wosik said to me a lot during my PhD, that corals live here at all. These are corals that experience tremendous swings in temperature, changes in nutrients. Why are there even corals living here? And yet, in some places, there are tremendously healthy coral reefs with really, really high coral cover. And not only that, there are these tremendous geologic frameworks of reefs in these areas that have built up over thousands and thousands of years. So at least for many millennia of their history, despite all the disturbances in these environments, reefs have been able to survive. We know from Rich's talk that though, wasn't always the case. There was about a more than 2,000 year period in the history of these reefs where things weren't so good for the reefs in Pacific Panama. And a lot of my PhD work was focused on trying to dig into what some of the causes of that decline would be. And it was really all of Dr. Glynn's work making observations about the bleaching events in 1982-83 and 97-98, and thinking about how dramatic a single climatological event can be for a reef ecosystem. And I know that many scientists hadn't observed a widespread coral bleaching event when Peter made his first observations in the early 80s. But I can say I personally had not observed a mass coral bleaching event and the impacts that it had had throughout my PhD and really until very recent years. So I relied really heavily on these observations to shape my thinking about how these reefs may have evolved over millennial timescales and what disturbances would be enough to shut down a coral reef for 2,000 years. And so, these events were really dramatic, and, not only did they cause the loss of coral cover, but they also took a long time for these reefs to recover from. So thinking about just how dramatic these events were in recent decades, I started to dig into the literature of what may have caused this decline in reef accretion in the past. And so very broadly speaking, if you look at the climate literature, the time when these reefs first started growing was a period of lower INSO variability. But INSO variability started to kick up right around the time that these reefs shut down. So it started increasing over that period. We did some geochemical reconstructions from the corals that we collected in the cores. And those showed that right at the onset of this reef shutdown, there were cooler temperatures and there was a rapid increase in upwelling. So we took that along with other studies that supported it to suggest that the onset of this reef shutdown was actually caused by La Nina. So the opposite of what we've observed in recent decades, but again, another climate event that can push these reefs past their very small envelope of tolerance into a state where they're no longer growing. After that time, there was a whole lot of climate variability. Not only did you have more La Nina, you had stronger and more intense El Nino events. All of this came together to continue to suppress the development of these reefs for about 2,000 years. But then reefs came back, which I think is a hopeful story and gives some hope for our future of coral reefs now. Our geochemical evidence suggests that there was weaker upwelling, which is supported by this idea that this was a period of less frequent La Nina events and probably less strong El Nino events. And all of this suggests that this transition between a state of reef growth and reef degradation is really underpinned by climate variability over these millennial timescales. So not only is it, as Rich said, impacting reefs over decadal timescales, it's impacting them over millennial timescales as well. One of the next questions we wanted to ask was, did this change in ENSO variability impact reefs elsewhere? And the first thing that we found was Jorge Cortés's work, where there's the hints of the same hiatus in Pacific Costa Rica, but also some further away places. There's hints of the same sort of timing of reef shutdown. So some super high latitude reefs in Japan, some really turbid inshore areas of the Great Barrier Reef, and some areas of Hawaii that experience really strong wave action. And what all of these places have in common is these are places that are already marginal environments for reefs to grow. And so it maybe didn't take very much to push those reefs just a little further past their tolerance into a state where you could have these prolonged shutdowns. And so I think these marginal environments can be really informative for thinking about the thresholds that reefs have to climate. This work then took me to where I am now at the U.S. Geological Survey, where I've mostly been focusing on reefs in Florida, which is another marginal environment because these are high latitude reefs. They experience broader swings in temperature than most of the Caribbean. And these are areas where you get periodic cold snaps that suppress reefs. Obviously now we only think about warming, but in Florida, cold snaps have been a really important part of the history of the reefs there. And like the Eastern Pacific, most of the reefs in Florida are poorly developed compared with the broader Caribbean. Most of the reefs are much, much thinner. So if we compare that to the reefs in the Eastern Pacific, you see, again, not as much reef has accumulated in these areas than elsewhere. And we have a whole bunch of cores that not just I took, but a whole bunch of folks took over the years that we recently compiled to look at accretion rates through time in Florida. And the trend in reef accretion throughout the Florida Keys looks like this. So there was a peak in the accretion of the reefs around 7,000 years ago, but it's really dramatically declined since this time. And we compared these data to the rate that sea level was rising at any particular time in the history of these reefs, and looked at where reefs were not keeping pace with sea level, because that should be kind of a minimum benchmark for how a reef should grow. And we found that by about 3,000 years ago, the majority of the reefs were no longer keeping pace with sea level. And in fact, if you were to knock off the surface of most reefs in the Florida Keys, they would date between about 3,000 to 4,000 years old. Again, we hypothesized that this had to do with climatic changes. So this is just a global climate anomaly, just showing broad-scale changes in temperature over the Holocene, where we go from the Holocene thermal maximum, a period that was slightly warmer than the recent past, not as warm as it is today, to a period of climatic cooling. And I'm not showing the data here, but this is a global temperature anomaly. But we actually have some preliminary geochemical data from corals in these cores that supports the idea that there was about a 1 degree cooling in South Florida over this period. So the hypothesis here is that Florida sits in the subtropics. We know now it's an area that gets these periodic cold snaps. That was probably less frequent during the Holocene thermal maximum because temperatures were just slightly warmer, putting them in a more optimal climatic envelope, and then they got pushed out of that envelope by very slight climatic cooling. Obviously now, anthropogenic warming is the dominant problem on reefs in Florida and around the world. And this has caused a further shift where we're starting to see these reefs begin to erode. This cooling of climate during the Holocene also impacted reefs further up the coast. So this is some recent work that I've been doing with Bill Precht and others, looking at the Holocene history of reefs in southeast Florida, so going up from the Florida Keys a little bit. And don't pay attention too much to the details of these data, but these are a bunch of radiocarbon ages from acroporid reefs throughout the region. And what I want you to take away from this is that between about 10,000 and about 8,000 years ago, we had reefs in Florida that went from the dry Tortugas all the way up the coast to southern Palm Beach County. But after about 8,000 years ago, reefs were no longer found in Palm Beach County. By about 6,000 years ago, we only had reefs extending up to Fort Lauderdale area, and by about 3,000 years ago, and these are, again, growing reefs. We had corals throughout all of these reefs. But by about 3,000 years ago, reefs were only growing in a couple places in the Dry Tortugas and a couple places off of Miami. And after that, reef accretion had essentially shut down. So to summarize, Peter Glynn's observations demonstrated to me, and I know to a lot of people, that climate can be an important factor controlling the development of reefs over decadal timescales. But our recent work has shown that this also is a major control of reef development over these longer millennial timescales. And this is especially true for these marginal reef environments. I think that's why places like the Eastern Pacific are so unique, because at those extremes, you can really observe these processes happening in real time in a way that you maybe can't observe on a reef that's more buffered from these impacts. A lot of my work and a lot of work from the geologic record shows that in the past, where we often see the historic collapse of reefs is due to cooling events, due to La Nina in the Eastern Pacific and due to large-scale climatic cooling. But warming is having a really similar impact today. And I think we can still learn some lessons from these historic reefs that can help us identify what those thresholds are. How much does it take to push a reef outside its optimal envelope to a point where it's no longer going to grow? And with that, thank you. But I especially want to thank Dr. Glynn for inspiring me and for, I think, inspiring so many people. So thank you.
We're all done. We're right on time. If you want to ask Lauren questions, you can do it now or at lunch or whatever. We're eating lunch at Salt. I don't know if Andrew, you want to take the reins back.
First, we do have some questions, time for questions for Lauren, if anybody has it.
Yeah. Repeat your question? Oh, right. Rich passed that off to me. No, so we did look at we looked at the depth to which erosion penetrates into the Posilopera framework and kind of used that to extrapolate out how much erosion could do over these long time periods, because there's kind of a horizon where erosion stops penetrating. And that turned out to be only 200 to 300 years worth of the reef framework would disappear, but not enough to account for a more than 2,000 year gap.
Yeah, just to add, these reefs they're loose coral will packed in sediment, and it's just packed to the point where live rivers just
Okay, thanks, Lauren. So just a quick couple of announcements. So we are heading into a lunch break now, which is a full hour and a half, so you should have plenty of time to catch up. Lunch is going to be in Salt's restaurant, which for those of you who don't know is if you go straight out of the auditorium and through the glass doors and follow your nose through, you'll find it. Everyone, I think, will be heading there. Lunch is a buffet style with a variety of different options, so you should find it all set up when we get in there. Dinner, however, this evening is a sit-down affair. A few people have asked if there are still any spaces left, and unfortunately, because we had to sort of hand in our numbers ahead of time for a sit-down event, there's currently no extra spaces. But I'd encourage people who come to the reception, and of course everyone's welcome to come to the reception, which is at 5:15. We may try to find ways of slipping people in to see the speeches for the dinner without necessarily supplying you with dinner. I hope that's okay. Also, I wanted to just thank the department and the school for supporting this symposium financially. It wouldn't have been possible without the generosity from the dean and from Martin Grosel, the chair of the department. And also, I want to give a special shout out to Josh Feingold for the provision of the photograph that so many of us are using and then in fact features on your badge, on your thing. It's just a wonderful photo that I think captures the spirit of Peter really well, and it's going to be used a lot, I think. So with that, so we start lunch now, and if we can all be back here by 1:30 p.m. for the first talk, which is going to be an afternoon plenary from Peter Glynn Jr., which promises to be a real treat. And thanks to all the speakers for staying on time so far.
131, if everybody could take their seat, please. Hello? Dear friends and colleagues, if you could take your seats so that we can begin. Good afternoon. This is the afternoon part of our Peter Glynn party here. My name is Bernhard Riegl. Great pleasure to welcome you all here. I have the privilege of introducing some very special speakers. We have two Peter Glynns talking to us this afternoon. Here's the younger one. I dare not say the prettier one because after that it'll be a host of people who have associations with Peter or students with Peter. Without further ado, let me open here the podium to Peter J. Glynn, who is Peter W. Glynn's son. So there's no coincidence in the name.
Thank you. Good afternoon. I'm honored to be here on my father's behalf, and thank you, darling. The title of my talk is From Diapers to Agent-Based Models, and I promise not to bore you with the details of my potty training years. I'll quickly move into more interesting topics. And what I really wanted to focus on initially was to talk about how my father instilled a love of nature and scientific inquiry in me. And it started in the early 70s. He would invite me on some of his research trips to the Uva and Secas Islands and also the Pearl Islands. And I remember I couldn't sleep the night before. I was so excited to go on these trips. And I believe my first one, I was 9 years old. And that's the first time I went scuba diving with my father. And I remember that my impression of going diving was that, because we had a pool and I'd swim in the pool and it was always really quiet. And my impression was, well, when I went scuba diving, it was going to be quiet underwater. So I remember going in and being just amazed by the beauty of the reef. And then I hear this snap, crackle, pop all over the reef, and it sounds like a bowl of Rice Krispies. And then I remember coming out of the water and saying, Dad, what is all this noise I was hearing? And he said, well, son, you got to see the beauty of the reef, and you also got to hear everything living in the reef, the snapping shrimp and grunting fish and so forth. So that was quite an impression for me. I recall one time, I think it was in the Pearl Islands, that we went on, I was on, actually the boat I recall, we saw some slides earlier of all the research vessels in Stry, and the Tethys was kind of an old vessel that I remember being on. And there was a Boston whaler that we had attached to it that we would take on, out to the reef to do diving. And I remember I was just sitting there kind of fiddling around, my hand in the water looking down, and I see this big shadow coming under the Boston whaler, and then seconds later a giant manta ray leaps out in front of the bow of the boat, and comes splashing down within a few feet of us, and it's just a sight that I'll never forget. It was just a remarkable thing to see. One of the neat things about in the early 70s is that there was hardly anyone around those reefs. They were very pristine, and my father told me that they were promulgating this myth around the locals that I think it was the Secas Island that was haunted, and so that kind of kept people away. And the benefit of that was that I remember diving in 5 or 6 feet of water and seeing 100-pound snappers, just huge schools of fish. It was just a remarkable amount of biomass that we encountered there. Lots of sharks. I remember seeing, black tips and white tips, and my father warned me about, one of these days you might see a big bull shark, and I thought he was just kind of joking around. So one time we went on a dive, and I got a little bit distracted and kind of went farther away from where everyone else was, and from the corner of my eye, I see this big shadowy figure approaching, and then I realized, okay, yeah, that's a bull shark. And so I just kind of sat there quietly waiting, and it got real close, and then it kind of swam away. And just seeing this primal creature feeding, and fortunately I was not on the menu that day, but I remember I couldn't wait to tell them about what I had seen. The other thing that was a lot of fun, there's been a lot of talk about fishing, And one of the fun things we used to do is off the tethies, off the stern of the boat, like we'd put down some lights at night, and I remember peering over and seeing all the plankton come up, and then I'd look more and I'd see little fish come up and larger fish right below them, so I got to see the whole chain of life there. And we'd throw some lines out there, and we could fill up a bucket about this big with fish in a matter of an hour or two. And we would eat like kings for the rest of the voyage, so that was really great. And one last story I wanted to share with you is one that's real memorable. Is one time we were out there anchored, and we saw a shrimp trawler that pulled up and anchored, and my father had the idea, why don't we head out there and see if we can get some fresh shrimp? And so the idea came up, well, let's bring some beer and some packs of cigarettes and trade them for shrimp. And so we got in the Boston Whaler and headed out there. And I remember the fishermen had kind of a scowl on their face when they saw us. They don't know, who are these crazy guys coming out here? And then my father held up the six-pack, and those scowls turned into big smiles. And so we approached, and they greedily let us on board. And my dad was negotiating, how many six-packs and packs of cigarettes for how many pounds of shrimp. And meanwhile, I was walking around the deck, and I was amazed to see all these really wondrous sea creatures, because they were fishing probably, for shrimp 1,000 feet or so deep. So I saw these anglers and all these interesting fish and crustaceans, and I started collecting them. And we got back to the tethies and got some kind of bell jar with formaldehyde, and I went back to school. I think I was probably 10 or 11, and the deal the teacher made with me was, I will let you go on these trips with your dad, but when you come back, you need to do a show and tell. So I was very excited. I had this big bell jar with all these interesting critters in it, and I bring it in and showed it to all the kids, and at least for a week, I was like king of the hill. They thought I was a really cool guy, but I think I lasted about a week, and then I was back down the totem pole again after that one. But as far as instilling scientific inquiry in me, I'd say that there's a couple things that come to mind. One of them was, I remember the study that my dad had adjacent to their bedroom, and he would go in there for hours, and this was before computers, so you just hear the clicking away, clack, 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 clack. He's typing away, and then he'd yell out, "Peter, I need you, come on in here." And he would sit me down, and I was proofreading. So he'd hand me his manuscript, and he'd sit there with his red pen, and he'd just sit there like this, and with his red pen, and I'd blah, 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 stop. And then I go, and then he'd grab it from me and make some markings. Okay, keep going. And this would go on back and forth for I don't know how many hours. And then I remember as a teenager, started, how teenagers are. I started getting irritated by it. It's like, oh God, here we go again. But thank you. I think my vocabulary and my command of the English language probably started with having to proofread your papers for you. And then the other thing that was pretty cool, in fact, both of us were talking about how neat it was that we got to see an actual photograph of what his laboratory looked like on the causeway in Panama. And I remember myself running around those rocks and fishing and throwing rocks and horsing around, and he'd yell, "Hey, I need you in here to help me." So I'd run up to the lab, and he'd have a weigh scale, and he said, "Okay, you see all those samples there? I need you to weigh them and record all the measurements," or calipers. I had to measure the length of isopods and God knows what, but he kept me very busy, and I remember that really struck me. I said, this is kind of fun. And then I remember you bought me a little notebook, and I'd walk around the rocks and with my little pen, and I was making my own recordings of things. I'm sure it made no sense, but it seemed at the time it did. And but as I was getting in my high school years, my father kind of dissuaded me from going into marine biology, and I think for good reason. I think at the time it was a very competitive field, and research dollars were hard to come by, and so I kind of pursued mathematics and computer science, and that's what I did for most of my career. And then, so now fast forward to 2015, and we did a family trip, including Bernhard Riegl and our friend Jorge Cortés. We went to Costa Rica, and we did several dives. It was kind of an exploratory trip, and I remember at the time that I thought it was really cool that, there were a lot of scientists talking, collaborating, and they included me in those conversations. And then Bernhard and my father said, you're a math guy and a computer science guy. We want you to start thinking about doing some modeling work. I said, okay, I'm not quite sure what that means, but that sounds interesting. And so he gave me their Galapagos paper from 2015. I believe that was the year. And I read that, and I said, okay, I read it. I liked it. Okay, here's 3 more. I read those. Here's 2 more. And you know, I kept reading and came up with some ideas. And you know, through hard work and collaboration, we actually published a paper in 2017 in the Marine Biology Journal, a modeling study that we had done. And so that was kind of my first foray, I guess, officially in the world, in my father's world. And then now in 2017, we did a trip to Uva and spent about a week there. And I think Bernhard said, you know, I think you're ready for another modeling study. So I said, okay. And we spent time on the reef, and we were doing a bunch of GPS sampling. Bernhard was on the surface. I was underwater, and I was basically antagonizing damselfish to see if they would respond in an aggressive fashion. I think they frequently did. And we did many readings, and then from there came the next study. And just last week, we had our study published in the Ecological Modeling Journal. So it was one of the hardest things I've ever done in my life, is to, other than raising children. There's my daughter over there. But it was a lot of hard work, a few rejections, and a lot of coaxing from my father and Bernhard, but we finally made it happen. So that's really the story I was going to tell you about my father. He pushed me to say, you got to go technical now. So I'm going to dive into the study, if you don't mind, and just to give you an overview of what we did, you know, based on that study. So that's going to be my next foray. But before we do that, I wanted to share with you a couple of photos. So this is at Uva, my dad taking copious notes, and you can see he gave me the dirty job of holding these spiny, sharp diademas and measuring their test diameters while he wrote them down. And I'm not sure whose hand that is. That might be Bernhard's or Juan Maté, you know, with his caliper there. And I really like this photo here. This is just showing you how beautiful, you know, the view is down to Ph.D, the village that we went from, and you know, very pristine, beautiful part of Panama there. Very difficult to get there, the logistics, but once you get there, it's really a fabulous place. And our last picture, this is us on a panga on Uva Reef, and the gentleman in the back is Kevon. He's the runs the dive-based COIB operations. Myself, my dad, and Juan Maté. That was really a fabulous trip. We really enjoyed it. So this is the study that was recently published on agent-based models that were developed, and it was really the idea of the model is to figure out whether the damselfish interactions with sea urchins has what kind of indirect they have on bioerosion. So that's really the gist of this particular study. And you know, some generally accepted wisdom. Some of these are referred to are accretive or positive on carbonate budgets, and some of them are negative. The top one, Hickson, Eakin, and my father, was that damselfish limit sea urchin grazing through their agonism by ejecting sea urchins from their algal lawns, and therefore protecting the underlying substrates. So that's an example of a positive effect on carbonate budgets. And the latter two are more of a negative effect. We have some San Marco in '87, the damselfish lawns providing shelter for microbioerodiers, so therefore enhancing the amount of bioerosion attributed to them. And then finally, San Marco in '83, the fact that the filamentous algae in their lawns outcompetes the crustose coralline algae, and then therefore diminishing the contribution that they have on the overall carbonate budget. So our hypothesis for this study is kind of a null hypothesis, I guess you could say, is that damselfish aggression towards sea urchins invading their territories has no indirect effect on bioerosion. In fact, on the contrary, the expectation at the beginning of the study was that I was going to confirm what is already known, is that they would have a positive effect on the overall carbonate budget. So this is already very familiar. We've seen this slide quite a bit already today, but here's Uva Island, Uva Reef, and Bernhard put together this really cool bathymetry map. These are the 2 sites that we surveyed. Basically, it was me on the surface, Bernhard on the surface, me underwater, antagonizing damselfish to see if they have an aggressive response, and every time that happened, we would take a GPS reading. And we did it on 2 different sites, Site 1 and Site 2. The model was focused on this site, and this is where the 4x5 monitoring plot that you've heard about earlier today is located. So what, 1983 is when it's established, and what, 30, 40 years later, we're still doing work on that monitoring plot. So the methodology for the study was to develop an agent-based model to simulate the interactions between urchins and damselfish, but also urchins with sea urchins to sea urchins. This has to do with the formation of aggregations and sufficient densities of sea urchins. This was something that Pierce and Arch found in '69, that they actually come together and behave as a single social unit. So any kind of stimuli, they will respond to it as a unit. They also graze as a unit, and they also spend considerably more time grazing than if they were not in this particular unit. This model was developed, actually, before I go there, it's important, this model was based on the Anenzo event, so we have to, some of the conditions of the model are high sea urchin abundances combined with low coral cover. There was like 90% mortality that took place in that event. The model was developed in R with a 1-year time frame for model runs with daily time steps. And there was 250 model runs with damselfish present and 250 without them present. And the spatial extent of the model is a 16 by 16 square meter study site with that 4 by 5 meter site that you've heard about inscribed within this study site. So as I said, the initial conditions were 90% coral mortality. And then every simulation run to produce enough variance in the results, damselfish territories were placed within the study site according to how long they are. The larger the damselfish, this is something that Eakin and my dad found, was the more aggressive they are. So the size of the damselfish is important, and also the distance from preferred habitat. The closer they are to preferred habitat, the larger they are, the more aggressive they are. They want to be close. And so this was something, and we gathered the data for this from our survey. I was able to produce some probability distributions to determine how to place damselfish according to this criteria. And then the other data that we used was from the 82, 83, and beyond, years beyond, was the sea urchin densities. This was from the post-ENSA Uva reef studies. You notice that they're pretty high, like in the reef base, which is a slightly deeper portion of the reef, there was about 40 individuals per square meter. And then on the shallower reef slope side, about half that many. And here's the agents in the model, the sea urchin diadema. I said mexicana, I think more accurately would be mexicanum. And then the damselfish, Sagastus acapulcoensis. And this is a spatial model. This is what I was referring to. This is 256 cells, 16 by 16. Here's a monitoring plot. The reason why there's a differentiation between the reef base and the reef slope, deep versus shallow, is that higher densities of urchins here, lower densities of urchins here to start with. And this is a monitoring plot that we've talked about frequently this morning. The one established by Bob Richmond in '83 corresponds to this. So this is a coral patch of high rugosity and considered to be a preferential habitat for damsel. And here's some of the results we found. This is thanks to Bernhard. He put together this density diagram, but I'll try and go through it. It's a little complicated. So if we go from top down vertically, this is 1 day in the model, so initial conditions, halfway through a model run, 180 days, and then 365. These colors, this represents the size of damselfish. The lighter the color, the smaller and the less aggressive. The darker the color, the larger and more aggressive. You notice that there is no change from day 1, day 180, and day 365. We kept that constant, and that was because Meerberg did a study where he found that in a matter of 24 hours, if there's any kind of damsel mortality, that they would be replaced within 24 hours. So I felt that was a reasonable assumption to take. So we move over to this middle column here, and this corresponds to sea urchin density. The lighter the color, the less individuals per square meter, the darker, the more. And you can see this is initial conditions. This is the reef base, so there's more dense urchins, and then less dense down here in the reef slope. This is what really struck me as interesting. We get down on day 180, and you start seeing very high densities of urchins forming. In fact, you look inside here within the 4x5 plot, you've got urchin densities in some of these cells of upwards of 300 individuals. And I was reading some of the literature, And there was observations of large aggregations of sea urchins back subsequent to the 82, 83 ENSO event. And notice that this looks like they're persistent like they stay, but in actuality what's happening is that I did implement in the model that they would dissolve away periodically, but I think what's happening is that they're becoming entrapped by the damselfish, through the agonism of the damselfish. Remember that close to the 4x5 are the larger, more aggressive ones. So that aggressive behavior might lead to entrapment of these urchins. And that's important when we move over here, when you see that I also have sea urchins aggregations. Aggregations are when they're that social unit that I indicated. Under certain conditions, in sufficient density, they will behave as a single social unit. Why is that important? Because they graze collectively as a unit. And not only that, they tend to stay longer. So they may stay, I think Pierce observed, sometimes weeks they would remain. In his studies, he found that they would remain for weeks within the same area. So that's the reasoning behind showing you the sea urchin aggregations. And you can see that these highly dense ones here actually did form aggregations that remained inside the monitoring plot for a period of time. So to share with you some of the correlations that we found, here in panel A, we have damselfish density, individuals per square meter, and we found a positive response between an increased damsel density. By the way, this represents 250 runs, model runs, each of these dots. And we found a linear response, and you can see on the y-axis that as the damselfish density increases, the number of sea urchin ejection events, in other words, urchins that are encountering sufficient agonism from damselfish will actually experience an ejection event where they're being ejected out of the lawn that they've invaded. So we see a positive response here. Go on to the next slide, and then we found a log linear relationship between the log of the sea urchin ejection events, which we have here, and a positive log linear response in the frequency of aggregation. So the more ejections that took place, the more likely you were going to have aggregations forming. And then to close the loop here, you notice here on the x-axis sea urchin aggregation frequency. So as the frequency of the aggregations increases, we found an exponential response in the amount of bioerosion that was attributed to that. So we found that to be a rather interesting result. I don't want to spend any time on these two, but I want to focus on this because I want you to see what the distributions, at least visually, look like. This represents count frequencies of when the 250 test cases where damselfish were present. You notice that the bioerosion scores are kind of, the density is like between 1.5 and 2.5 grams per square meter per day. But when damselfish were absent from this is the 250 runs where they're absent, it's clustering about 1. So it looks different. They look like different distributions, but of course, requiring more rigor, the statistical analysis actually demonstrated that, in fact, excuse me, that we found that there was a significant difference between the bioerosion score distributions for damselfish present and damselfish absent. Yes, sir. 5 minutes, thank you. This was confirmed with two-sided t-tests and randomization testing results. I was going to show you that. So I did a million randomizations, and what that means is 250 bioerosion scores, damselfish absent, 250 damselfish present, and basically shuffling 1 million times and then calculating the mean difference between them. And you notice a really nice Gaussian distribution from that. But we go over here, this delta star, that's the difference between the actual distribution that we found when 250 runs where damselfish were present from absent, and you can see how extreme of a result that is. So that helped confirm our results. I wanted to close with a slide here that is in the Coral Reef book that we talked about earlier. And you can see here on this has to do with this upper portion here is non-ENSO. So this corresponds, we have Panama and Galapagos, and this is the net carbonate production in a non-ENSO or normal conditions. Here we have under ENSO conditions. I'm talking about Panama, but I just think it's so dramatic to see what happened in the Galapagos here. This is these numbers here. This means non-econoid bioerosion, and these numbers a mean of 25 and a mean of 50. And I was talking to my father about this earlier, and I said, "My God, that is so dramatic." And he said, "Well, it's the Eusideris thorsi, the actual sea urchins in the Galapagos are incredible bioeroders. You know, with their Archimedes lantern, is that what it's called? Aristotle, sorry. And so tremendous amount of bioerosion. But let's go back over here to Uva, and you can see that under normal production and maximum production, you do around 10 kilograms per square meter per year. When we get down to about 40, we're about -9, 40 individuals per square meter, we're around -9. Our results indicate that you could have an additional 20% to that, so that could bring us down below 10, maybe -11 kilograms per square meter per year. And you know, discussions with my father, if you can imagine a lot of that bioerosion happening cryptically, where it's undermining the coral reef framework, and with any kind of hydrodynamic pressures, that could lead to really a lot of damage on the reef, on the coral reef's framework. And next steps, of course, I finished that study, and my father is pushing me to think of something else. So you all know what I'm talking about. One thought we have is, what about damselfish population distributions on the ETP reefs? Can they predict the spatial and morphological patterns? So what I mean by that is, the appearance, is it a lot of small patches? Is there some big ones with smaller ones interspersed? Could there be a model that might be predictive in nature? And so we're looking, we spoke to Paula Rodriguez-Toncoso about maybe doing some work in Mexico and Huatulco or Bahia Banderas and Puerto Vallarta. So that's kind of what the future holds. And I just want to acknowledge Dive Base Coiba for handling the logistics for our research trip. Obviously, Stry and Juan Maté for making it happen. And then we really had a good experience working with the editors, Dr. Fath and Dr. Wang in the Ecological Modeling Journal. And after a lot of hard work, we finally were able to get this study published. And that's it. Thank you. If there's any questions.
Thanks, Peter. Have you tried starting with different initial conditions for your damselfish populations and see what sort of results you get with bioerosion? Yes.
As I indicated earlier, we used actual data from UVA as far as how they're, basically I had probability distributions like what was the density of them 1 meter away from preferred habitat, 2 meters away from preferred habitat, and so forth. And then every model run, I did some sampling to figure out where to place them. So every model run had slightly different distributions of damselfish around the monitoring plot. So there was some variance there, but other than that, we just relied on the data that we had available to us from our site surveys.
Okay, thanks.
Thank you.
I guess
we have one more. Yeah.
Very nice. It's great to see this sort of work going on still. And I was intrigued by that first plot that you had that was showing where the damselfish were located. And they were all to the outside of the 4x5 plot.
Yes.
Were there no damselfish territories?
There were some, but they seemed to be quite small.
Interesting.
So my interpretation of that is that they were probably not particularly significant because of their size, their level of aggressiveness. That's a good question.
Yeah, I mean, it looks like an exclusion there. And
it is, purposely so. You're right. I mean, I could have put some small ones in there, but I felt that they probably wasn't going to have any significant impact on the results. So we did concentrate most of our sampling on the perimeter of the periphery of that particular monitoring plot. Yeah.
All right, well, I guess in the interest of time, we should move on. Thank you
Thank
very
you.
much,
Appreciate
Peter.
it.
There you see what happens when you go on holiday with father and son. It's my great pleasure now to introduce myself. my name, if I ever find the cursor. Does this thing have a cursor? Yeah, it's on the other side. If you just close it. We're going to close this. Yes, thank you very much. Okay. Just to introduce myself briefly, there's a lot of Peter Glynn students here, and I'm originally not one. However, I've been hanging around for over 20 years now, going in and out the lab and working with Peter. So I guess I'm the student who never finishes and just hangs there. And this is not about science at all. This is rather very personal. This is an explanation sort of what Peter did also to my outlook to science, and not that I'm particularly important or anything in this, or that this would be of any consequence, but I am just one in a long series of people that Peter has influenced and has taught how to do science and has put their science onto a track. And these couple of pictures that I'm going to show in the next few slides are simply a reflection on the kind of person and the kind of scientist that Peter is, and why he does have, and has had, and has been having such a big influence on so many people. So all of you who did your ecology readings know exactly where I've stolen this title from. Most of you will not know what this is. Peter will know. A young Peter Glynn hung around with a group of ne'er-do-wells, as he likes to call them. And he's not in this picture, but those were his friends. And he was moving through town, shooting a lot of pool, and you can just imagine a young Peter Glynn with a stogie hanging out of the corner of his mouth saying, "Hey man, you play me pool, and if I win, you pay." He was then, and he says for his own better, moved away to school in South Dakota, where he met at least one, if not several, professors who really impressed him. And through that, found his like in other things than pool and beer, and decided to stay with science in the end. So if we think of this, it's, all our lives are sort of, this is of course a Lorentz attractor, right? You all know this. Our life consists of basins of attraction into which we are being pulled. We start somewhere on our trajectory. We circle around one basin of attraction for a while, and then things happen that can move us over into another basin of attraction, and we may flip in and we may flip out. And it seems that for Peter, South Dakota was this bifurcation point, right, where he flipped into science. And this seems to have done something to him that made him want to pass this on. And he passed it on. And one of the people he passed this on is myself. My trajectory when I started out was very different. I came out of university in South Africa, and I went into the Middle East working in the parks, and I was seconded into the national park system in Egypt, and was running the parks on the Sinai. I had a lot of time to stand in lonely places and contemplate what's around. Not much. My major claim to fame is that I have found the only bona fide land shark. We met this thing sitting somewhere in the desert. I have no idea how it got there, but there already, far away from academia, really where just the faintest echoes of academia would reach, I came across this name Glynn. And whichever strange corner of that peninsula I would go to, there he had been before and written something. And I started asking myself, standing on this mountaintop, who the hell is Peter Glynn? So who the hell is Peter Glynn then? This is just going into the Google. If you ask yourself, who is he? Well, this year, 20,000 people will have cited his work. That's not nothing, right? He has a massive age index, one of the highest of this school, in fact, of any school. Very few people ever get the citation indices into those realms where he is. And he gets cited basically every day twice, right? There are 700 citations per year, over 700 citations per year, consistently since 2007. There you can see also what the Google has done to us. His citations, like all of ours, jump around 99, 2000 when the effective search engines come in. But he was cited even before to the tune of almost once a day, right? Without any search engines. So obviously, he's somebody who knows what he's talking about, talks about interesting things. And what are these interesting things? Well, for most of us, it's corals, right? Just to go back here, oops, sorry. He is really the one who tabled coral bleaching and the connection to the ENSO. Now, he's, of course, the first one to admit and say everywhere that it wasn't him who had this idea, but a colleague from South America. And he cites that, says that all the time. I've never seen him wear feathers of somebody else without citing exactly where that feather comes from. However, Peter was the one who really introduced this notion into science and made it completely clear that this is likely what's going on. But it's not only that was going on. It's not like he's a coral snob. He started with balanus and algae. He worked on acanthasta, anything that can have an impact. And there's nothing that's small and boring enough not to pique his interest. Now he's working on something that's actually not boring, but it's small. It's ctenophores that sit on gorgonians, and funny enough, get impacted by an echinoderm, by brittle stars. So with that, sort of the career goes in a full circle again. So this Peter Glynn that I had time to contemplate there on this mountaintop is obviously somebody who knows his marine biology. So the first time I had a chance to come over to the US, which was in the late 90s, I, of course, did my pilgrimage to his lab, and we met. And our closer association goes back to the year 2000, when a young Peter Glynn, Ian McIntyre, Jerry Wellington, Peter and Werner and Rosie Piller there joined us in Bali after the conference on a field trip where we went around. Peter likes to be with people, and Peter is good with people. And why do I have this picture of the students? Because really I should be in there. I wrote a paper for this proceedings, and it had to do with bleaching in the 98 event. And I came up with this really cool idea. I came up with this great idea that bleaching should not be so strong in upwelling areas and in areas with strong currents. Wasn't I smart? Fortunately, before I wrote this paper, I read the various papers that I had read before. And there was Peter Glynn again. Of course, it wasn't my idea. I had just a month previously probably read his papers, and it was in there, and I had just made his own idea my own, and I had been so terribly proud of myself. So I went to him, and I gave him my paper to read, and I said, and listen, by the way, Peter, I just tell you a story. I was so proud of my invention, which actually is yours, but I've cited your paper. And probably if you had come to me at the stage where I was then and Peter was then, I might have said, well, Worm, what do you think? Peter didn't say that. Peter, when I said, listen, I just reinvented your view of the world, but I cited you, he just, with full sincerity, said, well, thank you very much for that. I do appreciate. Which meant a lot, and which would mean a lot, and which shows the kind of person Peter Glynn is, right? He could have very well said, well, you little idiot, where did you think you have it from, right? But he didn't do that. He said, thank you very much. I really appreciate it. One thing he said after this field trip, he said, well, thank you, Bernhard, for taking us here. You are a scholar and a gentleman. At this point, I would like to return this and say, thank you very much, Peter. You are indeed a scholar and a gentleman, and you showed it at that time to somebody who you really didn't have to show it to. So I do appreciate that. And he's showing it, day in, day out to the students he's got. Why are there so many scientists in the Eastern Pacific? Well, because they can't really get away from Peter Glynn, right? He comes and he goes and visits them. This is in Chile. We were chasing some corals that he hadn't found enough in his book with Jerry Wellington. That's Jerry Wellington, Sergio Navarrete, Evie Wheaters in the back, and senior moment, Eric Bornemann to the left. He'll just chase after these things until he finds them. And he will always involve his friends and colleagues, ask them for guidance, and give them their academic due. I haven't to this day seen Peter ever produce anything like his big fat coral reefs of the Eastern Pacific where his collaborators wouldn't get more than their fair share of their due. And that is something that in today's competitive world really needs to be appreciated. There's also no place strange enough where one doesn't end up with Peter if one looks for some sample. This is a wonderful museum in Santiago in Chile, which looks now like it did 200 years ago, I guess. All sorts of pickled things. And he was writing something about Easter Island, and you know, I'm sure you've heard about these statues on Easter Island, and they're supposed to have something to do with collapse and survival and whatever. I have it on good authority, though, that this is all not true. In reality, this is a Polynesian rendition of Peter Glynn's face when he doesn't get the data that he needs, why he went out there. So really, but I'm sure especially students and co-workers will have seen that every now and then. Going out in the field with Peter is fun. One is never alone, and there's always several there. Why are there so many scientists in the Eastern Pacific? Well, because he just basically spawned every single one of us, or every other one of us. He is not only serious with his corals, he likes fish too. He doesn't like fish when he looks at corals. It can be as big as it wants to be. It can be right on top of him. If a big shark would take his leg off while he's looking at corals, he would probably only complain that it took him twice as long to swim from one coral to the next than when he had both legs, but that would probably be the only interest he shows in it. Peter's incredibly serious about fishing. He's a good fisherman, and when he goes out there, he catches. This, by the way, is not wanton destruction of rare Amazonian fish. This is willful destruction of rare Amazonian fish. They have infested this Panamanian ecosystem in the back, and they utterly destroy it. So therefore, it's not a problem if you pull a lot of them out, because they also taste very good and can be eaten. When you go on a holiday with him, as said, Peter Jr. has shown you what happens. There is just papers that come out, whether you want it or not. So for me, what Peter has done is I come from somewhere up in the frigid north, not quite that far north, but in the middle of Europe. And my trajectory first led me quite stably to the African East Coast and the Arabian region, where for many, many years I was perfectly happy, except wondering who that Peter Glynn was, and eventually being drawn into his gravity field. I ended up over in this part of the world and have ever since then had a fairly stable trajectory around this western region. So yes, this was a major shift in my outlook. And the other major shift, I used to work on things like fossils and corals and things that Jeremy Jackson once said about his own work where you're perfectly happy if the only people who are ever interested in it are the other half dozen of people who just happen to work on something similar right now, into something that is much more applied and really has some value to today's issues, ecological issues in that ocean. And that's what drew me increasingly more into the whole bleaching world, where I'm here with Peter nowadays. So Peter, thanks a lot. You made me involved into something quite strongly Eastern Pacific now, and also you've shown me, besides fishing, the other things that are very interesting in life. Why are there so many coral scientists around in the Eastern Pacific? Because you made them, because you maintain them. You teach them the important things, right? You see right there in the middle, people know what is important out there. And we owe you a great deal of gratitude for a lot of intellectual personal leadership, and just generally showing the way. And it's a great pleasure working with you and just in general dealing with you. So thank you very much. And with that, it's my great pleasure to introduce, and I apologize for going over for 2 minutes, Josh Feingold, a true Peter Glynn student, and I can't find the words for that.
Wow. So, Peter, it has been my pleasure and honor to continue to be your student. I think this is a sentiment that we all feel and can express. And like many people have already said, I wouldn't be where I am now if it were not for you in all of the ways that you've contributed to our lives. So I think it's interesting that Andrew started with an explanation of the decade thing, because it's basically the same deal for me. We started working together in the late 80s in the Galapagos Islands, and it's been my distinct pleasure to continue that work to this day. In fact, I think one of the things somebody said to me that had the most effect on my life, it was, again, another one of those situations in your office plastered with all of your reprints and shoeboxes of corals. And you said, Joshua, I have a student working in Costa Rica, Jorge, and I have a student working in Panama, Mark, but I don't have anybody working in the Galapagos Islands. Do you think you might be interested? And as Derek, I think, aptly stated, your mind immediately is screaming, yes, this is the most amazing thing anybody's ever said to me, but you also have to stay cool. And you say, well, let me think about it for a few minutes. And it's led to a wonderful trajectory. So 4 decades of Galapagos coral research with Peter Glynn. Let's see where this takes us. So I had to do this. This is basically a word cloud of all the titles from the talks that are given today. It's pretty clear what stands out, right? And I've known Peter a long time. You'll notice he's wearing a horse collar, BC, in this picture. And obviously showing, again, this is one of the first trips I did with him where we were working on the Mallepora corals in Panama. So before we get to Galapagos, we have to go to Panama for a minute. Here he is taking a picture of that famous Mallepora boschmai colony. And I had the distinct pleasure of, I think, being the person that found it. So we got to write a little note together. And it was kind of a really good education in how Peter is. He wrote this great paper on the first documentation of a marine extinction event. And then with grace and professionalism, we retracted it. Because as you know, when you see something, you know it exists. But how long does it take when you don't see something until it doesn't exist? And I'm still wondering about that. How many species are we losing that we don't talk about because, well, maybe I just haven't been in the right corner of Uva Island yet. And the other thing is, Peter is an avid fisherman. He catches some nice fish. So moving to Galapagos, this is my actual copy of his book, and I carried it with me on many occasions. I called it my Bible, because in so many respects it really was the working tome that ran my life for many years. And there he is with his good friend, Jerry Wellington, when we visited there and showed some of the places he had originally talked about in his book that now the corals were all kind of dead. But we know now that these are temporary situations, and hopefully they'll stay temporary, but we'll see how that trajectory goes in the future. So this is a variety of things that we worked on together. A lot of the things that I've been doing in the Galapagos, mostly related to population biology, but certainly forays into reproductive ecology, El Nino impacts, a bunch of cruises we did together. And of course, during all of this, I actually I think this actually reminds me of some funny conversation I had earlier with Jorge Cortés. He reminded me that when he finished his work, you were congratulating him as your first student. And I said, no, Peter, I was your first student. And you looked at us and said, master's students don't count. So that motivated me. I guess I was already your PhD student at that point, but it was a strong motivation to keep driving forward. And of course, I was honored that on the day of my graduation from the PhD, we were together having a beer in the field, there's no better place to be than having a beer with Peter in the field. What do you think? You meet a lot of scruffy characters. I think I'll hopefully change a little bit more as I get a little older and I can be a little scruffier looking to fit in better. But most of my work was at this place called Devil's Crown in the Galapagos. I was mentioning some of the people here that I started my work on coral communities in Academy Bay, and that didn't work out because I could only find 7. So you can't really do a dissertation on 7 corals. So we wound up on one of the trips going here and finding thousands of these fungiid corals that sort of formed the basis of my work. And we're still going back. There's still lots of fungiids. And we might be seeing an extirpation of that large fungiid in the middle, but that remains to be seen. I also worked on a lot of his reproductive stuff. Sue Colley is here. I think we published together with this on Cycloceras distorta egg production. I never thought I would get into histology, and I appreciate the opportunities to do that for a little bit of time. Opened my eyes into some of the important ways in which populations of corals can get maintained. But another topic we've been working on is Peter laid the groundwork for some of the coral populations in Galapagos. This was a picture taken by Charles Birkeland, and I think that was back before you could afford graduate students, so you had local help. But this was the post-Galapagos reef that you're all very familiar with from the Panama area, but this is what it looked like in Galapagos, and it disappeared in the El Nino. There were none found in the caldera until 1995, and then I wound up continuing a lot of these surveys for him. And most recently in 2019, we were back, and one of my students quantified populations there. They're starting to come back. It looks like a different morphology. We're learning more about the taxonomy of Postaloperos, so hopefully we'll get this nailed down a bit better. But this was basically what's happened. There were none until 95. The slow increase till 2007. There was another mortality event we think associated with La Nina, and now it's coming back. So I keep telling my students, I want to see a reef back there within the next couple decades. What do you guys think? What do you think, Peter? Is it going to happen? Don't want to say. Ever the scientist. So the legacy continues. I am now privileged to be able to pass on the torch to another generation of students. It was kind of funny. I'm sorry, I'm a little ahead of myself. So these are some of the products that came out of some of this work. There will be more, but this is the legacy that's passed on. There's Peter Glynn with Elizabeth Glynn, who's not relative, but it was kind of a fun circumstance. But this is the whole thing about Peter. He passes on his knowledge and then gets you all excited about continuing that trend. So one of my goals in life was to become a professor, and I remember another time when Peter Glynn fundamentally affected the trajectory of my life. He mentioned an opportunity that had arisen at Nova Southeastern University. Dick Dodge was a good friend of his, and Dick mentioned that they had a professor leave on short notice and needed somebody to teach an ecology class. He said, Josh, you said you always wanted to teach, and so why don't you check out this opportunity? So I did. And now I get to work with Bernhard, I get to work with a lot of, and still come back down to Miami. It's not too far. And again, thanks so much for that one word, that one suggestion that has profoundly changed all of us. And I like that little diagram Bernhard showed about the circular aspects of life. It's very true. So I will finish with the admonition that he told me early in my career that is still true. He would leave you in the field to take the torch and make sure you got the samples and bring it home and work it up. And I'll always appreciate all of these opportunities. And thank you for letting me express some of my appreciation for all of these things Peter Glynn has done. So thank you.
Any questions, please? Fire away. No?
Moving on.
If there's no questions, we move on. Our next speaker is going to be Nohora Galvis, who comes all the way from Colombia, from the ICRI Foundation. Nohora, it's all yours.
Thank you. Good afternoon. It's my pleasure to be here, and I have to thank the university who invited the university organizers who invited me to come here to share the experiences of being a student of Dr. Glynn. I have a different approach. I want the one who should have chosen Dr. Glynn. I'm going to talk about chronological order on my career, but I put Dr. Glynn in my career because I know the excellence of professor and researcher, so I want him to be my advisor. So to talk about monitoring geological aspects, already in the morning, many people talk about the geological changes on coral reefs. So I wanted to share with you an anecdote that I have in my first examination, my first exam with Dr. Glynn. There were 5 questions, and I just concentrated on the first question. It was about the evolution of coral reefs. I want to show him that I was eager to show him that I was a very good student. So I concentrated on the first question, evolution of coral reef. Many, many pages replying that question. He said, time is going to be up in 5 minutes, so I have to write 2 sentences in the other questions. But then he is a very good professor, he remembered that. And during the graduation ceremony, he gave me a book about the evolution of coral reefs. That was very nice gift that he remembered. Okay, the way on my career, how I developed my career since I was 2 years old, I started swimming. And then as a teenager, I started diving in the Eastern Pacific. But in Colombia, we have most coral reefs in the Caribbean Sea. So I went also to Cartagena. Most of my research is in Cartagena. When I got my diploma as a bachelor's degree, I also got a position at the government to work on coral reefs. In 2 years that I worked in Cartagena, I got a scholarship from USAID. I can pick any university in the United States, and I could pick any advisor or professor in the United States. But since I already have written a paper on the evaluation of this coral reef where I did my thesis, I cited Dr. Glynn, so I am in that record, those statistics I cited many times in that paper. And that was an ecological evaluation of a coral reef. And there was also another paper on the socioeconomic analysis in the Cartagena Bay. So when I came here, I said, Dr. Glynn, I want you to be my advisor. And he said, well, I have many students. You are interested in the Caribbean Sea. So he was very picky, and I have to convince him that I was really interested in coral reefs. I wanted to make a change in the decisions were made in the Colombia and the government. I was working in the government. My work was to come to learn from him how to improve the ecological how to put the decision-making taking into account the basis of ecological sciences. So he say, well, you are insisting, okay, I will accept you, but remember that you cannot get a PhD with just monitoring. I say, no, I only came here to do my master's degree because the scholarship year was for 2 years or so. So I completed that goal, that was to get the master's degree, and he gave me also a gift when I went back to Colombia, was a thermometer, calibrated thermometer. So I can give that to people working in the Pacific to measure in the Eastern Pacific the changes in temperature. So here's the map of Colombia. And well, I wrote here, as I say, chronologically, the 35 years of my career is first 10 years working in the government, for the government, and then 10 years working for the academia. I was a professor. I learned to be a professor also from Dr. Glynn because he paid attention to his students. And right now, I'm very satisfied being also a professor because as I see right now here, all the students remember all your thoughts and your advices. And now I also got students in the city that say, Professor Nohora, thank you for changing my life. So I think I learned to be a good professor because of you. And my topic is about monitoring, what knowledge we can get to the decision makers so we can improve management of coral reefs. Right now we are getting this number of reports. We have citizen science, a citizen science program, because we cannot be all the time in the coral reef researchers. And the scuba divers and the fishers are every day performing activities in the coral reefs, and they're taking pictures in a systematic way because they go to the same dive sites and the same fishing sites. We have a network of volunteers that report to us, and there's an observatory of coral reefs in the Foundation Eakin Colombia since 2008. So our 2 case studies are here in the Caribbean Sea. This is Varadero, already talked this morning, Monica, about it. And this is Burgana in the border close to Panama. In the middle is the National Park Corales del Rosario, where I did my first research that I mentioned to you in my thesis. We have to do 5 years in Colombia to get a bachelor's degree in my biology, and then we have to do 2 years of research. So when I came here, also Dr. Glynn said, but you already have a master's degree, because we have already done the research. And this, the pink case studies are hotspots. Have you heard about hotspots? The hotspots are a bottom-up protection scheme, and their coincidence with a resilient coral reef. The health of these 2 pink case studies is very outstanding. The coral health cover is between 80 and 70%. And talking to Alina now, yes, we confirm that we have seen currents influence also. There's the first meters from the surface are turbid water, and then the bottom is clear water. There's currents, open ocean coral, currents that may explain why they remain resilient. Okay. The first publication that I made here in the Bulletin of Marine Science was about monitoring. When I got my master's degree, I just focused on ecological indicators. And I'm sorry, here I was talking about restoration programs. But at that moment, at that year, we were just thinking about natural restoration. When you remove local threats, we can get natural restoration. And we are going to show you, I'm going to show you later a case study where I prove that when we remove the human activity that threatens a coral reef, we can get natural restoration. The socioeconomic indicators, I was interested because we need to manage people in coral reefs. So we need to identify what is the intensity of the activities, the frequency, the duration. And we have to relate pressure, state impact, response, and management kind of indicators. In total, we got 151 indicators. And that was presented in the Fort Lauderdale meeting in 1999, was that? In Fort Lauderdale. And socioeconomic, other socioeconomic indicators related to fisheries. So ideally, everything has to be measured, and the different institutions have to take care of different aspects related to their functions, to the responsibilities. And the coastal development, we can call in some areas unsustainable development that right now is going on, building larger ports and dredging, and trying to dredge some coral reefs. So that's why we need that information to empower communities, because we're working with the community, so they can also say we have the right to protect our, we call them treasures. You have treasures of coral reefs, so you have to protect your own treasures. So now, right now, it's a law in Colombia that the community has to be consulted for any of these projects, mega projects. After I finished the studies here, I went back to Colombia, and I got another scholarship from the Colombian Research Institution to go to other university in the United States. I went to Harvard. I got studies in environmental economics and policy analysis. So I learned how to measure how to measure the value of coral reefs. Although if we now look at the theory of value, what is very valuable doesn't have a price. But we need to show monetary values to decision makers. And in the Varadero case study, we measure that the project, they were thinking that there was just 60 million because of the environmental cost. We showed them that is 244 million, no more. They were not calculating that, so that could help also in the decision making to stop it. Besides, we write letters, of course, and there were other NGOs also working on the stopping of that project in Varadero. Right now, with this government, perhaps it's going to take again that project to dredge Varadero. Hopefully that remains stopped. Since in Varadero, we worked in 1989 with the fishers. They were the ones who were stopping any projects in 1990. They're supposed to be dredged at Varadero Reef. But now we are in 2020, and there are still some plans under this desk. I mean, people are still interested in dredging that reef. That's why we want to be independent NGO, because we advise the government saying, please don't do that, because there's a 500-year-old coral reef, and it's resilient. So, and it's valuable. After these studies, I also learned a master's degree in Holland, and I got a European scholarship, and I went to learn social sciences. And I learned that the relevance of involving people, but taking into account the multidisciplinary knowledge, working with more people, more professionals from different disciplines, and very important to do transdisciplinary research. That means integral research, because we're talking about how the decision should be adopted, taking into account the information. So we involve the stakeholders. This is not working very well. Well, the stakeholders are involved in all the process of the design of the research, in the co-production and the dissemination of the results. And here is Varadero This is not working. Well, the currents, I can show them here. The currents, the bottom currents are keeping the Varadero Reef in a very well state because in the surface is very turbid. But the open ocean keeps, thank you. Okay, thanks. This is Varadero, and this is the protected area. We did this as a multi-criteria analysis. And here, the red letters mean dive sites that have low performance in the indicators. And this area that is closer to Varadero Reef has been performance, very good performance of the indicators, socioeconomic, ecological indicators, and coincidentally is close to Varadero Reef, which is in a better resilient state. So we can show here to the ministry that it's important to protect Varadero because this information was from 2000 to 2008. The study lasted that amount of years, and this means that this reef should be included in the protected area. And that's what I was saying to you. These are the bottom currents that come with open ocean water. And the channel, thick channel, is in the surface. Come here and protect with the shadow these coral reefs, these beautiful big coral colonies that are 5 meters in diameter. So that's one of the explanations with the observation that we have. We have observations every day from divers, and the fishes go also there, and they are the ones who are protecting the reef also. So our research is applied research. And this is the other case studies in Burgana. We have developed, we have identified the activities, fisheries and diving sites. We monitor them because every day they go to the same sites. They go down by the mooring buoys, and so it's a georeference transect, and they videotape the transect. So we can see right now the resolutions of the government have changed because it can be this kind of research, and also the fishers have noticed that the improvement of this indicator, the native species of fishes, important for fisheries, they have been recovered. And this is what I say, the natural restoration of the coral reef has been shown good result because the industrial fisheries were banned in the area, so they coincide also with the control of the lionfish. There's biological lionfish control because they are bigger groupers now. There are also sharks in the area. There are many IUCN red-listed species in these areas that we call hotspots. And this is where we intervene with the community, so they are eating the lionfish. And this is very low population now in the area. There's another chart here, and here is the recovery of the fisheries, and there's a resolution from the government to increase the quota for fishing. I published that research in the International Coral Reef Symposium in Hawaii, where I met the two Peters. And these are my lessons learned from Dr. Glynn. I learned a lot from him, many different aspects that were our work value. It's very valuable, our work, that we have to be very careful what we publish, because I have to, for example, write in Spanish and English simultaneously sometimes. And monitoring, in situ monitoring is very relevant. For us, we need to influence decision-making, and we have to listen to our students and be example to them. I learned that from you, and thank you very much, Dr. Lee.
You know, Nohora, we keep the questions for later because otherwise we would be cheating. Don Olson. Where's Don? Oh, sorry. Who's coming here now of his time? Don, the moment I find the cursor, you're up. Don is going to tell us about wandering the world with developing fauna. Wanderings with developing fauna. There you go.
I originally misspelled it and said wandering instead of wandering, but I actually first met Peter when he first came here. He came to, I was in charge of producing what people wanted with our programmers in the remote sensing group. And he came and said, there's an El Nino in the Pacific. And I said, yes, I just got back from Colombia. So we sat down. We weren't getting real-time data then. We actually had to use a lesser product than we later produced. The title basically is I really started working with Peter from kind of the slow, non-productive side of academia. I was associate director of the marine science program, working out of the dean's office. And then Linda Farmer asked me if we'd put together something called saltwater semester and suggested that we train Peter to do invertebrate zoology. And I did physical oceanography. And I think our first trip, which we started off in January, and it's a horrible time to go to Key West and Fort Tortuga, Peter asked me, how can you stand going to sea? We had a nice cold front right up around Key West, and it's the night of the testosterone challenge. The young women went to bed. The young men tried to help the captain and threw up all over the ship. So these are basically pictures of my memories of Peter. We went to Bimini. This is the coral reef, little boat. We went to Bimini, I think, twice, Punta Cana and Fort Tortuga once. We have the students kind of captive. The first semester we did this, I thought, I don't really know much about they're an equation to me. The copepod is C1, C2. And I put a dot over the top of them and integrate them. So I thought I'd set in on invert zoo. This is the book. You can't carry it to Hawaii on a trip and try to study it all the way back on a red eye to take a test. I didn't really plan on taking Peter's test, but the TA came in, Peter was out of town, popped a test in front of me. I think I got a B maybe right out of the course. There's just a lot of stuff going on there. I think that students used to say that Peter can't write equations on the blackboard and get them quite right. And I can definitely not pronounce anything in Latin successfully. But together we make a pretty good team. There was some work on a program that I was involved with. I was the national chair. Peter Needler from Scripps was the international chair. Looking at the Pacific, one goal was to improve sea surface temperature by an order of magnitude. The other was to understand the currents. I worked some with Dick Backus at Woods Hole, who got me interested in biogeography, and I do diffusion in the ocean. And therefore, biogeography has to be all about currents and diffusion. And so I was interested in trying to think about Darwin's little gap, the East Pacific barrier. Again, I work in a remote sensing group, so when I think about the islands, Peter came and asked me about currents around Easter Island after he and Jerry Wellington got back. And he said, they don't seem to have anything to do with French Polynesia. And I said, well, we can look at that with our drifter sets, and I'll show you a drifter a little bit later. So we wrote a paper together on Easter Island. And then after that, and I'll show you the diagram out of that paper, Peter thought that, wow, Easter Island seems to have more in common with Galapagos than it does stuff farther west. So we started thinking about that. So we're going to do the currents and what happened to the current arrows. This is basically the figure showing that in lower. This is what's called the World Ocean Circulation Drifter Program. These are little satellite units you throw out of Ship of Opportunity. They drift for a year or two. Peter and I sold a program to NSF. These cost about $2,000 a piece, and I think we've thrown out over 100,000 over the years. And again, our first success was showing that we could correct temperature and improve 1 week forecasts on a international level. But we can also use them. They measure temperature. Where can corals grow? So when I think about the gap in the Eastern Pacific and how connected are different islands biogeographically. So when I think about, whoa, that didn't come out very good. That's the title, kind of blurry. So the first thing I did, of course, in my sensing is go look at our data and go back through the archives and find the warmest and coldest period ever. So we have a very cold December period and a very warm December period, looking at max and mins. And then we're going to look at these on drifters. These drifters, by the way, were used to basically improve the sea surface temperature accuracy to about 0.2 degrees, from more like 1 degree. This was an overkill slide for the Navy, showing that we have enough data to say something about it. You notice Bernhard is one of the authors. We kept adding authors, and I've gone through, I think, this is the second longest paper I've ever worked on. There's an older one. We started writing this in 2001, I think. So we kept adding authors. We also kept adding corals. So if we keep not publishing this, we maybe can really populate the corals. I think early on, I thought there were 7 corals in the Eastern Pacific, and Peter corrected me, no, there's 9. And I think the third version of the paper, he said, no, we need to get the corals right. They're not 9, they're 12. So the corals have actually been populating the Pacific, Eastern Pacific, across the barrier ever since. What do you do with these drifters to try to understand what's going on? And this is Rick Lumpkin from across the street, who's the current director of the drifter program at NOAA. This is what's called a splash diagram. So we're going to take every drifting unit that went through the little bullseye. So it went through that white circle out of our entire data set. And we're going to plot in days the time later it's somewhere. So it's 0 days when it's somewhere in the circle, that's Easter Island, and we're going to go out in time. And the idea is I can just see the little corals going across the seascape. The one thing you notice here is that Easter Island's up on a big hot spot instead of islands. This has good retention, but very, very poor connectivity with any place else. I'm not going to show you the French Polynesia, but all of the drifters from there go well south of Easter Island. So there shouldn't be any connection between Easter Island. So that's Easter Island. This is the Galapagos. So this is the barrier. And my first look at this, I have drifters that have gone from the Galapagos to Easter Island. Unfortunately, it takes, I think the quickest one's about 85 days. And the question is, can the little coral live that long? I'm very kind of leery of how you calculate competency time in the plankton. Cocos Island, I pronounced them Caicos after the one in the Indian Ocean, but is that right, or I had it the other way around? I don't know. Basically, we have drifters go in 4 days, back and forth between these islands. The residence time in the Galapagos is fairly good. Actually, I think it averages at about 40 days. The way these are done, it's the least time, so you're painting over things, so you don't see those retentive ones as well. And we can also get to Caicos in 80 days to the Line Islands. The Line Islands never get you back to the Eastern Pacific. In other words, there's none of the drifters, and I think Peter was always interested, there's an equatorial countercurrent in there, which should have been on a previous slide that plopped up, and the arrows are missing, but the countercurrent goes east towards Panama, but it's so narrow and has so many eddies that none of our drifters ever make it. They get washed into the equatorial currents and back into the Western Pacific far faster than that. So we wrote this, the coral population, the number of genera in the Pacific is going up as we slowly work on this paper. So I estimated that if we wait and hold off on this paper to about 2111 or so, we will basically be approaching the number of corals in Tahiti, roughly. That's kind of a Mark Twain type calculation. That's something, you know, mathematically it works out fine. It's, of course, ridiculous, and we stopped writing because it seemed too long for larval competency. There was connection. Why don't we just connect them across the Eastern Pacific from the south? And a paper came out. We put the paper aside, I think 4 years or 5 years, and a paper came out suggesting that in climate optimum about 8,000 years ago or so, there could have been corals on the coast of South America. And and then we had a couple anthropologists who are famous or infamous for arguing people got to Chile 14,000 years ago, and they were working in Peru, and some fishermen gave them this coral. My daughter sent me a coral. I gave it to Peter, and he said that's a cold water coral. But this one is, and I can't pronounce it, so I won't. The P word. The problem is there's more than one P word, right? Unfortunately. So this is looking at the splash diagrams from where this was found on the Peru coast, and it's days. By the way, the Panama link is weak. There's a lot of stuff. The Panama, it's about a month for the average time to get across here. So we submitted this to Science and Nature, because it's a way to make yourself famous, but they had too many coral papers that year, both of them. So we're rewriting it again. And I think since then, Peter has outproduced himself. I think there was about 20 papers in the next year that came out on genetics and other connections. So I think it's probably time maybe for me to get busy and write this, or should we really stop this impoverished, get rid of Darwin's famous barrier by just putting this paper off for a year or two more? I'll let you know what you think. We can meet in coffee between the co-authors. If somebody else is interested, we're also, we want to keep up the growth of co-authorship, if at all possible. We need about 3 or 4 more to keep up with the rate of the increase in corals in that part of the Pacific. Well, you can bring something. You can bring another climate model. You can bring a table of competency times. We could do a agent-based model. I call them IBMs, individual-based models, but I actually have a random walk that goes with the drifters, actually. With Claire Paris here, we've done millions of runs in the Caribbean. So we'll keep you informed. Don't stay up late at night waiting for this paper to appear somewhere. But eventually, and that's it.
Great. Thank you very much, Don.
What happened?
Any questions to Don? Otherwise, the coffee outside is waiting for you.
That was too baffling. Oh, you want to become a co-author? Oh.
No, I mean, rafting has been attributed to corals, if they can...
Yeah, that's part of the other, Yokel and others argue that rafting is important, and we've been trying to, that's what Peter and I do. We built all these fiberglass balls and launched them all over the ocean for rafting. My problem is, how does the coral jump off the ball? Well, fragments and jumps off, and what is the timing?
What's been invoked in Hawaii is multiple rafting events,
Yeah.
so that you end up having enough individuals to have a sexual encounter.
Yeah.
Because, all of the larval work I've done, it's really hard to keep a coral planula going for more than 30 days.
Well, my problem there is if you're doing it in a lab, how do you keep something? I worry about, and it could be either. It could be rafting, definitely. The first time I launched some drifters and did this, the first person who quoted me and wanted to talk about it was somebody from the Missouri Botanical Garden, and he wanted to talk about coconuts. How far can a coconut drift before? So,
pumice pieces.
Yeah.
Japanese fishing balls.
Yeah, no. That's clearly a possibility, and I think we have a paragraph that we've changed 2 or 3 times too, right? I'm not a fan of rafting, although our little drifters could provide rafts easily. Maybe maybe that's the reason the species there are increasing. But thanks for bringing that up. That is another hypothesis. Okay, coffee.
If everyone wants to start moving to their seats, Derek Manzello, that'd be great. Thank you. All right, we have made it to the fourth session today. We've heard a lot of talks from the Eastern Pacific, and we are about to jump into a different pool and start to look at some reefs that are more closer to our backyard. So with that, I'd like to introduce Jim Porter. Take it away.
Oh, thank you. Let me start by saying what a pleasure and a privilege it is to be here with Peter Glynn for this amazing honorarium symposium. My friend, my mentor, my Ph.D. advisor when I was a Smithsonian Tropical Institute Fellow in Panama. We are going to shift oceans here as I talk about coral species number and percent cover of coral reefs in the Caribbean. We are all familiar about the decline in terms of the number of species and percent coral cover throughout the Caribbean, in the Indo-Pacific, and across most of the globe. I'm going to talk to you today about long-term ecological dynamics that we have been looking at in our coral reef surveys funded by EPA and FWC, my friends from South Florida. Every element of this design is important, so let me tick them off before I begin to get into the data. We're looking at a region-wide analysis here from Key Largo to the Dry Tortugas. We're looking not only at coral reefs, but also water quality monitoring at 172 individual stations within the Florida Keys, matching all of our coral reef stations with water quality information as well. We look at percent coral cover from, that allows us to calculate delta coral cover versus time. But our survey was particularly unusual because we also took species number. And this was done not from the video data where species are not well represented in the photographs, but from a pair of individual divers who spent 15 minutes doing a timed species count in each and every one of these stations. And we have the nutrient data from the EPA Water Quality Monitoring Program. So what I'm going to do is to compare and contrast the patterns that we see with all of these data and how we can interpret coral species loss and coral percent cover loss, and to look at the best fit explanatory models that leads to a hypothesis for the differential patterns that I will be showing you between coral species number and percent coral cover. Now, there were 50 species of corals contained inside the 172 stations that we monitored annually. And what I can tell you is that 75% of these species were found in fewer stations at the end of the survey in 2014 than at the beginning in 1996. And 50% of these were found in significantly fewer stations. With a 95% confidence, I can tell you that coral species number in the Florida Keys is declining. Now, one species, Isophilostria rigida, I believe is now functionally extinct in the Florida Keys. You may know where there's a colony, but it's not in any of the 172 stations, nor in any of the traverses that we swim to get to those stations. This is an extinction event that we have measured in real time. There were 35% fewer stations that had Mycetophilia ferox, 29% fewer stations with Mycetophilia lamarckiana, 32% fewer stations with Acroporus cervicornis, and 21% fewer stations with Favia fragum. Now, it is important to say that there were 4% of the species that were found in more places. 2 of them were in significantly more places. One was Siderastria siderea, and the other was Stephanocenia michelini. Now, if you look at the number of species versus time in the Florida Keys, you see 2 trends. The first is a significant decline in the number of species found there, and also a monotonic decrease. For the Florida Keys, there is an average loss of 0.21 species per year, and this is time in, time out, and it doesn't matter where those data are taken from, whether it's deep reefs, shallow reefs, or patch reefs, you find the same result. For the deep reefs, it averages 0.51 species per year. For shallow reefs, it's 0.15 species per year. And for patch reefs, losing at a rate of 0.16 species per year. It also doesn't matter whether you are inside or outside of the special protected areas. The species loss rates for both SPA and non-SPAs were exactly the same at minus 0.33 species per year, again from the beginning of the survey till the end, and in a monotonic decline. So if you look at species number versus time in the Florida Keys, the average loss is 0.21 species per year, and what that means is that every 5 years, the Florida Keys, it loses an entire coral species. This is a frightening bit of information that goes hand in hand with the percent cover information that all of you are so familiar with. Now, what you would expect if there were a decline in the number of species is that you would also have a decline in the species diversity H prime, because H prime is so sensitive to the number of species that is included in the calculation. On the other hand, what you actually find with the Shannon index is that there is not much of a decline, no significant change over time. So over the same time period that there has been a loss of species number, there has been a rather no significant difference in terms of the species diversity. Now, there's only one way that that is possible to occur, and that is if the evenness of distribution of those species, the relative abundance of each species, has actually increased. And because we have abundance data from the video, we are capable of measuring evenness, and in fact, that is exactly what we found, that evenness has increased significantly over time. So what this means is that although Florida Keys are losing species each year, the Florida Keys are also becoming more homogeneous, an increase in the relative abundance of these different species over time. Now, let's also look at the species number versus time and try to understand the kinds of models that would accurately identify this trend. Only a one-line model, and specifically a linear fit one-line model, fits these data. This is highly significant, it's true for all sites, and it is a uniform monotonic reduction in species number. Okay, let's compare and contrast that loss of species number with the other kinds of data that are coming out of this analysis, and that is percent cover versus time. As with the species number, percent coral cover is also going down, and it is also highly significant reduction. Since the start of the survey in 1996, 50% of all corals in the Florida Keys have died. This is quantitative data. We have seen this, and we have measured it. It is, of course, not exactly a linear decline, and specifically the extraordinary El Nino of 1997, 1998, within our time frame of data, was the point at which there was a huge break in the coral loss data. And this pattern and the timing of the pattern loss is the same for both, whether you're looking at offshore deep reefs, patch reefs, or offshore shallow. Again, a significant decline and a break at the extraordinary El Nino year. So in this case, only a two-line model fits the data. Again, linear in both cases, but a break is required in order to fit the data. There's no other way to fit these data but contrasting patterns of linear models. The break always occurs in the extraordinary El Nino year, the bleaching year. Now, interestingly enough, hurricanes do not significantly influence the rate of coral loss. Everyone in this room knows that's not true for individual reefs, but over keys-wide analysis, it is true, and we see that as a result of that, we don't need to look at this geographic scale of the individual impact of hurricanes on individual reefs. Now, we also find relatively the same pattern, again, in special protected versus non-SPA reefs, but the loss of percent coral cover is actually greater in the protected areas, which is a little hard to deal with. As with S, SPA status does not protect against percent cover coral. They started out relatively the same. The SPA reefs at this point in time have less. SPA loss, coral cover is faster than non-SPA reefs. It wasn't true for species number, but it is true for percent cover. For S, only a one-line model fits the data. There are no breaks at any time, at any place. Therefore, controls over S and controls over percent coral cover must therefore be different. So for coral reef percent coral cover, temperature explains 95% of this variance. Let's compare and contrast that for S, where temperature explains only 12% of the variance. Once again, different controls. So, for a functional hypothesis to explain species loss versus time, it must apply to all locations in the Florida Keys, Key Largo to Key West, nearshore to offshore, apply uniformly to all reef types, patch reefs and berry reefs, apply to all years 1996 to present, do so consistently from year to year, and apply uniformly to all species with the possible exception of Siderastrea siderea and Stephanocenia mitchellini. For S, we know that temperature explains only 12% of the variance, so we looked into the data to find out what was going on, and when we add nutrient data, specifically soluble reactive phosphorus, nitrous oxide, dissolved in organic nitrogen, and ammonium, we find that those combined explain 60% of the variance. The nutrients correlate best with species number loss. We know that where the nutrients have been measured over a similar time frame, they are increasing, specifically our data from Lou Key. And we know that the Lou Key percent coral cover follows the trend that we have suggested for percent coral cover. And we also know, however, that correlation is not causation, so the mechanisms by which nutrient increase could influence species number is unclear, but with a 60% explanation of the variance, you've got to look under that rock. You just have to do it. So I would present to you the following hypothesis: that coral cover in the Florida Keys and probably worldwide is controlled by temperature, but that coral species number may be controlled instead by nutrients. And we do have the data that should allow us to explore this more carefully. We have the 97, 98 El Nino, and what we have are the data continuing to 2015, 2016. I'd like to know from those data whether S continues its inexorable loss and whether there was a break depending upon coral cover. Now, in closing, I want to give you one more observation from Peter. This picture was taken 50 years ago, almost exactly 50 years ago, in March in the Hollandaise Keys off San Blas in Panama. Peter was doing something then that has influenced me for the rest of my life, that had very little to what anyone else at that time was doing. Peter was talking to the press. Peter was teaching the next generation, me and his son, who was 9 then, speak up, speak out, be brave. Students now are taught that they can do that. Back then they were taught not to do that. Don't do that. Don't speak to the press. Don't share your ideas. Don't do those things. Peter said, do them. And I have lived my life with that lesson, Peter. Thank you so much.
We have time for one quick question, very quick.
Yes, over here.
The Florida Keys have made huge efforts to try to reduce septic tanks and septage, which is common. The Florida Keys have made a huge effort to try to reduce septage from going onto the reef track and reducing nutrients, and that happened late 90s into the early 2000s. Do you think that has made any difference at all?
I think it has made a great deal of difference, but maybe not directly for the nutrient increases which have continued. It's reduced the amount of nutrients that are coming from the Florida Keys themselves. But unfortunately, being a connected water system, elevated nutrients are continuing, and I think there are other additional sources for it. But one of the big things that those water quality enhancements have done is the reduction of bacteria and other pathogens that we're getting out to the Keys. We measured one, Serratia marcescens, and so that was a smart thing to do, even though it hasn't solved the global problem. But thank you for that question. All right.
At this time, it's my privilege to introduce Dr. Mary Alice Koffroth.
You'll turn me on when it comes time. Okay, so first of all, I'd like to thank the organizers for putting this event together. It's been great to see everybody, to hear all these great talks and everything. And of course, I'd like to say congratulations, Peter. Welcome to the club. So I can make this thing work. This is where I first met Peter, 1983. I traveled to Panama for, as many of others of us, to do a short-term fellowship. And Peter was also my advisor here. And so I actually spent time at NALS for a few days meeting Peter, and then I headed off down to San Blas, where Peter was over on the Eastern Pacific, noticing all this coral bleaching. And it turned out to be a very, as we all know, a very unusual and very disturbing summer. I arrive in San Blas, and the first thing we see, dying diademas. So this was basically we came right at the start of the diadema. This was one of the focal points where the pandemic started that spread throughout the Caribbean. And then I started working on my thesis, and we started noticing bleaching in the San Blas. And it was pretty disturbing. I'm seeing diadema die. The corals I want to work with are bleaching. I'm going, is this a sign? Should I not be doing a PhD? What's happening? But anyways, I continued my PhD work together with several other people. We began surveying the bleached corals in San Blas. We began monitoring coral recovery. Resulting in my first introduction to bleaching, we looked at the bleaching response to corals in the San Blas. That, I think, led Peter to invite myself and several others to contribute a chapter on his book on the global ecological consequences of the El Nino. So together with Howard Lasker and Jamie Oliver, we did this large survey of coral mortality and coral bleaching outside the Eastern Pacific region during the 82-83 ENSO. And so this just is a map that shows the different places where coral bleaching and coral mortality had been reported during 82-83. We looked at the literature, surveyed, generated information on where they were, what the potential causes were. And at the time, it may have been a little surprising. Of course, now it's not. The vast majority of bleaching and mortality events were correlated, temperature was given as a probable cause of this. I know at the time I was monitoring temperatures in the San Blas, and at the same time at bleaching, we saw this spike in temperature that continued. So anyway, basically we continued. I helped write this chapter, continued doing my research, and just these people at the top were not my committee. They were all my advisors while I was here at RSMAS. My first advisor was Dennis Taylor. After about a year, he left. I started working with Mike Roman. Then he left. I thought, this is telling me something. And so at this point, I was working on my Ph.D., and there were no coral reef biologists at RSMAS, but I knew they were interviewing people. So Jack Fell agreed to be my interim advisor. And then Peter and Alina showed up, and they were my co-advisors initially, and then eventually Alina became my advisor. So I probably was Peter's first RSMAS student. But I won't fight Mark and Jorge. Anyway, so I did my work, my thesis work, and at the same time, Peter took me to some wonderful places. This is the Barrow pit that we dove in when we were doing some reef surveys in Aruba in '87. It was a great trip, and you may recognize a couple people here. Anyway, so finished my PhD, went on to do other work. My postdocs led me into doing molecular work. That led me to start looking at really questions that we could address with these molecular techniques. Things like the sexual and vegetative reproduction in corals, how common was it? This led me to look at population structure and genetic diversity in corals. And basically having these tools, I began to then look at the symbiosis. And that's really what I've been working on for the past 2 decades, almost 3 decades. And so my work is focused on symbiosis and the diversity of the symbionts, and really focusing on what the dynamic of symbiosis is. One of my, really passions is to try to understand how this symbiosis is established. What factors affect the establishment of the symbiosis and the determination of the actual symbionts that are in the host throughout ontogeny and as an adult. So I looked at things like what are the environmental determinants, what are the parental or genetic effects. We also looked at a lot of physiological variation within the symbiont and how that affected the host. Hosting certain types of symbionts, how does that affect host physiology, host genetics, and so on. And one of these was how the symbionts affect stress. And so this is what I really want to focus the last bit of my talk on. As we all know, the world is getting warmer. We know this leads to increased ocean temperatures. We know this leads to increased coral bleaching. So here I am, come back full circle. So basically, we know that the intensity and the frequency of bleaching events are increasing. So what have been the outcomes of these events? Well, initially, I think the outcomes we would all say were recovery. We'd see in the late summer, early fall, we'd see coral bleaching, and then by the end of the fall, early winter, we'd see recovery. But now this is the outcome much more frequently. Much more often the corals die, and a lot of this is related to disease. But the other thing that everybody has been noticing is that there's variation in the response to this stress. So here we have a bleached Eusmelia surrounded by a couple small heads of Orbicella annularis, a bleached Pseudodiploria surrounded by seemingly healthy Antilogorgia, and then even within the same colony, an Orbicella colony, a Pseudorastra colony, we see variation in the bleaching response. And so this has led a lot of us to say, okay, so what's the role of the host? What's the role of the symbiont in these bleaching responses? And as Andrew mentioned at the very beginning, one of the things that a lot of people are asking, what's the potential for the host or the symbiont to adapt? We have this increased water temperature. Is there enough variation out there for the host or for the symbionts to adapt to this increased temperature? So what we're asking now is not really, can we create a super coral or a super symbiont that we can put out in the wild and survive the temperatures? What we're asking is, we know that a coral generally has a single dominant symbiont type, but we also know there's a lot of background symbiont types within that coral. So given that the world is changing, will these background symbionts, are there some thermal tolerant symbionts there that can actually help the coral and provide the coral resistance to the temperature? So what we've done is we've basically collected the natural populations within a coral host. We've reared them at 26 degrees, and we've reared them at 30 degrees. This is work that I'm doing in collaboration with Casey Turst at UC Northridge. And after having maintained them for 2 years at these temperatures, and actually now it's about 3 years, we've ended up with a bunch of genotypes that have a cold history and a bunch of genotypes that have a warm history. So now we want to ask, was selection, were there genotypes within that population that were more thermal tolerant, or even given the fact that the symbionts have a very rapid generation time, did mutations occur that may have given them the ability to survive at those temperatures? So what we've done is we've looked at the growth rate of these different genotypes at elevated temperatures. So we took those with a cold history and reared them at 26 and 30 degrees. We took those with a warm history and reared them at 26 and 30 degrees. So what are some possible outcomes? Well, one thing is that we could get selection. So here we have the genotypes with the cold history, the genotypes with the warm history. These were reared at 26, these were reared at 30. And that's how all my next couple slides are going to be. So basically, if selection occurred, you'd expect those with the warm history maybe to do better at 30 degrees than they did at 26. All the symbionts could just still be thermal sensitive, so that no matter what their history was, they'd still do better at 26 than they did at 30. Or maybe they're all thermal tolerant, or some of them are thermal tolerant, in which case you'd expect no matter what their history is, they'd do well at both temperatures. So we did this experiment. We reared the cold history and the warm history genotypes at both temperatures, and these are the results. So basically we have mean growth rate for the cold history, the warm history, reared at 26 degrees and reared at 30 degrees. And what we see is there's no effect of temperature. Whether they were reared at 26 or 30 degrees, didn't matter what their history was, they have the same, more or less the same growth rate. So that suggests selection did not occur. But there was a significant difference between genotype. We see some genotypes were pretty thermal sensitive, no matter what their history was. They grew well at both temperatures. And others were more thermal sensitive. They didn't grow as well at the temperatures. So then we said, okay, well, do these thermal tolerant symbionts confer benefits to their host? So we did the same type of experiment. This time we took newly settled polyps of Antilogorgia bipinnata. We inoculated them with each of the genotypes and then reared them at 26 and 30 degrees. We looked at symbiont uptake, if there was any variation with that. We also looked at and then we also looked at host survivorship. So this just shows the symbiont uptake, the mean number of cells per polyp after two months, with them again being reared at 26 or 30 degrees with a cold history or a warm history. And again, what we found there was no difference in temperature. It didn't matter which temperature there was reared at. Overall, there was no significant difference between temperatures or between genotypes. But we did find a significant temperature by genotype effect. So what this means is basically whether it grew well at 30 degrees or 26 degrees depended both on the temperature it was reared and also what genotype it was. We also looked at survivorship. So basically, at 1 month and at 2 months, there was no difference between genotypes, but there was a significant temperature effect. So what we saw, those that were reared at 26, those polyps that were reared at 26, did better than those that were reared at 30, regardless of what the symbiont genotype was. But I also want to point out that those who were reared at 30 actually did survive reasonably well for the first month. So to summarize, what we saw was that basically there was variation at the level of the genotype. Some were thermal tolerant, some weren't quite as thermal tolerant. And there was also no evidence of adaptation. Those that had the history of being reared at the warm temperature didn't really do any better at 30 degrees than they did at 26 degrees. We also saw that the holobiont does survive at least for a period of time at 30 degrees. So this kind of gives us a ray of hope. We have a thermal tolerant symbiont with a host that can survive with that symbiont. So maybe this will give, if there's those corals out there have these symbionts that can be selected for, it'll give at least the ability for the corals to survive for a bit longer to maybe we get our act together and we can do something to reduce or at least stop this warming. And so with that, I'd like to thank my co-authors, 3 undergrads that worked on this project, my technician, my collaborator, a score of undergrad volunteers. This is only about a fifth of them. About 40 students come in for 4 months to take care of all these polyps. We had like, I don't know, thousands of polyps that we were growing. And then Howard Lasker and also the staff at KML. I'm happy to ask any questions. And again, congratulations, Peter.
Time for a question?
Yes.
So some of your data supports the symbiote shuffling hypothesis, and some of it seems to contradict it. How do you sort that through?
I think, well, what we were looking at, we were not, in this particular case, they were not shuffling symbionts, different species. They were shuffling different genotypes. And I think that's I also think that if the coral harbors different species, which most corals, a lot of corals don't harbor more than one species. But if they harbor different species, then yes, there's definitely, I mean, Andrew's shown that there's shuffling. But what I'm saying is within a single coral community where you have the same species, there are variations in those genotypes that can be shuffled around.
Yeah, did you go? You can also speak into this microphone if you. All right. While he's getting suited up, it is my pleasure to introduce Bill Precht, who will be talking about disease and death.
So I came to RSMAS in 1986, and I came here.
Try it now.
Is that better? Okay, sorry. So as I was saying, I came here in 1986 to work on my dissertation, and specifically to do work on the geology of reefs in Belize. And I specifically came here to work with Dr. Robert Ginsberg, who's a world-renowned carbonate sedimentologist. And it was one of those fortuitous things that sort of happened that, you know, serendipity. Bob Ginsberg was supposed to lead a field trip for the oil industry personnel that supported his lab, and they were supposed to take a trip to the Bahamas. And lo and behold, there was a hurricane heading towards the Bahamas, and he had just had these 15 geologists from all these oil companies arrive in Miami, and he can't go to the Bahamas. And then after that, the hurricane was supposed to come to Florida, so a Florida trip was out of the question as well. So Bob Ginsberg, the organizer that he was, put together a trip to Belize in literally 12 hours. He just got everybody involved and said, okay, we're shuffling from the Bahamas to Belize. We're going to run this week-long program in Belize. So we head down to Belize, and we go to one of his favorite reefs, a reef called Channel Key, which is where he had had a former master's student do a master's thesis on the reef at Channel Key, where he had courted. And it was a reef that was comprised of Acropora cervicornis, and it was one of the most amazing reefs that you've ever seen. And as most of the people in this room know, that the reefs of the Caribbean back in the 1970s and 80s were dominated by 2 species of Acropora. Acropora palmata in shallow water, and Acropora cervicornis either in the back reef or in deeper waters out on the forereef. And this is the reef. an aerial view of Channel Key Reef, which is one of the rhomboid shoal reefs in the lagoon of Belize. And this, again, was an area that Bob Ginsberg, Gene Shin, and others at the University of Miami and USGS had been studying for about 10 years. So there's a lot of data on this reef. And when we went out, we noticed corals that looked like this. And there were not just one or two corals that looked like this, but hundreds and hundreds of corals that looked like this. And Bob Ginsberg said to all the oil company people, you see all that white on those corals? That's from fireworm mortality. And basically what fireworms do is they will engorge themselves over the branch tips and will eat and suck the tissue right off the coral, leaving a white skeleton. And I'm looking at these corals, and I'm going, it's very hard to pass a bifurcated tip. How does the fireworm do that? Can't do that. So I didn't want to disagree with my advisor and say, I don't think that's what's going on. So I kept my mouth shut. And then in the spring of 1998, I had the amazing fortune to take Peter Glynn's coral reef ecology class. It was actually coral reef ecology and physiology. It was co-taught by Dr. Lena Jmont as well. And our cohorts in that class, most of them are in this room today, which include Nancy Gassman, Josh Feingold, Mark Eakin, Jorge Cortés, and on and on and on. And it was really probably the best class and the greatest experience I had in RSMAS, and one where I also determined that I probably shouldn't have been in geology, and I probably should have been in the biology department studying ecology. But the other great thing that happened in the spring of 1988 was I met Rich Aronson and started working with Rich. So now I'm basically working with Peter and Rich, and after we're talking about coral diseases in the coral reef ecology class one day with Dr. Glynn, I came up to him and I said, I have some pictures I want to show you from Belize. And I showed him this picture, and he said, well, Bill, are you familiar with the work of Bill Gladfelter down in the Virgin Islands where he described white band disease? And I'm, no. So I proceed to go to the RSMAS library, and I go and I find this wonderful paper published in 1982 in the Bulletin of Marine Science. Published in 1982 based upon research done in 1976. And this is the way science was done back then, right? You would type a paper on a regular typewriter and you'd mail it into a journal, and then it'd probably take 2 years before you'd get your paper accepted instead of the instant gratification that we have with certain journals today. But Bill Gladfelter had seen this disease, and when he was writing this paper, he asked a bunch of other scientists working around the Caribbean if they had seen something similar. And you can see the quote that I have, it has also been observed in Panama, Peter Glynn, Pers Com. So obviously Peter was there at the birth of white band disease as well, but that paper and then a subsequent, and that was basically the white band disease on Palmata that Gladfelter pointed out, but it had also been occurring in Cervicornis. Nancy Knowlton had written a paper in 1981, actually predating the Gladfelter 82 paper on white band disease in Cervicornis. And basically, that we went out, and this is what Channel Key Reef looked like in the Belize Lagoon in 1986, the first time I went to see it. And by 1990, that's what it looked like. And we had a 99+% mortality of Cervicornis at all depths on every reef in the Belize Lagoon. Catastrophic. It was unbelievable. Rich and I then cored these reefs, and we published a paper together on this in 1997 in Journal Paleobiology, and we did some coring work with it, and this is basically a close-up of the white band disease. Then, when we're working on our coring project, Peter Glynn's colleague from the Smithsonian and longtime friend Ian McIntyre was working with us down at Caribou Key in Belize. And Ian makes a comment about white band disease and about the paper we had published. And he said, you know, it's really weird that you saw that disease break out in 1986, 87, 88 in the lagoon, because on the fore reef it happened in 1983. And this doesn't fit normal models of the way diseases are transmitted, especially for what we learned from the 83, 84 diadema plague, where you could actually almost time the first arrival of your disease at the next location following the ocean currents. And why would the fore reef die in 1983 and the lagoon not get affected till 1986? So how did I know that 1983 was the year that this happened? Well, at the picnic table at dinner, Ian tells the story of when everybody was first starting to think about white band disease right after Gladfelter's paper came out. Jerry Wellington, again, longtime colleague of Peter's, was at University of Houston and wanted to have a PhD student study the potential effects and go to a place in the Caribbean where the disease had not yet affected the reefs. And called Ian McIntyre and said, Ian, when was the last time you were in Belize? And he said, I was there in September of 1982. And he said, how did the cervicornis look? He said, healthy as a clam, looked great, fore reef was beautiful. So Jerry sets up a trip with his student to go to Caribou Key, get some Smithsonian funding, shows up at the lab in June of 1983, and 100% of the cervicornis on the fore reef had died. So in 1 year, from 1982 to 1983, the corals had essentially disappeared from the Belizean fore reef. And I didn't know that story until after Rich and I had already done our work in the lagoon. So Rich and I are just having conversations, and we basically looked at each other and said, you know what we need to do is we need to do an analysis Caribbean-wide and try to figure out where and when white band disease affected these different places. So we wrote a chapter that was in a book on evolutionary paleoecology. And this chapter is the chapter that we wrote. And that table, or those 2 pages of tables, were basically based upon a month worth's work in the RSMAS library, going through the stacks and going through gray literature, CARICOMP publications, lab publications, gray literature, abstracts from different meetings. And basically what we were looking for is any mention of a cropper of cervicornis or a cropper of palmata that was found dead in place or had been identified to have died by disease. And all of a sudden, when we started putting this together, all of a sudden there wasn't a place in the Caribbean that was not affected by white band disease. And this included palmata and cervicornis. And that led to Jim Porter asking us to give a presentation at an ASLO meeting, which resulted in white band disease and the changing face of Caribbean reefs paper that Rich and I wrote. And I almost shamefully say that paper's been cited more than Bill Gladfelter's original 1982 paper. And it's really a shame because that 82 paper is really the foundation for all disease work on Caribbean coral reefs. It is the foundation from the shoulders on which every paper on coral disease stands, basically. And when we put this map together, we could not believe the results. And this was even before meta statistical analysis had really taken off. What this led to was for us to core all the reefs throughout the Belizean lagoon to see what the history was of these reefs. And it led to a very, very close long-term relationship with Ian McIntyre. Over the years, we took more than 50 cores together throughout the Belize lagoon. Those are just 2 of the papers we wrote with Ian on our work. And it all stemmed from this disease that Peter guided me to, basically as a side project that was related to an oil company field trip. That, you know, so you just never know where you jump off. But that 1988 coral reef ecology class was probably the most important class I've ever taken in college anywhere. And the friendships that I made, both from Peter and all the students that I'm still very close with, I will never forget. Thank you.
We have time for questions.
Peter.
Where these occurrences of disease have taken place, has there been any demonstrable recovery of any sort, or is it pretty much a permanent degradation?
It has been a pretty much devastating story for the acroporids Caribbean-wide. And there's been a probably Caribbean-wide, you know, people throw different statistics around, but certainly greater than a 95% total loss in the acroporid population from the 1970s till today. Very few areas have recovered, and they're listed as critically endangered, both species, on the IUCN Red List, and they're listed as threatened on the US ESA.
Thank you.
All right. Thank you, Bill. Next, we have John Van Leer with the solution.
Good afternoon, and thanks for the opportunity, and thank you, Peter. Question of what corals really need. They actually need to have global emissions go below zero. That means we need to not only reduce emissions, which people talk about frequently, but we actually need to sequester carbon in a serious way. So my personal background, I don't have a direct connection to Peter, but as a graduate student at MIT in Woods Hole, I was one of the first graduates in that program, and so when I finished, I was looking for a place to work, and I knew Klaus Root from Miami. He'd been at Woods Hole, and so he said, you ought to look at what was going on with RSMAS. Pardon? Okay. Tell you what, we'll take the middleman out of the equation here. All right, so the attractions to coming to Miami was the opportunity to build a moored robotic profiler, something I'd wanted to do as a graduate student, but the people at Woods Hole said it wouldn't possibly work, so I couldn't do it. It's a sailor's paradise down here in South Florida, and had an opportunity to live in Key Biscayne. There were luxuriant coral reefs at the time in both Florida and the Bahamas. As a junior in college, I'd hitchhiked down to Key West from Washington, D.C. with 650 pounds of diving gear to see the reefs off of Key West, and they were magnificent. And to corroborate your story about the noise with reefs, they were noisy. I mean, it was just incredible, the coral cover, the quality of them. And so when I was looking for a place to go to work, a place that had beautiful corals like that was clearly on the list. And of course, then we had the Gulf Stream on our doorstep. And in addition, we had tropically focused faculty down here, so you could learn something about reefs and geology, as well as the physical oceanography, which I was working on. And of course, RSMAS in those days had excellent ship resources, so that was a net thing. And so working in Walton Smith's version of RSMAS, I was able to go to diverse seminars across disciplines, which was just fantastic. And it was like sitting at a smorgasbord, really inspiring with good coral reef biology and geology. Unfortunately, we lost many of the best known coral reef researchers during the last years of Warren Worcester's term as dean. So then we went into a period where we didn't have that kind of diversity and that kind of interest going. And finally, Peter Glynn came up from Panama riding on his white horse, possibly a bleached horse. And then the coral reef research in Miami not only was a renaissance, but it was almost an explosion. It was really quite amazing and gratifying to see coral reefs come back. So one of the things that we haven't really talked about up to now is what is the future, and it's not a very pleasant one if we keep doing what we're doing. And this is a graph by the IPCC, a special report on oceans and cryospheres from Chapter 4. It actually goes out and looks at 3 centuries time frame. In South Florida, we typically look at 50 years, maybe 100 years long-term planning. And there's a tremendous difference in sea level rise between trying to remove CO2 and essentially get down to the 2 degrees C above pre-industrial levels. So one set of curves continues to rise, has very wide error bounds. There are a lot of unpleasant things that can happen if we allow emissions to continue. So the other one begins to slow down in rise. So it means that corals could possibly keep up with something like that. Really quite a different kind of a future that we can have. There's another special report that came out in the fall of 2018 responding to what the island nations and low-lying countries had asked for at the Paris meeting. What could we do to stay within 1.5 degrees C above pre-industrial? And that is really the most practical thing that we can do. It does require emissions reductions of about 50% by 2030 and 90% reduction by 2050. And that is widely considered as improbable, unlikely, impossible. You pick the adjective. So I've been working with a group called Citizens Climate Lobby, which actually works with Congress to try to reduce emissions, and they do it in a courteous and a bipartisan way. That sounds like an oxymoron in this time, a bipartisan way. RPC 2.6 requires rapid emissions reductions. 1.5, you've got to go a lot further than that. And so the graph to the left shows the difference in sea level rise depending on a bunch of different climate scenarios, 4 of them. 8.5 is essentially what we're doing now, and it's a pretty undesirable future if you happen to study corals. The 2.6 is much more favorable for corals in the future, as well as humanity in general, and so that's what the program CCL has been working on endeavors to do. So there are things that are already being worked at scale. You can work to elect candidates committed to strong emissions reductions. That's the usual political process. You can lobby to pass legislation which prices carbon high enough to account for its damage so the economy will drive the required transformation. That's what CCL, Friends Committee, and a number of other groups do. You can litigate cases up to the Supreme Court if necessary. That's what James Hansen is doing with his children's lawsuit. You can block construction of pipelines or export terminals by all means possible. That's what Greenpeace does. You can promote institutional divestment of carbon securities. That's what 350.org does. They have succeeded in about 11 trillion so far. There are also large numbers of individuals who have converted their lives to achieve negative carbon emissions and complete carbon divestment. But what change could actually occur faster? And the answer is banking. There is an excellent article written by Bill McKibben in the New Yorker, September 17th last year. Basically, money is the oxygen which fires the global carbon emissions burn. What if banking and asset management and insurance industries moved away from fossil fuels? That is the fastest thing, and I think there's a better chance now than ever before. So why would bankers abandon fossil fuel? We talk about climate tipping points. There are a lot of economic tipping points as well. One of them is that the price of solar photovoltaic panels have dropped 90% in the last decade. The price of wind turbines have gone down about 70%. The price of lithium-ion batteries to store the energy that you produce from intermittent sources has gone down at least 75%. And most recently, the coronavirus is reducing the demand for oil and the price of oil internationally. So if you're a banker, are you going to finance more fracking if you already have an oversupply of oil? So today, 30% of global car buyers, according to a AAA survey, plan to buy an EV in their next purchase. Grid-scale lithium-ion batteries are much cheaper and more efficient to operate than gas-fired peaker plants. That's tremendously significant because it means it's putting peaker plants out of business. In Southwest Australia, they put in a giant battery, and essentially the peaker plants lost 40 million in year one, 110 million in year two, and they're out of business. GE has based their future turbine sales on peaker plants, which are not happening because of the efficiency of the batteries. They not only store energy, but they dispatch it in a millisecond to the highest bidder. You cannot compete with that. With a peaker plant takes 20 or 30 minutes to warm up and get up to production. Another thing is the banker's children might actually take part in Fridays for Our Future or Extinction Rebellion. So he might have to deal with his own children. So for continued existence of corals, we often unpackage Christmas toys and they say batteries needed. Well, if we're going to have a transition to renewable energy that's intermittent, we better have batteries to store the energy in. And those batteries better be abundant, they better be cheap, and so that's a necessary condition. It's by no means a sufficient condition. It's something we need to do to reduce demand. So we see gigafactories sprouting up. The first one was in Nevada. When it's finished, it will produce 100 gigawatt hours of lithium-ion batteries per year for at least 30% lower price and net zero energy. It essentially uses sunlight, it uses geothermal power, and it uses wind power, and doesn't require any excess energy. So basically now Tesla has 4 gigafactories, 2 well underway and 2 more in the pipeline. There are 5 other battery makers that have gone to large scale. We only need 100 of these things to store the energy for the entire world, so we are on the way. If you have not gotten a copy of Drawdown, this basically discusses the other options in not only power and transportation. 80 drawdown solutions save $44 trillion over 3 decades compared to business as usual, and can reverse global warming if done fast enough. We'd be nuts not to open this toolbox. We should do it for the profit, even if there were no global warming. So this is the pie showing the different options. They group by sectors. You notice that food is actually the biggest one, bigger than electricity generation, land use, women and girls education, and also family planning is a huge component of this because everything scales with population. And so there are materials, buildings, and so you see there's a lot more we have to do besides lithium-ion batteries. And so this is the Energy Innovation and Carbon Dividend Act, H.R. 763, which is actually a bipartisan bill, which is already in Congress, going through the committee structure in the House of Representatives, and this will enable those climate solutions. So for over a decade, CCL volunteers have been interacting with their Congress member to support this. It's got to be a big solution. It's got to be a lasting solution. Therefore, it has to be bipartisan, and it has to be legislation. So this policy will reduce America's emissions by 40%, first 12 years, 90% by 2050. It's good for the people, improves health, saves lives, and so forth, and it is revenue neutral. That's what makes it palatable to both sides of the aisle. And it's good for the economy, and it's bipartisan. And because it's global warming, not USA warming, there has to be a border adjustment so that people who offshore production have to pay for the embedded carbon at the borderline, and exporters are compensated as well. And so this is what it looks like with 1,200 CCL volunteers preparing to lobby Capitol Hill. This is saturation lobbying, but polite and respectful. And if you'd like to learn more about this, we have a meeting going on next Saturday over in the Unger building. If you'd like to put your shoulder to the wheel and help make this happen, we can use all the help we can get. Thank you.
Sorry, we do not have time for questions. All right. It's now my privilege to introduce yet another colleague that I respect very much and a collaborator as well. Proud to have her as a collaborator, Dr. Nikki Traylor-Knowles.
Thank you. Thank you so much for having me. It's a pleasure to be here and an honor to speak today. When I was coming up with the title and thinking about what's my connection to Peter, there's actually quite a bit. And one of the things that initially, when I came up with the title, I thought of this, which if you're familiar with the Muppets, the Rainbow Connection. But the way that I like to think about it is through the Apostle Opera connection. And I'm going to talk about this today because it's actually kind of wild how many connections I have with Peter, and I didn't really fully realize it until coming up with this presentation. So it's really humbling, and I realize how much Peter's work has actually helped shape my work now, which is very different, and I'll talk briefly about it. But his work has helped foundationally, in both with the corals that he has worked on and the things that he has found with bleaching, to help shape my current research platform. So I kind of came up with a timeline for this because it was interesting. So when I started as an undergrad, I was in Baltimore, and there's no tropical corals there, so I started working on Apostle damicornis. We were using a lot that were sourced from the Pittsburgh Zoo, but we also used some that were sourced from RSMAS, and specifically from trips that Peter had made. Then in 2005, this is a huge confession, I actually applied for Peter's lab. I didn't get in, but I did apply. Ended up going to Boston University, and again, I started working again on Apostle Opera, and I actually worked a lot on the Pan 10, the Panama 10. I don't know if the pointer's here. Which they're here growing. You can see they look like they're Apostle Opera on steroids. We fed them a lot. So I did a lot of work on wound healing on those specific coral genotype that Peter had collected in 2005. Then I was at Hopkins Marine Station, and while I didn't actually overlap with Peter, but that was a connection that we had of both being there at different times and a shared experience. And then just as a side note, I had my honeymoon in Panama, so that's a connection. And then I came to RSMAS and was actually hired once Peter decided to retire. And so now I've had the pleasure of actually finally getting to work with Peter and his corals. So it's been just amazing. He's helped me have better connections here. For example, my student Mike Conley just was able to go to the Galapagos and work, and we're trying to start some field work there, and so it's through those connections that we were able to do that. And also working with the Invertebrate Museum, Peter was instrumental in introducing me to Nancy Voss and helping me kind of work on the transition of being a part of that. So I thank you so much, Peter, and I don't know that you realized how much influence that you've had. So this is my lab, and my lab primarily studies coral immunity, and we're really interested in how immunity can shape coral health. And so we take a lot of different approaches for this, and the sizes of these circles are sort of to denote sort of the areas of focus, but we use a lot of different organisms to study these different areas. And we focus on a lot of different techniques to get at answers within these areas. Ooh, it's very touchy. And we also do a lot of different types of experiments, looking at different types of cells, which today I'll talk about immune cells. But all of this is with the understanding of trying to understand coral health. Sorry, it's just moving. So my question is, why do I want to work on Passalopora damicornis? I get this question a lot because being in the Caribbean, I should focus mainly on Caribbean corals. However, Passalopora, this is Pan 10. We now are growing a bunch of these in my lab. It's weedy and able to survive in these kind of crappy environments, has stress tolerance, and it's really hardy in lab culture. And one of the things that I'm particularly interested in is how that ability to survive in crappy environments, I guess that's subjective, but how the immune system is involved with that. So one of the first goals that I had when I got to RSMAS was to sequence the genome of Passalopora damicornis. And in particular, it was the corals, this Pan 10, that was collected in 2005 by Peter Glynn and colleagues. And so I won't go into the hardcore details of this, but just to say that what we did is when we sequenced the genome, we actually compared it to three other available genomes at the time. And what we found is that our genome, in relative size and assembly and completeness, it was the best at the time. So we were really proud of that and are really excited because this is a really valuable resource. But coming back to the idea of understanding Passalopora as a model for immunity, we wanted to look at, if we look at all the different genes that might be shared amongst all these different corals, I wanted to look specifically at the genes here to see if we see enrichment for immune genes, and that might be one of the reasons that we see such a high ability for these corals to survive in seemingly fringe environments. And you don't have to understand anything that's being set up here, but the main thing to take away from this is that there is enrichment for immune pathways, and these are things that I'm hoping to, in the future, actually start to study at a more cellular level. So the other thing that has been a big drive for me has been to understand the cell biology of corals. And Passalopora is a great model for this. And again, because we can keep it in the lab and it's very easy to propagate, we can get lots of different replication of it. And it's also, there's a lot of methods that have already been developed for doing cell culture with these corals. However, with coral immune cells, it's been sort of a mystery. So we have documentation coming from histology, but these are preserved samples, right? And so there's a lot of changes that can occur when you're preserving a sample. So we started using something called fluorescence-activated cell sorting. And this is a way in which, it's used a lot in immunology, it's a way in which to sort out different cell types based on their size as well as their granularity. And so we collaborate with people in Israel as well as the medical school to do this. And long story short, we found that there are a lot of different cell populations. There's about 12 in total. But the question is coming back to where are the actual immune cells? So is this population exist? Because there has been some discussion that maybe corals don't actually have immune cells. They just have general digestive cells, and it's not really an immune cell. So we did an experiment where we took coral cells and we exposed them to beads or bacteria. And this is, again, Poecilopora. And what we did is look at the amount of uptake of bacteria-positive cells. So what you can see is that in this circle here, this shift is indicating that there is bacteria within these cells. And this over here is a Symbiodiniaceae. And we know this based on the size and the fluorescence that's being given off. And what we can then do is actually collect these cells and look at them live. And I'll show at the end a really awesome video of one of these cells. Just for quantification, we looked at then trying to figure out what is the percentage of the cells that may be able to phagocytose, so actually engulf a bacteria or bead. And we saw that between 2 and 8%, depending on the type of bead or bacteria that we were using. And I don't have time to show this, but it is different from what we're seeing for digestive cells. So there seems to be a unique cell population that is doing this. So we were very excited about that. But nothing, you can look at all these graphs and things and whatever, but really pictures and videos to me are what are really exciting because you want to actually see it. So these are some pictures of these potential immune cells that we found, where we have these round morphology that are almost more reminiscent of a type of digestive cell, but we think that they are having an immune function. And then we also have these more amoeba-site type morphology. So they look visually, and they actually are visually different in terms of size and granularity. And so we're investigating these more. And so this is a picture of one of these cells moving, and you can see that there's actually Philopodia that are coming out, and it's searching through its environment. The green that you see is actually beads that have been phagocytosed within the cell. So this is the first time that we've actually had a video of this, and I'm really excited about this because I think that now the next step is to culture these and to start actually really investigating what they're doing and what are their limits and their interactions with other cell types as well as other types of bacteria, viruses, whatnot. So again, my take has been very little. I really focus in on genes and cell biology, but all of this has been bridged, again, by this coral. And now I guess we can add in these other tools with this coral. And one of my goals is to continue working on this coral and to continue propagating it to be a model that many different people can use, and we can build up a lot of resources for this on this particular genotype. So thank you, Peter. Thank you so much. And I'd like to thank all the graduate students that were involved with this and the funding, as well as my collaborators. Thank you.
Time for a question? Yeah.
So, thanks, that was great. Have you thought about collecting Poecilopora damicornis from the Western Pacific and maybe the Central Pacific? Mm-hmm. Because they're really different out there.
Right.
And they're very vulnerable to temperature shocks. You're saying it's a really tolerant species. I would say quite the opposite from where I come from.
Well, there's a lot of discussion, too, in terms of just the genetics of these, right? So there's Poecilopora acuta that, is quite different. And I think that is true. It's a natural next step to look at and compare those two. So I know a group in France is actually sequencing the Poecilopora acuta genome. So it'll be great to actually do a full-on comparison. Yeah.
Thanks. Yeah.
Thank you. Yeah, Peter.
Hi, Nikki.
Hi.
I was wondering if you're at the point or will you be, where you can actually test for memory of cell response?
Yeah, you're reading my mind. That's the next step. Yeah, so we would love to see if there's some sort of competency that is learned within these cells, because if that were there, that could be a tool for tolerance. So we haven't done that yet, but that's in line.
So there's nothing been published on that?
No, not in hard corals.
There
There's
you
some-
go. That would be a real breakthrough.
Yeah, yeah.
Thank you.
Yeah, thank you.
Next, it's my pleasure to introduce Dr. Elena Schmott. Take it away. Thank you.
Thank you. I want to again thank Andrew. Can you guys hear me? I can't tell if this thing is, no, hang on. Like here? Maybe point the other direction. How does that? Any better? Okay. I want to thank Andrew and the other organizers for inviting me to come help celebrate Peter's long, very productive career. So I'm retired now, but still working part-time with no pay. That's what happens to some of us when we get older. And I guess everybody's trying to say who was earliest in Peter's life. And other than Peter Jr., I guess I was 17 when I met Peter for the first time, and I found out that it was the first time he taught the marine ecology course in La Parguera, Puerto Rico, when I took it. So I'm going to leave most of my comments about Peter until dinner time, where I'm signed up to say a few emotional words. But for now, I want to say that, you know, when I had to come up with an idea, what am I going to present here that's new, different, whatever, I came up with the idea that maybe not all of our attempts at research are 100% productive, or let's say we don't answer the question. And there's something that I've struggled with for many decades, that with coral reproduction, which I started just before I came here from Florida State. And it's something where Peter and I have talked about over the years, but we've never actually worked together on this. But anyway, I hope this will be of interest. And it is a conundrum, and I hope there are going to be young people here that might become interested in it, might want to take it on. So what do I mean by the ups and downs of coral settlement and recruitment? An unresolved conundrum. So let's see, what do I do here? Okay, so what is this conundrum? So, you know, when we're coral reef ecologists, we swim around, the corals are everywhere on top of the reef, right? But as I started, my interest in coral reproductive biology began as a graduate student in Rhode Island, where I worked with Astrangia pulculata, then Danae. And one of the first things that I started doing was looking at a reproductive cycle, because I wanted to know when it got its zooxanthellae, and based on the literature back then, coral larvae the corals were all planulators, and they all came pre-equipped with zooxanthellae, and so I was trying to figure out, well, when does this happen for Astrangia? And it turns out they were spawners, and they acquire zooxanthellae sometime later. I never really was able to get them to settle. They were sort of a strange coral. And actually, Peter had invited me to give a presentation down in Panama when I visited in '75, I believe, at STRI. And then after I finished my Ph.D. on Astrangia, and I was trying to figure out what do I do next, I said, well, I want to continue working on coral reproduction. There's a lot of things we just really did not know in 1980 about coral reproduction. So I started in La Parguera with that course. I used to live in Puerto Rico, and so Caribbean was my home, and I wrote my NSF proposal, got funding to study the reproductive cycles of Caribbean corals. And after a few years doing that and figured out when took me about 10 years to figure out when they actually spawn and actually observe it for the first time. Well, now we had the ability to collect the larvae, and then it took a few years to figure out how to raise them all the way through development and get them to settle. And then now what? Well, what's the point of coral reproduction? It's to replace and augment coral populations. So the important part of all of this was what happens to the larvae once they become competent and want to become corals. So, but when I started playing around with coral settlement, the first thing that came about is like they always wanted to settle in undersides and cryptic parts of the substrate, not on top of the reef where we surveyed for coral recruits and things like that. So then started a long series of experiments trying to understand the settlement behavior of these animals. And I'm not going to emphasize too much the data. I'm going to try to just hit the highlights of what we learned along the way. And this work took large numbers of paid staff, volunteers, graduate students, postdocs, and anybody that wanted to come along and help. I want to specifically thank Margaret Miller, who I think is maybe still here, and she was instrumental to everything, and Ernesto Weil. Without them and many others, I didn't put all the names up here, this work would not be here. So the first step was to, just to summarize, this is like 10 years of work, trying to figure out how their reproductive cycle, when to go out diving, what time of night, what to do with them after they spawned, how to raise them. The first year, actually, I think, Susan was I think we poured the larvae down the drain because we didn't think they were doing anything because they looked totally different from anything I'd ever seen published. I literally was, after 24, 48 hours of no sleep, said, oh, this didn't work. And then I found out the next year, this is what they look like. So anyway, so after finally being able to raise them, we got to the point where we could actually try to do experiments. And all the data I'm going to present today, most of it is going to be using field age settlement plates to try to reproduce the natural environment as much as possible in the laboratory. These are all quarried limestone that were like, you could see the cut coral through it, so it had all the nooks and crannies and for the larvae to climb into. And then I learned how to use fluorescent microscopy. Actually, Mike Schmale's microscope was the first one I used, and help us like, wow, look at those little guys, they really pop out. These are Orbicella fabulata. You're going to see some slides with Montastro because they're old slides. And then from there, we could do the experiments that I'm going to talk about next. So the first thing, the first time we did a whole bunch of coral settlement, Margaret was involved with this. This is Wellwood and Little Grecian. We had all these plates that we had aged out there. And when we bring the plates in to look at where we got settlers, they're all on the bottom, not on the top. I'm like, huh, that's weird. So I said, well, let me do an experiment here to see, because this is, I would think, well, maybe they're avoiding the light, right? We know that there are a lot of organisms that want to hide from the light when they're swimming around as larvae. So I took a tile that was settled, that was well aged, and I broke it in half, and then I put it in the laboratory in a trough, and I put half of it facing up and half of it in the original orientation. And the larvae settled on the bottom side of the plate, regardless of what direction they were facing. So light was not a cue in terms of settlement. And I'm just going to sort of do pop-ups here in terms of what did I learn from this. Is like, well, light is not a signal. It's got to be something else. And it's probably dependent on something that is growing on these plates. And what's growing on the plates is not the same on the top and the bottom. So the next thing we did then was carefully map under the microscope every settler on every plate. And we see by doing a Poisson analysis that they have an aggregated settlement pattern compared to a Poisson distribution. And then if you plot the percent of the spat versus the relative cover of different types of organisms, I'm too short, I can't point above that thing. We see that they seem to associate more with more than frequently with certain types of substrate that were in greater proportion than what the substrate cover was. So the lesson learned is that there seems to be settlement cues that are associated with different types of organisms that are attracting the larvae to settle. Now, there's been work going on by others showing that coral and algae might be important. Microbial films was not really much known at the time. But at this point, I had an opportunity to move to work back in La Parguera. Some of these reefs will be very familiar to Peter. And we selected, with Ernesto Weil's help, we selected 3 sites: inshore, mid-shore reef, and way offshore at the shelf edge. And we put settlement plates out, 15 to 20 per site, 2 depths at the 2 inner stations. And we had all of these plates all aged, and we randomly, after spawning, we brought them into the lab, and we put them on these skewers so that 3 different aquariums, the plates were arranged so that every combination of top and bottom were possible. So the top for inshore shallow was with inshore, offshore deep, et cetera, all the way through. So the larvae were dumped in and allowed to swim around and decide where they wanted to settle within this system. And the important part here is that the larvae really could choose between a top and a bottom and whether they liked offshore or inshore or deeper or shallower. They had access, every combination was possible. And when you look at what we got, first result, which was interesting, was that the settlement density increased with where the plates were aged, and there was more settlement offshore than inshore, and the middle shore was in between. But in every single case for 2, 4, 5 sites, the bottoms always had more settlers than the top. So a larva would swim between 2 plates and say, this is the top, I don't like it. This is the bottom, aha, I like it. No matter what the top and what the bottom came from. So what is it about the undersides of these plates that makes them so attractive to corals? So we spent a lot of time taking all of these 80 plates and mapping out the larvae and also characterizing the community structure of the bottoms and the tops. So here's MDS of the tops versus bottom. You can see they really separate out from each other. So they're different community structure. This is using primer. And then just at the bottoms, because that's where most of the settlement is, the 3 sites really differed in terms of their bottom composition. We're really excited, right? We're going to get someplace. Not really. So we do know that the bottoms are definitely more attracted to the larvae, and the sites are different from each other. Now can we see which taxa actually explain where the coral settled? And sadly, the answer is no. And so, I've looked at this thing for a long time, and I can't make any sense of it, so I'm not going to waste your time. So the point is that while we expected microfilm and CCA, the higher the cover of that, we would see more settlement, but that didn't quite happen. And so I started to realize, well, I'm an old-fashioned biologist, and maybe these methods we're using are not sensitive enough to really look at it. We need more sophisticated methodologies, molecular methods, to try to identify what's on the bottom that is attracting the larvae, and that's why I wrote here, the devil's in the details. So, but I'm not one to give up. And so we decided to come up with one great new experiment. I said, well, if I can't figure out what the cue is, at least I can figure out what are the environmental cues that determine what's growing on the top and the bottom. And the obvious ones are how much light you get, right? Upside gets lots of light. It also gets a lot of grazing. So we did this sort of Jerry Wellington type of, and I love Jerry, and it was so sad to lose him. This control experiment, these racks, this is what they looked like. We had little control blocks, and then we had shaded, and we had grazing, reduced grazing, and then we had all the cage controls for reduced grazing and shading and controls and whatever. So we had all of these different treatments. This time I used little blocks of limestone. It was very expensive to do this. Because we had tops and bottoms, but a lot of times they were close to the sides, and on plates you have almost no side. So I figured maybe if they had more sides, we would get different kind of results. So these plates were aged. We went to do the study, and this was the summer of 2006. The summer of 2006 followed the summer of 2005 when there was the major bleaching. We didn't get any spawn for a crop for Orbicella, so therefore we used a cropper palmata, same pattern, and no pattern that we could figure out. So therefore, we did not find an expected effect of shading and reduced grazing on the substrate composition that would affect salmon patterns. So again, we're back to maybe other techniques are necessary. And I'm going to skip this slide because it's basically the same thing. So my conclusions are that coral larvae have a mind of their own. It's not clear what the signal is. It's probably microbial, some kind of a chemical given off by something. And I think somebody else that knows how to do these methods need to take this on. And before I finish, this is pure, gratuitous self-promotion. So what did I do when I gave up doing this? Well, I got a big NOAA grant that lasted for almost 10 years. It ends now on June 30th, to develop new instrumentation for measuring coral physiology in situ. And it's called Kiss Me, and Ian's telling me I'm out of time, but here it is, and a few people here have been trained on it. It's basically like an EKG for corals, and papers are actually now coming out and published by it. We welcome any inquiries because we're trying to get more people interested in using it. Thank you very much.
Our next speaker needs no introduction. And it so happens that if you have a seminar about you, you can speak as long as you like. So I'm not going to keep track of time. It's my distinct privilege to introduce for some closing comments, Dr. Peter Glynn.
Thank you, Ian. Good friends, colleagues, brothers and sisters. I'm just going to say a few words, just 2 slides actually, because this evening at the banquet I'll be giving a longer presentation and maybe some more emotional moments then. So these are some passing thoughts that I put together when I read over the titles of the different talks that would be given today. The first, accelerated population and economic growth are not necessarily good things. And these ideas were espoused in a very controversial book, The Population Bomb, by both husband and wife Ehrlichs in 1968. It was brought up again by the Club of Rome in a book called The Limits to Growth, Meadows et al., 1972. And skipping down now from the green, is this on?
You can also just press that.
No, I don't want that.
A pen? Like a laser pointer?
That would work, yeah. Thank you. So I've noticed over the years that there have been many changes, not only in terms of global climate change, global warming, and so on, but also in increasing population and the effects of increasing population on different coral sites and other diving sites that we visited when I was in high school in the 1950s. And we see that the world population is projected, right now is about 7.8 billion people. The UN projection for 2050 goes up to 9.7 times 9.7 billion people. So that is a substantial increase. When I graduated from high school, the world population was at 1.5 billion. In 2018, the projection was 3.3 billion. So there's been a doubling in the world's population. And when I was in high school, I remember we lived in Coronado, California, which is right close to the Mexican border in Baja California. And a friend of mine, Stephen Flynn, purchased a World War II surplus Jeep. And at low tide, we could drive in that Jeep along the beach in a southerly direction and cross the border and go into Mexico and do diving and collecting and observing of marine life and so on. And then after catching some lobsters and cooking some abalone over the fire on the beach, we would wait till the next low tide and then drive the Jeep back to the USA. So you can imagine there were no fences, no guards, no problems. We were perfectly welcome in Baja California. In fact, some of the people who lived in little homes where we were diving invited us into their places for tortilla and habichuelas, frijoles, and so on. We offered them some of the catch that we made and so on. And it was a very free, open situation. Now try to drive a Jeep across that beach. You can't do it. There's a huge fence there. There are all kinds of restrictions. It's highly guarded. And it's a very different world. Coming to another point, this has always been something that has impressed me during my professional career. A statement made by Preston Cloud, a very famous geologist, the purpose of life, to live it with as much grace and integrity as possible, to enjoy and improve it while you have it, and to leave the world no worse for having been there. And I try to follow that as much as I can. I know it's very difficult now because, you know, we all have automobiles, we use electric power, we all pollute to some extent. But I think that is something that would be wonderful if we could all add to in some way. And a spin-off from that, considering greater resource use and limiting resources, and coupled with greater waste disposal and other impacts of humans on the environment, is it possible for us to leave planet Earth a little better off for our having been here? And I think John had some solutions to this, and I hope that we're able to carry some of those forward. And this is something that I hope that all of us keep in mind and that we are able to contribute toward making planet Earth a little bit better off, but certainly no more damage due to our presence here. And then finally, I think this is an important thing. Embrace your passion for marine biological and coral studies. And I don't mean to be preaching to the choir because already I know you have your passions for this subject area, but it's something that for the coming students and so on, if they develop a real sincere interest, a love for this type of science, then this is something you should hold on to and never let go. You should strive to push it forward and enjoy it and embrace it as much as you can. So, acknowledgments and dedication. I would like to dedicate the talks today and my talk this evening to Nancy Voss and family. Nancy is no longer with us. She is now living in New Jersey. She has left the Marine Invertebrate Museum. But the Voss family in general has contributed about 70 years of biological research, marine biological research in South Florida. And this is a picture of Nancy when she was a graduate student at RSMAS. And this picture was taken in 1954. And here she is sketching some fish larvae. She was probably the first graduate degree at RSMAS, a master's degree, which was earned in 1954. And for many years, several decades, she was our stalwart director and curator in the Invertebrate Museum. I'm going to tell you a secret about that picture. There are two people in that picture. Where's the second one? Well, Nancy was pregnant with her son, Bob, who is a curator at the American Museum in New York. And he studies fossil mammals expert in the field. And so thanks to Rob, he was able to come to Miami and get Nancy and take her up to New Jersey, so she lives close by to him, so she's able to enjoy her family life up there now. So I've listed some people who I've associated with, students, colleagues, and so on. And I'm not going to get into the discussion of who was my first student, where, and when. I'm going to let you figure that out. But I have here Alina Schmont at the beginning of the list, University of Puerto Rico in the 1960s. After I received my Ph.D. at Stanford, I got a job at the University of Puerto Rico in the Marine Laboratory in La Parguera. And I used to give a summer course there. I think it was 6 weeks. Alina, was it 6 weeks? Yeah, 6 weeks. And we would make trips out to some of the cayos, the reefs, and we would spend sometimes 3 or 4 nights out there sampling water quality, surveying coral reefs, and trying to learn as much as possible about reef function on those trips. So Alina was a student, one of my first students in that course, 17 years old. And fortunately, coral reefs became a passion of Alina's, and then she continued on and has had a very illustrious career. Congratulations, Alina. Then we come to Jim Porter. Excuse me. So Jim Porter, as he mentioned, came to STRI, the Smithsonian Tropical Research Institute in Panama, in the early 70s. And conducted comparative studies comparing coral reefs on the Caribbean side of Panama with on the Pacific side. And I had some very enjoyable field excursions with Jim, joint research projects that we got engaged in and so on. And so I'm proud to say that Jim also went on and had a very illustrious career. Just recently retired in the last 2 years? Last year. And Jim tells me that he has flunked at retirement. And I'm proud to say I'm flunking retirement too. I can't let it go. Research is so much fun, mentoring students and teaching and so on. It's hard to let it go. And then Mitchell Colgan. I met and started working with Mitchell at the University of Guam in the 1980s when I was invited out there to teach a course at the behest of Chuck Berkland. And so Mitchell and I became very good friends. And as he told you this morning, I turned him into a marine biologist, from a marine biologist into a dirty geologist. But nonetheless, the close association of coral reef functions in biology and geology overlap to a great extent, so he was able to contribute some very substantial understanding of bioerosion and intermittent coral growth in relation to El Nino events. Then we have several people in the 1980s, and this is where I'm going to avoid the controversy because I grouped them all together in the 1980s. Margaret Basile, Aaron Yedid. And then we come to the 1990s with Andrew Baker, Jorge Cortés, Mark Eakin, Josh Feingold, Julie Furman, David Obura, David Smith, Juan Maté. And the 1990s, and we have the little asterisk here. So we have an acknowledgement for these good folks, and also Dean Roni Avissar for allowing this retirement symposium to take place. And so I take my hat off to these good people for putting this together. It's been a tremendous undertaking. Andrew has it. And I think I told Andrew at one point, I'm sorry, I'm not going to be around to help with his retirement. So he can't get even with me. And then from 2000 to 2009, we have another set of students and colleagues, Ileana Baums, Hector Reyes Bonilla from Mexico, Rob Carter, and I think I'm a joint sponsor or with you, Alina. I think we were jointly on his committee. Pardon me?
I left, so you had to take over.
Oh, I had to take over, okay. But at one point, I think we had to jointly sign the dissertation. Yeah, and so Gina Hawkinsmith, Derek Manzello, Allison Molding, Jill Richardson, Tyler Smith, Juan Maté, Bernardo Vargas Angel, and these were all from 2000 to 2009. Ian Enox, Carolyn Margolin. Ian, you are not my last student. I think Carolyn was. Is that right? Yeah.
Last accepted.
Okay.
Yeah.
Oh, last acceptance.
I had 4 years and there was absolutely no deviation.
Okay. I only gave him 4 years. And Bernhard Riegl, he's an NSU outlier, but he has very much become a part of our team, doing research in the Eastern Pacific. And then, as I mentioned, the deanery has provided financial support for all of these activities here. And so I thank all of you for attending. This has really been a great experience for me. When I see some of these pictures, the old pictures, it's quite emotional for me. I don't know where the time has gone, but I really appreciate all of this effort that you've put in, and I'm very proud of all of you guys and gals who went on and made this a very distinguished career in your life cycle. So thank you very much. And for those of you who are going to be at the banquet tonight, I will spend more time with you because I have more stories to tell, and so there will be a little bit more of my time involved this evening. So thank you all, and hope to see you again soon. Thank you. Wow. We need a picture of this. Well, we're going to have a picture now. Yeah, Andrew, we're having a joint picture now. What's that? A picture now of the group.
And now the fun really starts. So before everybody leaves, we have a few announcements and important things. Right from here, we're going to go to take a group photograph, which, if you heard these talks, is very important. It's the only way we remember who was here and what happened. And it's going to take place on the steps leading up to the library. So we're going to go out of the auditorium as a group and then head down and try and take it there. As soon as we finish the photograph, we're going to come back this way to go into Salt, where we were for lunch, where everyone's invited to a reception. And that will be followed by dinner at 6:45. And I'm pleased to say that if anybody would like to stay for dinner to witness the talks, you're welcome to do so. We may or may not be able to feed you if you haven't pre-reserved a ticket, but you should please feel welcome to come along and at very least see the talks. As far as the reception goes, if you have a dinner icon on your badge, you should also have got by now a wristband, a gold wristband, to make you feel particularly special. This is really important because without the gold wristband, you cannot get free drinks all night. So if you have not got one of these bands, please pick one up from one of the students who'll put one on you at the back of the room before we go to the photograph. If you do not have one of these and you don't have a badge on here, you still get a free drink. But what was it, John? What was the name of that book? Money is the fuel on which the fires of global warming burns. So free drinks tickets and alcohol appear to be the fuel upon which productivity at RSMAS burns. So I couldn't leave these drink tickets over at the desk because they would just vanish in a flash. So please come and get a ticket. If you don't have one of these, please come and get a ticket from me. If you have one of these, you don't need these. Okay, and I think that's it. Once again, I want to just thank everybody for coming. I think we've got a great evening ahead of us. As I said, this is where the fun really starts, and I want to, again, thank Peter for his amazing contributions to the field, and look forward to seeing everybody tonight. So we're going to head straight to the photograph.